Well, good morning, everybody, and welcome on behalf of the staff of the Kluge Center. I'm John Haskell, the director of the Kluge Center. Our program today is Life as It Could Be, Astrobiology, Synthetic Biology, and the Future of Life. A couple things I want to mention before we get rolling. Today's program is being live streamed and filmed for placement on the library's YouTube and iTunes channel. And we're also tweeting, or as some of us like to say, twittering, but that just shows our age. Hashtag Future of Life. We would like for people to silence their cell phones and other electronic devices as they might interfere. Today the program will be led by Louis Campos, who is the Blumberg NASA Library of Congress Chair in Astrobiology, and I'll introduce him in a minute. A couple things about the Kluge Center in case you don't know what we're up to here. It was created 17 years ago due to a generous contribution from philanthropist John W. Kluge. Each year we create a scholarly community that involves about 100 researchers and residents, including senior scholars, postdoctoral fellows, and doctoral candidates from the US and other countries. They come here to use the world's largest library and to access its unique collections. We do a couple of other things, major things here at the center. One of them is to administer the Kluge Prize, which will be administered every 2 years. The next one will be in 2018, which is awarded by the librarian to recognize achievement in the study of the humanities. In addition, the Kluge Center is actively involved in informing members of Congress and their staff through congressional conversations and dinner and democracy events. Now to today's business, let me tell you a little bit about the Blumberg Chair in Astrobiology, and it's the result of a unique and rewarding collaboration between NASA and the Library of Congress. It takes its name from Nobel Prize winner Baruch Barry Blumberg, founder of the NASA Astrobiology Institute and a founding member of the Library of Congress's Scholars Council. Dr. Blumberg envisioned the creation of a chair in the Kluge Center that would focus on the humanistic and societal impacts of astrobiology. Basically, the idea was to connect scientific discoveries in the field with the best thinking in the humanities and social sciences. Placing scientific knowledge within the context of historical and philosophical thinking gives us the chance to consider questions of meaning and value that are obviously important for us as human beings. Please take note also, one thing, just a kind of advertisement that we're accepting applications for the chair, what will be the fifth chair for 2018 and 2019. Any of the past chairs can tell you, this is a fantastic opportunity to spend a year at the Kluge Center. The deadline's December 1. In fact, we have two of the former chairs sitting here with us. The inaugural chair is David Grinspoon. David, why don't you stand up for a second, everybody can see. David's at the Planetary Science Institute here in DC, and we're glad to have him here, showing a little love to the Kluge Center and the astrobiology chair. And Mr. Nathaniel Comfort is here. He's with Johns Hopkins. He was the third chair. The second chair, Stephen Dick, is on travel, or he would be here. He sent his regrets this morning. Nathaniel is with Johns Hopkins at the Department of the History of Medicine. But let's talk about Luis for a second. As I mentioned, he's the fourth chair in resident. He's on the faculty in the Department of History at the University of New Mexico. He joined the department in 2012 as its historian of science and as senior fellow with the Robert Wood Johnson Foundation Center for Health Policy. Luis's scholarship integrates archival discoveries with contemporary field work at the intersection of genetics and society. He's the author of Radium and the Secret Life, which came out through the University of Chicago Press in 2015, and he's the co-editor of Making Mutations: Objects, Practices, Contexts. He's received grants and fellowships from a number of institutions, including the Max Planck Institute for the History of Science, the National Science Foundation, the American Philosophical Society, the National Center for the Humanities, the Mellon Foundation, and the Feminist Research Institute. Before he takes the stage, we're going to hear a few words from our collaborator, Dr. Mary Wojtek, the senior scientist for astrobiology at NASA. Please help me in welcoming Mary to the stage.
So this is a wonderful event today. This is the fourth time that I've been here, and it's been my pleasure to also agree that this has been a tremendously productive collaboration between NASA and the Library of Congress. So I'm a senior scientist for astrobiology, and NASA is the agency for the federal government that's interested in understanding the origin and evolution of life on Earth so that we can actually understand how life began here, what sort of niches on our planet it explored, so what are the limits to life, in order that we can develop a strategy for searching for life beyond Earth. And so, in fact, actually one of the most exciting things for us is that this year the authorization bill for NASA included language adding to the Space Act that established the agency that put as the 10th goal for the agency would be to understand the origin, evolution, distribution, and future of life in the universe. And so all of our researchers are part of the effort, our engineers in designing instruments and missions, and now the Library of Congress is participating in this incredible endeavor. So over the past few years, we've had different chairs that have worked with the assets at the library to understand the societal impacts, historical perspectives on exploration, what does it mean to think about life both as we know it, and today we're going to hear about life as it could be, which is certainly important to us because as we actually invest in strategies for looking for life elsewhere, and now not only do we have our own solar system, but as you look at up in the night sky and every star you see is another solar system, so we have solar systems of solar systems of solar systems, so billions of planets that potentially could harbor life. And so as we consider the strategy for looking for that, we know what we know about life here on Earth, but we need to think about what life could be so that we don't necessarily get someplace and completely miss it because we've remained myopic in our search. So I think we're going to learn a lot today, both about what we know about life in the past and what life could be in the future. So thank you, Luis, for putting this together. I think this is going to be truly exciting. They just keep getting better. I don't know what else to say, but thank you.
Good morning. Welcome, everyone. I'm really glad you found your way to our symposium here today. And welcome to everyone online and to those of you watching in posterity in the future. So, I'm just delighted to have everyone here today to think about life as it could be, astrobiology, synthetic biology, and the future of life. Rarely does one get to organize a conference in the subjunctive, life as it could be, right? Life as it could have been, as it could be now, as yet undetected here on Earth, or unknown elsewhere. As it could be at some point to come, as we might make it. Life as it could be incorporates all of these possibilities. So, first, astrobiology. We've just heard from Mary about how astrobiology seeks to understand life in the universe from its origins to its future. And it's often characterized as focusing on these three fundamental questions. How does life begin and evolve? Does it exist elsewhere in the universe? And what is the future of life here on Earth and beyond? Or, as some other folks have summarized it, how did we get here? Where are we going? And are we alone, or is there anything else alive out there? By exploring the limits of life here on Earth and integrating this knowledge with what we know of space and planetary environments elsewhere, astrobiology seeks to characterize life as a general phenomenon of the universe, where the history of life on Earth is one singularly important case, but hopefully only one out of many of what we might encounter. And astrobiology brings together investigators from many different disciplines, biology, chemistry, geology, planetary science, and many others, to consider both the cosmic possibilities for life and the compelling questions of habitability. What made Earth habitable? Are there other habitable planets, or abodes for life, as we used to say, elsewhere in the universe? Yes, here we go. But might we think about life's limits in other ways too? 4 years ago, in 2013, at the first Blumberg Symposium, the suggestion was made that we might also want to think about a world in which biology proliferates, in which we have a lot of different biologies. What if what nature has provided is not the only biology possible? What if we might face, they asked, not the total depletion of biodiversity in the 21st or 22nd century, as we so often rightly fear, but find ourselves instead to be at the edge of a new Cambrian explosion of biodiversity? What if we began to think about what we could do with biology, if we thought of biology as a kind of technological platform? Could we reconstruct life to suit our purposes? These aims to engineer life and to engineer biology are at the heart of a field that today is known as synthetic biology. Most of biotechnology has yet to be imagined, let alone made true, Drew Endy said at the Synthetic Biology 7.0 meeting in Singapore just this past summer. What might it be like, he wondered, if we were able to re-implement life in a manner of our own choosing? What if we could move beyond the attributes that we inherited from the continent of natural lineages, he said. Movement through space was a useful metaphor for movement through possibility. You can imagine arriving on a new continent, he said, where we are totally unconstrained by the lineages that pre-exist in nature, and there's going to be systems in that biology, and the synthesis is going to be essentially, intellectually, to come up with the imaginative things. So instead of just imagining the world as it exists and as we inherit it from nature, he suggested, I think it's becoming increasingly important that we imagine how worlds might be and how we might choose to design and to construct them. So synthetic biology moves beyond the naturally given to engineer new forms of life based on human design and engineering in an effort to explore the full potentials of living systems, a question that in other ways astrobiologists have long had on their minds. Okay. On the more wild side, you might have read about some visions for de-extincting woolly mammoths and the possible intersections of synthetic biology with conservation. But you may also have heard about efforts to engineer microbial metabolisms for new commercial or industrial applications and to bring biological engineering solutions to bear on real-world problems. What if we could create new pharmaceuticals in the laboratory instead of having to collect them from endangered species? What if we could produce new forms of biofuels or other commodities, or boot up new organisms through genome transplant, or design minimal cells with the smallest possible number of genes? We can now synthesize DNA sequences from chemicals off the shelf, and advances in genome engineering are offering worlds of possibility. How might we best develop an industrial platform for future innovation? And I think we're going to hear about that later this morning. Any technological purpose that you could imagine being served by biology could fall within the realm of synthetic biology. And by actually engineering new organisms in the laboratory that could live in different environments, at different temperatures, or different pH or other conditions, synthetic biologists are already expanding the possibilities for life here on Earth. And someday these efforts might have an important role to play in engineering new life forms designed and adapted to live in space or on other worlds. By reconstructing inferred ancestral sequences, synthetic biologists might help us gain astrobiological insights into the environments that may have existed on the early Earth in the distant past. And by re-engineering metabolisms and redesigning hereditary systems or inventing new genetic codes, they might help us better understand how nature may have made life elsewhere, as Mary suggested this morning. With Earth as our test bed, some synthetic biologists are now beginning to ask about other possible biologies and alien biospheres, whether biochemistry as we know it is universal, or if a bestiary of alternative biochemistries are possible. Are there fundamental principles or design rules of biology? Might we develop a periodic table of biology, or do the possibilities of a biological universe exceed even the human imagination? These are compelling questions for synthetic biologists as well as for astrobiologists, who have long suspected that the life that we know here on Earth is but a subset of the vast combinatorial possibilities in the universe. Just what are the limits of life? So whether modeling the historical origin of life, searching for life elsewhere in the universe, or remaking the stuff of life itself in the laboratory, both astrobiologists and synthetic biologists, and there's a lot of overlap, are actively exploring life's outer and inner limits, looking to understand and to realize life's full potential. I've been researching this topic here over the past year, and it's been a wonderful year to be researching in the library as the Blumberg Chair, and I've discovered that there's no shortage of material, that generations of investigators over decades have been fascinated by the prospects of life as it could be. Evolution is one long sermon on the text of the infinite plasticity of living matter, wrote the celebrated naturalist Julian Huxley as early as the 1930s. And 20 years later, in 1953, he returned to the theme, and he highlighted the role of human ingenuity in evolution. He said, in broad terms, the destiny of man on Earth has been made clear by evolutionary biology. It is to be the agent of the world processes of evolution, the sole agent capable of leading it to new heights and enabling it, he said, to ever fresh realizations of new possibilities for living substance, leading life into regions of new evolutionary opportunity. So decades ago, already heralding a different sort of Anthropocene, Huxley noted that man finds himself in the unexpected position of business manager for the cosmic process of evolution. That there can be no action higher or more noble, he said, than raising the inherent possibilities of life. By the 1950s, understanding the cosmic process of evolution meant studying the origin of life for many, and that was a common touchstone for many forerunners of both astrobiology and synthetic biology. For many who were inspired by the famous Urey-Miller experiment, which zapped gases with electricity to produce amino acids, the famous attempt to study with experiment the historical origin of life on Earth, this also meant trying to understand how life might be synthesized in the test tube, and that's often how it was talked about. But by the 1960s, such claims were both commonplace but increasingly disturbing to many professional exobiologists. And the historian Jim Strick has noted that one exobiologist entering a laboratory at NASA Ames was, quote, a bit horrified to find when I first got there a secretary answering the telephone with, life synthesis. The lab was later renamed Chemical Evolution. So rather than be horrified at these deep historical linkages, I propose that we probe them and that we seek to better understand the ways in which the intertwined histories of synthetic biology and astrobiology complicate any easy distinction between discovery and invention. So, synthesizing life from scratch was a key experimental approach for investigating the potentialities and the prospects of life. And the date of the Moscow International Symposium on the Origin of Life on Earth in August 1957, noted one scientist, will probably be remembered in later years as the time when it finally became respectable in scientific circles to admit a more than ideological interest in the problem of how to make life in the laboratory. But not only were the study of the origin of life and the effort to make life in the test tube related quests at mid-century, such efforts were frequently framed within a larger cosmic context and with consideration of how life might have emerged in other planets as well. So already by mid-century, my point is, the forerunners of astrobiology and synthetic biology were conceptually joined at the hip. The construction of life by nature or by humans was of deep interest. Already by the early 60s, Joshua Lederberg, the man who coined the term exobiology, was drawing explicit connections between our illuminating the chemical mechanism of terrestrial life, he said, and the technical realization of spaceflight, saying that the challenge of planetary exploration has made us think more deeply about the general principles of earthly life. The prime questions of exobiology, he said, life beyond the Earth, concern molecular biology. Do the Martian organisms, he asked, use DNA and amino acids as we do? Or are there other solutions to the basic problem of the architecture of evolution? Within 10 years, the same sorts of questions about the nature of possible organisms anywhere were being actively debated among those whose first allegiances were to the use of synthetic methods. The age of synthesis is in its infancy, but clearly discernible, James Danieli wrote in 1972, describing how the preceding decade had brought the first synthesis of a protein, a gene, a virus, and a cell, and I think we may hear more about this later as well. Nothing, he said, with such dramatic implications has ever been seen in biology before. Evolution would be accelerated. He estimated the synthesis of all existing genes and others will soon make it possible to carry out the equivalent of 10 to the 8th to 10 to the 9th years of evolution in 1 year. Only a minute proportion of novel genomes that might be constructed can ever have existed at any time on Earth, he said, but new life synthesis techniques make it possible to explore all possible combinations of genes which are viable. So, while space might hold astronomical possibilities for life, life already held astronomical possibilities within itself. In the near future, he foresaw, we would no longer be restricted to recombinations of genes already contained within a species or derivable by mutation from those genes. And artificial cell synthesis, as it becomes available, he says all genes available in the biosphere by synthesis or mutation will be available, so the rate and the range of operation will be increased. And he foresaw new industrial futures. Industry will evolve toward the increased use of photosynthesis and biologically styled industrial synthesis, he said. Use of metals will give way to sequence-determined polymers. Food supplies and environmental control will depend upon new crop plants, innovations in forestry, and the derivation of crops from the sea. The present mechanical-style computer industry, he foresaw, may be displaced by biological-style computer design. Increased automation and control of industry by biological-style computers may accompany a disappearance of the classical need to work. The goals of society, he said, may become, on the one hand, much more decisively the experience of the inner world, of the total range of possible experience, and on the other hand, he said, the intensive exploration of the galaxy. Life synthesis, in other words, would lead to nothing less than a utopian society of leisure time with abundant resources for the thoughtful consideration and exploration of the galaxy. Once again, the futures of synthesis and of space exploration were never far apart. Danieli also theorized how we might design organisms for Mars, and he thought the process would be similar in conceptual terms to developing organisms for the treatment of sewage. The ultimate challenge, he said, was simply to learn how to take components from different organisms and put them together and get a working cell. Eventually, he said, we hope to be able to set boundaries to what components can be mixed together. So whether for practical purposes on Earth or for extraterrestrial purposes on Mars, what's clear is that even at the very dawn of recombinant DNA techniques that we now call genetic engineering, a shared vision of life as it could be was already simultaneously astrobiological and synthetic biological. It's also striking that the emerging concerns in astrobiology over back and forward contamination, what the scholar Audra Wolfe has called germs in space, and a topic that we now know as planetary protection, had a second life also in synthetic biology. The same language of contamination and containment and of evolutionary or laboratory barriers traveled across both fields, aided clearly in part by a popular new sci-fi thriller written by Michael Crichton in 1969 called The Andromeda Strain. Exobiological concerns, and this particular novel, were explicitly and repeatedly invoked in the mid-1970s by molecular biologists who had more terrestrial concerns about the potential hazards of genetically engineered forms of life newly constructed in the laboratory through the use of recombinant DNA technologies. So I think it's a remarkable fact that through the intervention of a work of fiction, exobiology became a part of the famous 1975 Asilomar Conference proposing laboratory safeguards for recombinant DNA work on Earth. Such cases suggest that we would do well to seek to understand the unexpected and sometimes unruly cultural narratives that condition and may even deeply structure the emergence of a new science. Sometimes fictions matter. They may frame public discourse about a field of science. They may prove useful as speculative tools for scientists thinking their ways through abstract possibilities. Sometimes such fictions, or sometimes we call them thought experiments, may be generative of new insights. However unruly these other cultural framings may be, they are important sites of historical investigation for anyone interested in a better understanding of the nature of the relation between science and society, and perhaps even the nature of scientific cognition itself. It's less a matter of boundary drawing and drawing lines between fiction and what some might call faction than understanding how speculative thought itself plays a key role not to be underestimated in comprehending life as it could be. But this overlap between astrobiology and synthetic biology was not limited to the potentials of new organisms. Ideas of biological design led all the way up the abstraction hierarchy, from systems of heredity and organisms to ecosystems and planets. So perhaps it's not surprising that the history of terraforming has also formed part of this earlier genealogy of the overlapping space between synthetic biology and astrobiology. Carl Sagan, after all, had envisioned what he called biological amplification to terraform Venus and envisioned successfully growing plants on Mars. The deliberate construction of habitable worlds using biology is a synthetic biological aspect of a much larger history of astrobiological approaches to habitability, and another key place where these fields overlapped. Later visionaries like Freeman Dyson, of course, have dreamt of habitable oases strewn across the solar system. Biological engineering, he said, will come to the rescue. We shall learn to grow trees on comets and redesign leaves so that seen from far away, the comet will look like a small potato sprouting an immense growth of stems and foliage. Perhaps, he said, we shall start a wave of life which will spread from comet to comet and without end until we have achieved the greening of the galaxy. Others, like Frederick Turner, have proposed visions for deploying living technology as a means of conveyance. Someday, he envisioned, our spaceships may look like inside-out trees or jellyfish. There's no reason, he said, why we'll need huge edifices made out of riveted plates of metal. If we're growing our machines and appliances ad hoc as we need them, and redissolving them when we want them out of the way, and if their shapes are customized perfectly to the task at hand and to human aesthetics, our devices will probably look like plants or animals or exquisite little works of art. Now, This is, to a first approximation, not what most astrobiologists today would consider part of astrobiology, but just what counts as astrobiology or synthetic biology is what I think is interesting here historically. The past is a foreign country, and things look a little bit differently there from how we talk about it today. These sorts of ideas look and sound downright futuristic, but in fact they have a long history, and they date back at least to the famous X-ray crystallographer J.D. Bernal, who as early as 1929 suggested that our future spaceship will get all of its nourishment in the form of energy through its outer surface. It would be forced to resemble, on the whole, he said, an enormously complicated single-celled plant. Now, Bernal was truly a visionary, and in 1929, his book, The World, the Flesh, and the Devil, it still makes for compelling reading today. He said, the stage will be reached when materials can be produced which are not merely modifications of what nature has given us, but are made to specifications of a molecular architecture. And for one place in which we can imitate nature, we will be able to improve on her in 10, and furnish the model so for organic materials with more varied properties and capable of withstanding more rigorous conditions. We should have a world of fabric materials, light and elastic, strong only for the purposes for which they are being used, a world which will imitate, he said, the balanced perfection of a living body. And then he says, after we've mastered the making of synthetic foods, we will leave the planet and make our permanent home in space. A space vessel spreading its large metallic wings, acres in extent, he said, to the full might be blown to the limit of Neptune's orbit. And when we have learned, he said, to live on solar energy and also to emancipate ourselves from the Earth's surface, the possibilities of the spread of humanity will be multiplied accordingly. And we reach cosmic vegetables by page 19. Making life itself will be only a preliminary stage, he says. Men will not be content to manufacture life, they will want to improve on it. For one material out of which nature has been forced to make life, man will have 1,000. Living and organized material will be as much at the call as metals are today, and gradually living material will come to substitute more and more. The new life would be more plastic, more directly controllable, and at the same time more variable and more permanent than that produced by the triumphant opportunism of nature, he says. Such a change would be as important as that in which life first appeared on the Earth's surface, and it might be as gradual and as imperceptible. The need to determine the desirable form of the humanly controlled universe, he suggested, is nothing more nor less than art. So our synthetic futures of life in space are as cosmic in their significance, he suggests, as the origin of life on Earth itself. Now, there are many possible links between astrobiology and synthetic biology, and as you've seen, some familiar, many not, and some more useful or entertaining than others. These particular visions go far afield in some cases from what we consider to be the proper scope or ambit of astrobiology today, but that's the beauty of history, that it brings together things that from our present perspective might seem like odd bedfellows, but which in fact have long legacies and are behind some of the ways that we structure our thought today. But whatever our human futures in space, and every envisioned future for the expansion of the human sphere of influence throughout the solar system inevitably says more about its own time and place than the future that it inhabits, what I find especially compelling about these earlier visions of life as it could be are not their fantastical dimensions, though those are fun to talk about in a talk like this, but the ways in which cultural contexts are deeply constitutive of even our most natural of natural sciences and our most basic claims about life in the universe. Take, for example, Craig Venter, who, along with sequencing the human genome, has also been busy sequencing the microbial genomes of the ocean to be used in part as a database for designing future organisms, he says, at the rate of 1 million chromosomes a day. But in addition to talking about making chromosomes here on Earth, Venter has also said that his recent projects designing minimal genomes for minimal cells, he succeeded in producing in the laboratory the smallest genome of a self-replicating organism with only 2 protein-encoding genes and 43 RNA genes, was inspired by discussions in the mid-90s about the possibility of nanobacteria found in Martian meteorites. One could observe and wonder how small life could be morphologically, he said, or one could experiment and find out genetically. So wondering about life as it could be from Mars, it turns out, was a direct inspiration for him in wondering about life as it could be down here, and not how we might find it in nature, but how we might make it to be. He's also said more recently he wants to put a DNA sequencing machine on the surface of Mars in a bid to prove the existence of extraterrestrial life and to sequence and beam back this digital DNA from the planet. Now, for a historian, that gives a new and surprising twist to the place of silicon in the tale of life as it could be. The idea that life could be based on silicon instead of carbon is an old and usually dismissed trope in the earlier history of astrobiology, and I have references to that over decades. But with the digitization of biology and the reinvigoration of this metaphor of the genetic code and its cross-fertilization with computer code, and with the rise of tremendous advances in sequencing and synthesizing, all of which enable us to equate biological systems with abstract conceptualizations of information, ideas of silicon-based life have taken on a new and surprising twist that we didn't foresee. In 2010, Venter said that he couldn't think of an organization that has more potential to use synthetic genomes in synthetic biology than NASA. But the idea that we might be taking life up into space for scientific as well as more baldly expansionistic reasons was already envisioned decades earlier. It was perfectly obvious in 1953, for instance, that living matter is a substance of very lightweight construction. Some contemporary synthetic biologists, like Harvard's George Church, have even come to view DNA itself as a kind of spaceship that could do amazing things. "We've received a great gift that biology has given to us," he said. "It's as if a master engineer parked a spacecraft in our backyard with not so many manuals, but lots of goodies in it that are self-explanatory. Why not imagine DNA as an ideal spaceship for transporting life to space, a kind of genetic Sputnik, a thread continuing from H.J. Muller's visions earlier in the century, which I'll talk about this afternoon." So Church has dreamed of launching DNA into space in a grand new vision of directed panspermia. "The time may soon come," he says, "when we need to shoot our spawn, the 6 most common elements of life, sulfur, carbon, hydrogen, phosphorus, oxygen, and nitrogen, into the void of the universe." Such a proposal for the seeding of the universe, rather than life on our planet being the result of earlier seeding, presents a new twist on Arrhenius's idea of panspermia from a century ago. This is the inversion of that idea, that it's we who should seed the universe with engineered organisms. So sometimes it seems the dreams of synthetic biology might run athwart those of astrobiology. But sending DNA to the moon was another of Church's dreams for the future of life. And having recoded all 53,000 words and 11 images in his 2012 book, Regenesis, into DNA, and made 70 billion copies of it on a chip smaller than the period at the end of a sentence, Church not only realized his dream of literary immortality, but provided proof of concept of DNA as a means of data storage with 1,000,000 times the density of current disk drives. The incredible stability of DNA as a storage medium has also drawn attention from companies interested in preserving films and recordings, with some firms even eyeing the moon as a long-term storage location. Having worked to encode an entire early silent film based on a Jules Verne story, A Trip to the Moon, 1902, into DNA, Church offers a fascinating contemporary synthetic biology vision of a genetic moonshot. He wants to literally take this DNA film, he says, and, quote, "shoot it to the moon the way Jules Verne intended." While aiming for the moon has long been a feature of Church's dreams, he's also never been afraid to shoot far beyond it. The increasing interchangeability of biological life with its digital representations, and the thought of bringing billions and billions of possible new genomes together with the vast enormity of space even led him to suggest the concept of remote printing of DNA. That, quote, "fractions of the human genome could be sent to suitable planets embedded in bacteria designed for interstellar travel, and once there, the human would be reassembled." So with apologies to Sagan, it sounds something like a digital dandelion going to seed. So some specific futures for life were envisioned 50 years ago, some others definitely weren't. And sometimes an older vision is reworked in new and surprising ways. But the question of the best relations between astrobiology and synthetic biology, even if the terms used were different, has long been a focus of concern. One 1962 National Academies report saw cosmobiology, man into space, and environmental biology as fundamentally related. While the search for extraterrestrial life came first in importance for this committee, the immense task, they said, for the biological engineer of putting man into space adequately protected came second. And the report noted that in a real sense, all three are 3 aspects of one and the same undertaking, a general extension of the scope of nature that the biologist can bring within his grasp. So history can trouble our common sense assumptions that we're perched precipitously on a frontier of innovation and nothing else. But history also never repeats itself, the novelist Julian Barnes once said. No, history just burps, and we taste again that raw onion sandwich it swallowed centuries ago. So today there is a lot of excitement around this field known as space synthetic biology, using engineered organisms in space travel as biological tools, whether for building materials or mining or pharmaceuticals or other aims. These are all exciting areas of research and they're well worth exploring. But I also hope to have suggested this morning that when we talk about the intersections between astrobiology and synthetic biology, that we not restrict ourselves to only thinking about space synthetic biology as we know it today, but that we consider a much larger realm of concepts and experiments and possibilities for life as it could be. How the astronomically large possibilities for life, of systems of heredity and their mutations, of the niches they might exist in, for example, resonated and resonate still with the literal astronomical possibilities revealed by astronomy itself. How the statistics in thinking about the one might inform and reveal the statistics of thinking about the other. How even as astronomy envisions the space of possible abodes for life, the combinatorial possibilities of synthetic life might be framed in astrobiological ways. And what we might make of the ironic fact that DNA synthesis is being heralded today as a new world full of promise, even as we no longer think of life synthesis as the appropriate descriptor or goal for either astrobiology or for synthetic biology. How do we best interpret these shifts from life as we know it to life as we grow it, or the complex relationships between plant forms and platforms? Looking at life as it could be in these broader ways might help us to find other astrobiological points of inspiration for synthetic biology, or perhaps synthetic biological inspirations behind astrobiology. As Woody Sullivan and John Barris have recently written, in some sense, consideration of how alien life might be constructed is the future of astrobiology. They and many others have envisioned what they call life with a different architecture, consisting of alternative genetic and protein alphabets, alternative metabolisms, even alternative solvents, they say, that bring us to the point where life becomes truly weird. These can be examined in the laboratory, they say, thus learning about biochemical possibilities in ways inaccessible to those who simply study and analyze Terran life. And those of you who attended last year's symposium will recall hearing about some of these fascinating possibilities from the chemist Stephen Benner. So there's doubtless many other possible connections between these two fields here waiting to be uncovered. My hope here is that today in our discussions we'll bring some of these other connections to light, and hopefully in ways that might not emerge in other sorts of venues. Two fields that might have seemed otherwise so unrelated to each other, in fact, have a longer and more fascinatingly intertwined history than we might have suspected, and even today they continue to generate shared legacies. We have a fantastic crew with us here today to help us explore these points of intersection between, as we might say, life elsewhere and life else how. I think we'll come to see there are as many different ways of thinking about this relationship as there are investigators in the arts and the sciences and the humanities. And we'll have some very different presentation styles as well, reflecting our different disciplinary identities. Historians like to read, as you've noticed, but other folks will prefer to extemporize or to perform in other ways. And I think that as we range across the cutting-edge science and the new humanistic scholarship to the productive provocations of art and design, we'll discover that these separate worlds, like astrobiology and synthetic biology, are perhaps not so distant from each other seem either. As you can see from the program, after this introductory prelude, our event today will be structured into 3 sessions, from the laboratory, from the library, and from the studio. Each of these sessions will be inaugurated by a wonderful performing duo and introduced and moderated by distinguished guests from the public square, from the worlds of science journalism and science communication. Our first session from the laboratory includes leaders in astrobiology and synthetic biology from government, from the academy, and from industry to help us get to speed on the state of the current science. Giada Arni from NASA Goddard Space Flight Center will start us off by providing the larger cosmic context for astrobiology and stoking our interest in newly discovered exoplanets and the ever-present question of habitability. One of my advisors with a penchant for bad puns once told me that there's no need to improve stellar evolution, but just how many worlds are out there and around these other stars, and how do we discover them? What can we know about them, and what do we wish we could find out for their prospects for life? Next, we'll bring things back down to Earth, and we'll think more about how we might conceive of the Earth as a planetary environment in the cosmic context, how we might go about finding new ways to detect biosignatures of life on the early Earth, which might as well be an alien planet from our perspective today. So Betul Kajar from the University of Arizona will tell us about her fascinating efforts to use synthetic techniques to reconstruct ancient protein phenotypes and to explore what she calls the very large solution space of the possibilities for life as it could be, and which may also give us insights into the determinacy or contingency of evolutionary processes. And wrapping up our morning session, Jason Kelly, the founder and head of Ginkgo Bioworks in Boston, the largest designer of synthetic DNA in the world, will present, or user, I guess, we'll hear more, will present on the industrial aims and the prospects of synthetic biology, including the astronomical rates of growth and improvements in the cost of new technologies that have transformed direct synthesis into a powerful new method for synthetic biology. And I couldn't think of a better moderator for this session than Antonio Regalado, the senior editor for biomedicine at the MIT Tech Review, who many of you have doubtless read in his column, Rewriting Life. Our second session is called From the Library, and will showcase work from scholars who analyze science from perspectives that go beyond the scientific. We often travel under the name of humanists and many other labels. You'll hear a little bit more from me about a really wonderful set of discoveries that I made in the library here this year. The anthropologist Stefan Helmreich from MIT will address the limits of life as he sees it in his ethnographic work across a variety of related fields, all concerned with life, artificial life, weird life, and life as it could be, an alien ocean of living forms, and he's written the book on the alien ocean. And our last presenter in this session will be Markus Schmidt from Biofaction in Vienna, who will explore questions of the relationship of what's been called xenobiology to astrobiology and some other really fascinating work. We're very fortunate to have as our moderator for this session Valerie Thompson, the book review editor at Science Magazine. And when we return from the afternoon break, we'll have our third and final session from the studio, where we'll turn to highlight the work of artists and designers engaged in fascinating projects at the intersections of these fields. We'll hear more about Daisy Ginsburg, Andy Gracie, and Joe Davis this afternoon, but I think you'll be amazed, and I'm just delighted our moderator for this session is the incomparable Nicola Twilley. As a former Librarian of Congress, Archibald MacLeish once wrote, "To see feelingly, only poetry can teach us." And given that astronomer James Jeans in 1943 said that astronomy is the most poetical and the most aesthetically gratifying of the sciences, we're fortunate indeed to have poetry and performance infuse our day. So Jennifer Joy and Banu Murada Mohammed, our Sci-Art performers, will be performing a brief thematic piece at the beginning of each of our sessions, bringing the beauty and the wisdom of performance and spoken word to our conversations here today. As Nabokov once wrote, the difference between the comic side of things and their cosmic side depends upon one sibilant. So, one last thought. I dwell in the past, and I think towards and with the past when I think of our current moment. I'll say more about that this afternoon, but it was a special delight when I was getting ready for today to discover in the archives that it was exactly 50 years ago, this coming weekend, that a conference was held here in Washington, D.C. on the future environment of a democracy. Now, the fate of our democracy is a perennial topic here on Capitol Hill. But what I find fascinating is that even 50 years ago, they were envisioning an event that brought together art, science, technology, and spirit when thinking about the future. So let me conclude with one final thought about how our own efforts here today share features in common with the other business of we the people here on this Hill. One might say that astrobiology and synthetic biology share a common quest to explore nature's designs and our own. Astrobiology and synthetic biology are, like politics, the arts of the possible. And with that, welcome to the Library of Congress.
When the moon slid over the sun, we three, laughing, crying, laughing, disbelieving. When the moon slid over the sun, we three, feeling the cold breeze on our skin, chilling us and waving the grass in front of us. When the moon slid over the sun, we three, laughing, crying, hugging, crying, laughing, shouting. And the moon wore a white sun halo that sent its rays reaching up and down and out. And here on Earth, a seeming dusk or dawn rimmed the horizon in every direction. When the moon slid over the sun, we three laugh, cried, and the birds were quiet, but the crowd around us, in the same awe, in the same shared, grateful, stunned awe, laugh, cried, and the children pointed and shouted, cooed and oohed and awed and hugged and jumped up and down. The day the moon slid over the sun. Thank you so much. So when Luis and I were talking about what pieces we should do, Banu and I, and then me alone, for this symposium, I shared with him this poem that I wrote Can you guess when I wrote it? It was 24 hours after. It was such an amazing experience. Those of you who saw the eclipse can relate. So Luis was saying that for him, it was an interesting moment of really knowing that you're on a planet, seeing the eclipse. And of course, since it seems so far that life seems to require a planet, we thought that would be a good beginning. My name is Jennifer Joy, and I am a writer and performer and director, and I am passionate about science. So I situate my work, all of my work, in the intersections between science and art, theater, performance. I have a podcast series on evolution. I have a performance troupe called the Sci-Art 6. We do a lot of work about the environment and environmental science. And I have some solo work that I do also. The Physics of Love is about cosmology, the scientific history of the universe. And my new piece is called The Chaos Theory of Now, which is where I'm playing with chaos theory and also complexity theory and emergence, and just looking at the situation we find ourselves in here in America today. And so for my next piece, this is the opening to that new piece, The Chaos Theory of Now. In the beginning, there was nothing. But then, something. The cosmos itself, but completely compressed. All of the possibilities in an impossibly tiny and infinitely heavy ball. And then, chaos. An infinitesimally small butterfly flapped its tiny wings inside that ball, unfurling a chain of events that resulted in our Big Bang. Had we seen that beginning, to us, it would have been chaos. But it was not chaos to itself. No, to itself, it was form and not form. It was possibility. With bosons and quarks and leptons and hadrons blinking in and out of existence. Their opposites, anti-bosons and anti-quarks, appearing to annihilate them. Parthenogenetic, our universe, gave birth to itself. A nothingness gave birth. But a nothingness containing entire universes, infinite numbers of tables of elements in its silent vastness. Its potentialities were endless, and it created universe after universe after universe, a happy artist, fat with possibility. Its potentialities were endless. The universe we live in could not possibly be the only one. No, that chaotic creativity surely invented infinite variations of form and structure, infinite ways of being a universe. We live in only one.
Thank you, Jennifer. I think I'm waiting for the chairs to be brought on. Okay. Very interesting performance. Can Can I invite our panelists up? Okay, thank you. Welcome to the first panel of today's astrobiological triptych from the laboratory, or as I like to say, from the laboratory. I am a journalist. I work at MIT Technology Review. I cover the revolution in genetic technologies. These days, mostly that's CRISPR, the new, fast, cheap, and maybe out of control way of editing genes. We also cover gene therapy, an older technology which is now coming to a hospital near you. People are being cured with one-time genetic fixes, hemophilia, immune disorders, and cancer. So for the first time, the genetically modified, they walk amongst us. That is really something new, and maybe in the future they'll walk on other worlds. We do have these worlds to visit and to populate, so I will go ahead and introduce our first speaker. She is Giada Arni. She is a astrobiologist and planetary scientist at NASA Goddard, and I think that she's working on a telescope with which we might not only hear some other civilization, but actually be able to see them or see traces of them. So let's hear about first contact.
Hi, everyone, and thank you so much for inviting me to this spectacular building. The Library of Congress is so amazing to be able to speak at today. I'm Giada Arni, a planetary scientist and astrobiologist at NASA Goddard. Let's see. Wait for my slide to come up. And also a member of the NASA Astrobiology Institute team, the Virtual Planetary Laboratory. And the purpose of this talk is to provide a cosmic context for the search for life beyond Earth. Because in many ways, life is a planetary process that can modify its environment on a global level. And some of those modifications might be detectable across interstellar distances. One of the most profound questions in the history of the human species is, are we alone in the universe? For centuries, we've pondered this question without actually being able to definitively answer it. But now, thanks to upcoming space telescopes, that might be about to change. We might actually, within our lifetimes, know that the answer to this question is no for the first time in human history. Now, when I was a kid, the only planets that we knew about in the entire universe were the planets of our solar system. But recently, that has all changed thanks to the exoplanet revolution. Exoplanets are what we call planets orbiting distant stars. The first one ever discovered was called 51 Pegasus b, and its discovery was announced in 1995, just recently. But since then, things have snowballed, and we now know of thousands of exoplanets. But even that is just beginning to scratch the surface, because we know now that most stars have planets. And what that means is that there are hundreds of billions, with a B, of planets within our galaxy. So that well will not run dry anytime soon. Those of us who study exoplanets are going to be busy for a very, very long time. So if you want to know, are we alone in the universe, step one is to find a habitable planet. How do we do that? Well, with current technology, we use two major techniques to search for exoplanets. One of those techniques is called the radial velocity method. The radial velocity method takes advantage of the fact that as a planet orbits its star, its gravity tugs on the star, and so the star wobbles a little bit as the planet is orbiting around it. We can actually measure that wobble with our telescopes today and use it to infer the presence of the planet that's doing the tugging. And from that, we can learn things about the planet's orbit as well as the mass of the planet. The second major technique that we use to learn about and discover exoplanets today is called the transit method. The transit method looks at small dips in the brightness of the star that occur when the planet passes directly in front of the star from our point of view. Now, when these periodic dimmings happen, we know that there's a planet there, and we can infer, again, things about the planet's orbit, as well as the size of the planet based on how much light it blocks out from the star. So, with these two techniques, we have found thousands of exoplanets today. But notice how these techniques are indirect techniques. You are not actually seeing the planet itself with either of these methods. Instead, you're indirectly inferring that the planet is there from how it interacts with its parent star. And so the kinds of information that we're able to obtain about these planets from these methods is inherently limited. What we really need to do if we want to understand planetary habitability, and not just potential habitability, is we have to actually see the planets directly. But this is a major challenge. How do you determine if a planet is habitable? Well, you have to see it. How do you see it? Well, unfortunately, there's a star there, and that star is 10 billion times brighter than an Earth-like planet. This is kind of like trying to see a firefly right next to a floodlight, except much, much, much harder even than that. So somehow, you have to block out that floodlight that is the star in order to see that tiny planet that's right next to it. This is a huge technological challenge. We heard some words this morning about the total solar eclipse that happened in August. And I'm curious how many people in this room actually saw the total solar eclipse? Lots of people. Who saw totality during the eclipse? So if you saw totality, it was amazing. And you also saw a part of the sun that few people have seen before because it's normally totally washed out by the glare of the star. And that's that ring of light that you see around the sun called the solar corona. The corona is the extremely hot and tenuous outer layers of the sun that reach out into space for millions of miles. Normally, you can't see the corona because the sun is overwhelmingly brighter than it. But during those magical couple of minutes during a total solar eclipse, when the moon is directly in front of the sun, blocking out the light, that allows the corona to come into view. Now, if you're a scientist who studies the sun's corona, you can't just sit there and wait for a total solar eclipse to come to you to collect data. That would be ridiculous. These things don't happen very often. And so what we do is we can build telescopes that are equipped with something called a coronagraph that does inside the telescope what the moon is doing to the sun in this image. It blocks out the sunlight so that you can see dim things that are around the sun, like the corona. We have a space telescope that does exactly this. This is a false color photograph taken with the Solar and Heliospheric Observatory, or SOHO telescope. And SOHO has a coronagraph inside of it, which is an opaque disk that sits in the optical path. And so you can point SOHO at the sun and block out the sunlight, and then that would allow you to see details that you wouldn't normally be able to see. These beautiful details you can see in the solar corona in this image here. What's also cool about this particular SOHO image is that you can see planets near the sun, some of the planets of our solar system. Now, these planets are so close to the sun in this image that if the coronagraph was not in place, you wouldn't be able to see them. They'd be totally washed out in the glare, similarly to how exoplanets are perpetually washed out in the glare of their parent stars. So what we could do to see exoplanets is we can build powerful future space telescopes equipped with sophisticated starlight suppression technologies like sophisticated coronagraphs that can do exactly what you're seeing in this picture here. So imagine pointing a future space telescope at a distant star and then nullifying that starlight so that you can see planets that are orbiting that star. And this is how we could directly see other potentially Earth-like worlds around other suns. Now, unfortunately, we don't have the technology to do this yet, but we really, really want to make these measurements. And so we're studying the designs for potential future space telescopes that might be able to enable us to do these kinds of studies. One of these potential future space telescopes that I'm part of a design study for is called the LUVOIR telescope. So LUVOIR stands for the Large Ultraviolet Optical Infrared Telescope. Astronomers love acronyms like that. They just love those kinds of acronyms. Fortunately, I take solace in the fact that if this telescope were to actually be selected and fly, it would get a different, much better name. But right now we call it LUVOIR. LUVOIR would be a very large space telescope. You can kind of think of it as the much more powerful great-grandchild to the Hubble Space Telescope. And it would be designed to be able to suppress starlight and allow us to characterize planets that are orbiting distant stars and search for signs of life in their atmospheres. Okay, so let's say we're in the future and we found some potentially Earth-like planets. The next step is we have to characterize those planets. Unfortunately, even with a mission like LUVOIR, you are not going to get a picture of an exoplanet that looks anything like this. Your picture of an exoplanet is going to look more like this. You're going to get a dot. You're going to get basically a pixel per planet, right? So you just get a point on your detector. What do you do with that? Because that doesn't look like a lot of information, right? How do you extract information from that dot? Well, fortunately, at the moment, we already know how to get some of these basic properties from planets that I've described before, things like their mass, things like their radius, their orbital period, that is how long it takes them to go around their stars, also properties of the stars themselves, like the temperature of the star, the age of the system. All of these things we already know how to learn. But when we're thinking about planetary habitability and if there's life, we really need to go farther. We want to understand what are these planets like, and those very basic properties are not satisfying when we want to get to a deeper level. What we want to know is things like, what is the atmosphere made of? Does it have liquid water, which is important, which is really central to our understanding of planetary habitability? Does it have geological activity, which is important for maintaining habitability over long geological timescales? And of course, is the planet alive? And all of that is a lot to ask from a pale blue dot. But fortunately, even though we don't have the technology to yet do this, we know how to do it. There's another dimension of information that comes along with these pale blue dots, and that's their spectra. You can shine the light from a planet through an instrument in the telescope called a spectrograph that splits up that light into a rainbow of colors. And that spectrum is incredibly valuable and information-rich. This is a cartoon spectrum of the sun. If you were to shine sunlight through a prism and look at it very, very closely, you would see these dark lines interspersed throughout the bright colors. These are called absorption lines, and they're caused by absorptions from different atoms in the atmosphere of our sun absorbing light at very particular colors. Now, we know, for example, from measurements in the laboratory that sodium, for instance, absorbs light in that sort of double line pattern that you see here in the orange-yellow part of the spectrum. So if we see that pattern in the spectrum of something out in space, whether it's a star, it's a galaxy, it's a planet, we know that there's sodium there. And so this is a science called spectroscopy, and spectroscopy is absolutely central to how we do modern astronomy. It's what allows us to measure the chemical composition of things that we'll never be able to actually reach out and touch and bring a sample of back to the laboratory. We use spectra to do this. Okay, so let's pretend you're an astronaut and you're in the distant future and you're traveling to a nearby star system, but you want to look back at Earth to check up on how our planet is doing. So you have a telescope with you and you point that telescope at Earth, but you're so far away that all you can see through that telescope is a dot. But fortunately, you are smart enough to bring a spectrograph along with you so that you can get a spectrum of the Earth as well. Well, if you're interested in planetary habitability, probably the first thing you would look for is water features in that planet's spectrum. And fortunately, Earth's spectrum is awash in water features. This is the #1 priority when we're searching for habitable planets elsewhere. We want to see, is there water? The next thing we might look for is greenhouse gases. So greenhouse gases are sort of like a blanket over our atmosphere. They trap heat and keep our planet warm. We want to understand what the temperature, what the climate of a planet is like to understand the stability of liquid water on the surface. You don't want a planet that's too hot or too cold, which is partially a function of how far away you are from your star, but it's also a function of the composition of your atmosphere. We can take a census of the greenhouse gases that are present from the planet's spectrum, and then feed that into climate models that would attempt to constrain what the surface temperature of that planet might be. We can also look for things like gases produced by geological activity. Volcanism produces gases like sulfur dioxide and compounds like sulfuric acid, volcanic ash. We could see all of these things from a planet's spectrum. And so from a spectrum, we're able to see a lot of things that are relevant and important to planetary habitability. From the spectrum, we can learn a lot about what a planet is like. But of course, understanding whether a planet is habitable is different from understanding whether it's inhabited. Detecting life on a planet is hard. It's very hard. But as I mentioned earlier, life is a planetary process. And actually, in this room, we are all taking advantage of that fact right now because we're breathing oxygen. And the oxygen in our atmosphere was put there by life. Before the evolution of oxygenic photosynthesis and the buildup of oxygen in our planet's atmosphere, we did not have breathable oxygen. If you were to go back in time to early Earth, you'd have to bring an oxygen tank with you because you wouldn't be able to breathe the air on our planet billions of years ago. Life has changed the composition of our atmosphere in a fundamental way, and this change is detectable across interstellar distances. So one of the things that we would look for on an exoplanet like the Earth is oxygen. If you were to shut off photosynthesis all at once, oxygen on our planet would disappear. You need a continuously replenishing source to have an oxygenated atmosphere. And so that's what oxygenic photosynthesis does for our planet. And in particular, most of the oxygen in our atmosphere actually comes from microbes. It comes from a class of organisms called cyanobacteria. And these cyanobacteria have oxygenated our atmosphere. And so the oxygen we see in our spectrum is sort of a way for the cyanobacteria to say hello to the rest of the universe. Another gas that we could look for that's produced by life is methane. Most of the methane on Earth is produced by biology. It's produced by microbes called methanogens that live in various places, including the guts of cows, as well as hydrothermal vents. And so these gases are what are signs of life in our planet's atmosphere, and we call them biosignatures. Really, the search for life on distant planets is the search for biosignatures. And this is what we're going to be looking for with our future telescopes. We're not even talking about intelligent life here, right? We're talking about metabolisms. We're detecting the products of specific metabolisms that occur on distant exoplanets. But what about other kinds of life? Because, there's more than one way to make a metabolism, as I'm sure some of the wonderful people on this panel will tell you. So what about novel biosignatures, which are gases that have not been well considered in the traditional biosignature framework? Well, unfortunately, we can't actually measure novel biosignatures in the atmosphere of an exoplanet yet, but we can prepare for it. Whenever we launch a future space telescope, we are inevitably surprised by what we find. Hubble surprised us. The James Webb Space Telescope, which is launching next year, super exciting, is bound to surprise us. And if we ever have a telescope that's able to directly image exoplanets, it's going to amaze us with what we found. It's going to answer questions that we didn't even think to ask. And so we're trying to prepare, and we're trying to think about what might be out there. And one way we do that is we simulate planets with computer simulations. And this is part of what I do for my own research. We try to simulate planets in the computer to understand what biosignatures might look like in the spectra of exoplanets to future telescopes. So let's say we want to do that. So first we have to assume some basic things about the planet, like its radius, its mass, its gravity. And then we have to assume some things about the star, because planets don't just sit there in isolation. They have a parent star that interacts with them. And so we assume the properties of the star. Then we have to think about how thick is your atmosphere and what is the composition of that atmosphere. And then you have to think about what biology is present on that planet. And really what I'm talking about here is what metabolisms are present on that planet. What are they eating from the environment? And then what do they then excrete back into the environment? Because some of those gases that they might produce could end up going into the atmosphere to become detectable biosignatures. But it's actually not that simple, because if we look at the planet's atmosphere in detail, there could be some interesting things going on that we could discover with our models. For example, this biologically produced gas will interact with other gases in the planet's atmosphere. Let's say there's this other gas that's represented with these red triangles. Our biological gas can interact with that gas through atmospheric chemistry and get transformed into a new kind of gas. The atmosphere of a planet is also impacted by the star. This is why it's very important to consider how the star affects planetary environments. High energy ultraviolet radiation from the star can break apart gases in the planet's atmosphere into smaller component parts. This is a process called photolysis. And through this processing of photolysis and atmospheric chemistry, what you wind up with in the planet's atmosphere in the end might not be intuitive. It might not be the gas you actually started off with being produced by the biosphere itself. And so this is why it's really, really important when considering biosignatures, and particularly novel biosignatures, that you have to think about them in the context of the whole planetary environment, because these planets are a series of interacting systems. And so if you just isolate one system from the others, you might not get the right answer. And in this case, we're building up a gas that we did not, necessarily expect to build up just from what was produced at the surface. One study that has looked at this in detail in exactly the way that I described thought about organic sulfur gases, which are gases that contain carbon and sulfur atoms. Organic sulfur gases are a relatively minor product of life on Earth, but you might imagine there could be exoplanets where this is one of the dominant things the biosphere produces. The particular organism on this slide can produce this gas, CH3SH, or methyl mercaptan. And when that methyl mercaptan enters the atmosphere, it can participate in atmospheric chemistry and photochemistry. It can get broken up by light into smaller pieces. And some of those smaller pieces, those CH3 methyl radicals, can react together to form C2H6, or ethane. So notice here that even though the biology is producing CH3SH, what's actually building up, and what's actually your biosignature here, is an excess of ethane. So this is not intuitive. You would not necessarily expect this unless you actually run these computer models. In this case, the excess of ethane is actually the most detectable biosignature of this interesting type of metabolism. Now, I performed a follow-on study to the one that I just described, and I found that these organic sulfur gases can also produce an excess of haze particles. When I say haze, what I'm talking about is something that's chemically similar to smogs that build up around industrialized cities. So think of Los Angeles and all that brown muck that happens in the air there sometimes. So these haze particles, they're similar to smog, but in this case, this is a sign of life on a planet, a sign of microbial life on a planet. This is an interesting, novel type of biosignature. But organic sulfur gases are not just the only types of novel gases that a biosphere might produce. My colleague, Nikki Peranto at NASA Ames, she grows anoxygenic photosynthesizers in the laboratory. These are photosynthetic organisms that don't produce oxygen. They produce something else. And so she's trying to figure out what that something else is. So she measures it. And we can feed then these novel potential biosignature gases into our computer models to then try to understand what they do in their environment. And then from those computer models, we can figure out what is the novel detectable biosignature that's produced. Because sometimes it's not intuitive, sometimes it's surprising. And then we can feed that forward into our designs of future space telescopes so that we can design them to be able to detect the interesting gases that we want to see in the future. Although I do think it's important to point out that sometimes things don't go as we want them to. Sometimes biologically produced gases are not detectable in a planet's spectrum, or they might get converted through atmospheric photochemistry into something that's not interesting at all. And so even though this is a sad possibility, we do have to keep it in mind. Not all biological products are going to be ultimately detectable as biosignatures. So in conclusion, what I've talked about is how large future space telescopes, like the LUVOIR mission concept that I'm involved with, may allow us to directly image exoplanets in the future. And then we'll be able to take spectra from those planets to look for biosignatures in their atmospheres. But since we can't do this yet, Currently, we're using computer modeling and laboratory studies to allow us to anticipate what kinds of novel biosignatures might be out there. Thanks.
Thank you very much, Giada. After we're done with our panelists, we will have a Q&A, so think of your questions. Our next speaker is from the place where our own worlds collide, East and West, city of Istanbul. She is now a professor, an assistant professor at the University of Arizona. She's the co-creator, the creator of an online network for astrobiology. And I think we're going to hear about the ancient enzymes that may have created our own biosignature. So.
Thank you very much for having me here. Today I want to tell you a story that comes from a molecular biology lab in order to illuminate the past so that we understand our present and also the future. And this is forward. What brings us together as astrobiologists is the question, are we alone? And what you're looking here is a person that is holding a flashlight to the cosmos, hoping that maybe he will see something. This is not what we do. But in some ways, this is similar to what we do in the lab when we study biological organisms. That biology works in so many different ways, in so many little and important ways, and we are trying to understand how biology works in general with the tools available to us. And in a way, I find this similar to what we do. Knowing how life started on our Earth, I think will allow us to learn more about biology and teach us a lot about the working processes of organisms at the molecular level. Earth's past is an alien planet. As Louis put it in the morning, that Earth is a foreign country. And Earth is a foreign planet as well. And the question is whether we can reconstruct the past biology of Earth. Some of you already may be thinking that we have this problem, that there is only one life, and that is the life that we have on our planet. So we have an n=1 problem. We need to understand what we have here. But then you may also ask, if past is a different environment, and if it's a different planet, in fact, and we know that biology adapts to its environment, can we also infer that past organisms may be different? Could they be exhibiting different functions, different molecular mechanisms that are yet waiting to be revealed? And that is what we study in the lab as synthetic biologists with interest in astrobiology. And the challenge here is to design systems in the lab where we can study and control and test and repeat and answer specific questions that will not be able to allow us gather information at the molecular level, but then connect what we learn at the molecular level to the information that we observe at the stellar scale, as Giada was explaining. How is that going to work? In one level, we have this huge stellar level data. It's so big that we cannot see it. And on the other hand, we have smaller scale data at the molecular level. It is so small, and we cannot see it with naked eye as well. So how do we connect these two different scales of information and then make sense of it, and then know that when we detect life or any signal, it is in fact a biological organism or event? In order to understand the past, we are mostly limited with what is recorded on the rocks. On the left, you're looking at, on the right, you're looking at this artistic expression of ancient life. It is very different. I wouldn't want to go there. Very dramatic, because it is a very dramatic place to be. Yet microbes lived microbes somehow found a way to survive under these conditions that would kill us. And on the right, you're looking at the rock formation and these mats that are produced by microbes themselves. Rocks are great. They provide us so much information about our past, not just the organisms, but also the environment of our planet. But they are not evolved systems in general. Rocks need to be found, and they are not here because they are naturally selected, just like biology. They are in a way here because of some certainly random events that we may not be ever able to repeat. And then we have biology that tells us a story about the past, and now we have the exact opposite problem, that life evolves, and the DNA itself is an ever-evolving and changing material itself. If you think of DNA as a tape, and I know, hopefully some of you know what tape is, unlike my students who just stare at me when I say tape. But tape itself, we can think of life as a tape, and tape itself has changed, right? We need to rewind and then listen to the story of life again. But how are we going to do that when we know that we cannot do that unless we build a time machine? But now we have some tools, like the one that's allowing you to see where you are located in the phylogenetic tree. We build these trees to understand where we stand in relation to other organisms, and these trees are built by using information, DNA and protein and genomic information. So we have this that allows us to see where we stand on this tree in relation to other organisms. And most of actually information that we gathered about the past from a biological perspective came from microbes. What you're looking here is in the form of different colors, even cocktails, are the microbes, and each color represents the environment in which these microbes were obtained. And this is a study recently published by the lab of Jan Glass at Georgia Tech, that each microbe was then, after being collected from the field, brought to the lab, and then there was this growth simulation in the tiny incubators that will simulate the environmental conditions. So in a way, how JADA simulates planets, we can simulate microbial growth conditions in the lab. And we learned so much about the environments and the evolution of these organisms using these sort of techniques in the laboratory. But then really, truly, how do we get back to the past and even all the way to the beginnings, to life's origins? And the motivation here is then, again, can we reconstruct Earth's past biology? And this is a beautiful quote that I like from Haldane. Plausible histories for any molecule must be heavily impacted by the plausible histories of other molecules that together with it formed a living organism. We cannot separate ourselves from our past. And yet we have to still work with the whole billions of years of biological recording. So when we are thinking about life today, we really are challenging with the problem of over 4 billion years of history. So you may not forget what happened last year, but for a biological organism, it is 4 billion years of history. That's a lot of history. So we will be always constrained by this past, unless we make life from nothing. So I believe this is always going to be a challenge for a molecular biologist interested in using current organisms in order to understand the past with the tools of synthetic biology. And we are in a very exciting time to answer these questions in the lab. We have a lot of tools that are provided to us that are developed to answer questions that are not related to astrobiology, but immediate needs of our society. And these are questions that are related to how to respond to an outbreak or to how to prevent a pandemic. And these solutions answer questions that probably, in some cases, would need to respond in the scale of months or perhaps years. But as astrobiologists, we are dealing with the scale of billions of years. So our time scale is different, but in some ways, I think, can use these tools that are developed for completely different reasons. And that is what I would like to discuss in the next slides with you. That at the DNA level, now we can not only look at the DNA and look at its structure, but also understand its underpinning functions. And this study was published only 5 years ago, and showing us the whole genome sequence of bacteria that lives in our gut. And now it is such a straightforward and simple and speedy method to do in our labs that it is. Even when I was preparing these slides, I was thinking to myself that I cannot believe this was only published 5 years ago, because it is such a simple and straightforward method right now. So we can look at the 4,000 genes of the simple bacteria, and not just bacteria itself, but these genomes that are as big as the human genome itself. So how can we use these tools for the questions that are interesting for astrobiologists? So we will shift the gears a little bit, and I want to talk to you about how can we use the DNA sequence of today in order to trace our way back into the past, and then engineer organisms, and then study these engineered organisms in order to understand past life and astrobiology. And majority of the tools that are given to us in order to resurrect the ancestral genes, in order to resurrect ancient proteins, comes from this field of paleogenetics. That is the ability to create ancient proteins by using phylogenetic trees that I was mentioning earlier. And you're looking at an example here that's word. And this is, in a way, represents a protein sequence. In some ways, may represent a DNA sequence. So you are looking at the ancestor of this whole 4 words that all start with the different letter with the exception of W's. So there are 3 W's and 1 G's here, so the ancestor will be a W as well. So not only we can predict the ancestor of all these 4 words by inferring each sequence using statistical methods, by also using the bifurcation points in each tree, each node, we can estimate and infer the timing of the bifurcation. So in a way, this is similar to what linguists do, that when they resurrect an ancient language, that they look at the current languages, current words, and then try to infer an ancient word, an ancient representation, and then they try to time it. It is very similar, and this is what we do in the lab to resurrect ancient sequences. And the questions then are, can we use biology in order to understand Earth history? We talked about metabolic processes. Can we resurrect ancient metabolic processes? And can we also access the universal biological features? We talk about DNA. If alien life has DNA, and if alien life also has a way to go from DNA to protein, that will make this mechanism a universal process. But how do these processes evolve in general? Is life going to be the same elsewhere in the universe? Or is life the way we have it here is a result of a fluke accident? And engineering living organisms, which I know we will hear a lot about, is another tool that I think can provide a lot of insights into resurrecting ancient behavior. And one example here is a very popular example, is CRISPR, that I represent as the scissor, with the scissor here, that you can think of CRISPR methodology as a molecular way of inserting or deleting any gene you want to any organism you want. That's the promise of the system. And it is very popular. The way we know this is that it's picked up by Hollywood. That if you have any science fiction aficionados in the room, in the previous episode of X-Files, and I'm sorry for spoiling this if you haven't watched it, but alien DNA was all that could save us, including Scully, and the methodology that was used in X-Files was CRISPR. So even Scully's genome can be engineered, presumably. And so how do we then tie these things for astrobiological questions? We heard about cyanobacteria and the importance of cyanobacteria in generating oxygen in our environment. So this cyanobacterial driven invention happened over 2 billion years ago. We have fossils that suggest that cyanobacteria existed as early as 2 billion years into the past. By changing the CO2 concentration into O2, cyanobacteria definitely impacted our past and also, I think, affected in a way that now we are here all together. And at the heart of this organism and this metabolism that we refer to as carbon fixation, there is an enzyme called rubisco. So rubisco takes carbon dioxide from the environment and then integrates carbon dioxide into this process of carbon fixation so that this metabolism leads to the production of biomass. It is, again, catalyzing the first step. It is the most important enzyme for this step and thus for the biomass production. And it also has been the topic of many synthetic biology studies. And a lot of scientists been trying, they tried and they still are working on improving the function of rubisco. But we don't know how this enzyme evolved. We don't know how old this enzyme is, even though we attribute a lot of our history and life itself to the presence of this enzyme. So in my lab, we resurrected the ancient versions of this rubisco enzyme. And we wanted to understand how this enzyme behaved prior to the rise of oxygen. And currently, we are engineering this enzyme inside cyanobacteria. We want to understand if we have a rubisco enzyme that is 3 billion years old, that will be before the rise of oxygen on our planet. So how will this ancient enzyme change the behavior of an organism that significantly impacted our environment? And I believe that one of the main contributions of synthetic biology to astrobiology will be the ability to engineer an organism that will represent an ancient heritage, an ancient state of our current biota. But when we talk about our last universal common ancestor, we no longer are in the time scale of 2 billion. You have to go 2 billion further. And now you have to work at the 4 billion scale, because that's when we think the first organism existed. So how do we then generate and resurrect an ancient organism? And I think that will be the key, and I think that will be the main contribution coming from synthetic biology. Not only design organisms with metabolisms that may represent an ancient state, but make an enzyme and make an organism that it has no prior genetic history. And I want to end by asking, is our past the new future? I believe that while studying these past organisms, past proteins, we have a chance to access different possibilities that are very alien to us, that are representing past states, but I think they will be so new and different that will be providing us a new future. Thank you very much.
Thank you, Betu Kagar. Our next presenter is Jason Kelly. He's the co-founder, CEO of Ginkgo Bioworks. Jason is sort of part of the world that I write about, the practical, the commercial use of genetic technology. And his company has really become kind of the premier one that is in evolution as a business, I would say. But he will tell us more. Jason.
Awesome. Okay, there we go. Yeah, so I want to thank Luis for organizing this. I thought the talk this morning, kind of giving all these historical references back between that connection between astrobiology and synthetic biology was shocking, actually. And so I wouldn't have actually I wouldn't have put the two together as closely as that sort of suggested, but then these couple talks here make it very clear to me. So what I'm hopeful to give you all is sort of an introduction to the synthetic biology half of that Venn diagram that Luis showed, and I'll use Ginkgo as sort of a vector to tell you what's going on, and I'll start with sort of a picture of what really is synthetic biology, which to me is really thinking of biology as something that can be designed. So I'll talk about that. And then I'll also, being the kind of token industry representative here today. I'll talk a little bit about what's happening out in the marketplace, what's actually drawing this technology out. Why can you believe that more and more investment's going to go in, and so we're going to see this technology keep improving? So I'll show you some of the kind of new and exciting things that are happening, and then a little taste of how the technology actually works. Why is this happening now and not 10 or 15 years ago? So I'll start by saying here we go. Which button is it? The right one? Can you guys hit it in the back? Or? Anything is possible. I'll give you the preview. We talk about biology as something that can be designed. That's sort of I think, kind of a shocking idea, if you just think of sort of biology that you've kind of interacted with out in the world. You don't think of it, when we think of engineering a new product, we're either writing computer code or we're building something, physical, mechanical engineering. But the idea to design biology is strange. But I'd argue it's not that weird of a concept. And you really only need to understand sort of one facet of biology to understand why that's the case. And that's that at the heart of biology is digital code in the form of DNA, right? And so, let me try it again. Oh, yeah, look at that. Here we go. So to put a little punch behind that, this is an article in Wired magazine from a couple of years ago. It says, "Move over Johnny Ive, biologists are the next rock star designers." And this is our head of design, Patrick Boyle. I really love this article, obviously. But again, it sounds shocking, but it's not that shocking, right? And we've heard today already a couple times about DNA being this sort of code that underlies organisms. I mean, that's amazing, right? So evolution over the last, we've just heard, several billion years, invented a tape, a code that's a physical thing, right? It's actually a chemical, but when you get in there, it's digital, very similar to what we ended up inventing in computers as a way to sort of encode memory and run programs, right? And biology did this on its own, and we've, over the last, really 15 years, gotten the ability to read that code, and now, just in the last really 5 years or so, gotten the ability at scale to write that code. And if you're a software developer, a software developer, if you can read and write code, you can program, right? And it's sort of a fair question, Jason, how expensive is this to do? How predictable is it? Right? All those are good questions. But is biology fundamentally designable? The answer is yes, because at the heart of it is digital code that we can read and write, right? So we know it is. And so that's really what synthetic biology builds on, that very fact. Okay? So I'm going to talk about those markets first. So what's really happened over the last several years is the cost for reading and writing that code and designing biology is falling, and it's starting to move biotechnology into a bunch of markets very different from where it used to be. So really what you've thought of with biotech to date has largely been things like pharmaceuticals, right? But this was an event on Fifth Avenue in New York last year. It's a pair of sneakers there in the middle. I'll talk about this a little bit in a minute, but it's a partnership between Adidas and a biotechnology company that's genetically engineering yeast to produce spider silk, okay? And what I love about this photo is all the people crowding around with iPhones, taking pictures, right? This is what a biotech product launch is going to look like in the future, right? It's going to look more like an Apple product event rather than a successful clinical trial, right? And that's fundamentally new. That entry of biotechnology into these consumer markets is a new thing. So I'll give you my personal experience with it. So that's me on the left there at the, if you don't know what the WPC is, that's the World Perfumery Congress. Anybody been to the World Perfumery Congress? In this crowd, it might actually get a hand. Usually it's impossible. So it's a pretty fun meeting. I'll explain the picture. So it's sort of a trade show like any other. Everyone's got a booth, and they're showing off their fragrances. But it's in a contained room. So pretty soon the whole room smells like 50 different fragrances, right? It's a disaster. And so they've invented these smell domes where you literally put your head up in there, and then they spritz out a little bit of the fragrance, so you can just smell the one fragrance. So it's a really it's an amazing industry, and not where I thought I would be when I was doing my PhD at MIT, but really been fun to learn about this industry. And one of the things I found out was, a lot of the ingredients that end up in your products are made with technologies that really haven't changed in the last 150 years. This is how we make rose oil. You like literally pick rose petals and toss them in the air and extract the rose oil. And so we looked at this as biological designers and said, well, that's fundamentally a biological process, the way that rose makes rose oil. Could we instead have a brewer's yeast, the kind of yeast you would use to make beer, put it in a brewery like this, except instead of having it make beer, have it make rose oil, okay? And the only challenge with that is, how do you make a yeast smell like a rose? And so I'll tell you how to do that, so you can leave today as sort of novice biological designers. So first thing you need to do is go look at that rose and look at its genome. Look at the code inside the rose to find the genes that encode for the ability to make that fragrance. Right? And again, this is in there just like in any other organisms, and so we do that with the technology of DNA sequencing, DNA reading. And so you can see in red, I've shown one of the genes that contributes to that rose oil. The next thing you need to do is you need to, in a computer, redesign that DNA code so it'll work in the brewer's yeast. So you're using the rose gene as a template, but you're going to make some changes so that it's a little more likely to have the yeast be able to produce this gene and do the same function. All right? But that's still not enough, because that's just in a computer, right? These A, T, C's, and G's. Remember, biology is physical. The bits, the code, in this case, is not just electrons on your processor. It's literally a piece of chemistry, right? It's a physical code. And so what do we do to get that? Well, we interact with the DNA printing industry, and we write an email like this, where we specify a sequence of letters up to about 10,000 A, T, C's, or G's. We hit send. And 4 to 6 weeks later, a tube comes in the mail from FedEx with the piece of DNA we asked for, okay? So this is a technology that really over the last 5 years has been dramatically improving. It's not as well understood as DNA reading. This is the technology of DNA writing, printing, synthesis, okay? And so I'll talk a bit more about this later, as Ginkgo is the largest designer of printed DNA in the world. About 30% of the genes printed worldwide are printed by us, okay? All right, so once you have that DNA, you now put it into the genome of the organism. You grow it up in these little teeny breweries in front of Anirudh here in this device, and you produce some rose oil. And what you're going to do then is test it. We'll pass it on to our partner. In this case, it's a French fragrance company called Robertet. They'll tell us if they like it or not. We'll make some additional edits to the genome and be able to iterate and create sort of a new and novel product there, a novel rose oil, which really hasn't seen one in the last 100-odd years. And what I'll say is, in the fragrance industry, there hasn't, the first biotech product was in 2014, so the first product made with this technology. Up until then, you'd had no biotechnology in that industry. And I'm going to talk about a few other industries like that too. So this is something that's completely being enabled by the falling costs. Now today, there's probably about 10 ingredients in the industry produced with this technology. Okay, so I'll give another example. I mentioned that shoe at the beginning. So there's a few companies that are working on making spider silk using biotechnology, right? So spider silk is very interesting, all right? So your silk today comes from silkworm silk. Spider silk threads, particularly dragline spider silk, is like really strong for its weight. So it'd be really interesting material for like performance athletic wear and things like that. So why don't we use it? The answer is, if you put 10 spiders in a box, they'll just fight until there's one spider left, right? Like you cannot farm spiders, and also a spider farm would be completely horrifying. So no spider farms, thankfully. But what the folks at these three companies, AM Silk, Spiber, and Bolt Threads, are doing is they're doing the same process we talked about with the rose. They're looking at the genome of that spider. They're finding the code that encodes for the silk. They're moving it into, in this case, brewer's yeast, and they're brewing it up, except instead of beer coming out, spider silk's coming out. All right, and the real category leader in this space is Bolt Threads out in California, and they just announced back in April their first product. It's a tie. You can see me modeling it on the right there. And this is the first biotech textile, right? And if you rewind the clock on pharmaceuticals, in 1982, human insulin was the first biotech therapeutic. Now today, half your drugs are therapeutics made with biotechnology. So if you're in the textile industry today, if you're Nike, if you're Adidas, you have to start thinking that you might be a biotechnology company, right? And so this, when biotechnology is able to enter these industries, it's pretty powerful technology. It's flexible. So I think you're seeing the very beginnings, but there could be big opportunities in these spaces. Another example where we're seeing a lot of activity right now is in the animal protein space. So up in the upper right there is a burger. It's from a company called Impossible Foods. It's a veggie burger, okay? Except it's a veggie burger that bleeds, all right? And so how does it do that? Well, what they've done is they've taken the gene for heme, which is the one of the key components in blood, okay? And they put that gene into, once again, a yeast or a bacteria that you can grow up in a big fermenter and produce essentially an animal-free blood that you can then add to your otherwise, your plant-based veggie burger and get, I've tried it, it's pretty good for a meat lover, a pretty good veggie burger. And so this is a company that's raised now about $180 million in capital in Silicon Valley and is really going after, could we replace ground beef with a true plant-based substitute that even a meat lover would like. You're seeing similar activity in the milk space, a company called Perfect Day trying to do the same thing with milk proteins, egg whites. Could we replace a lot of these key food animal proteins with a process that doesn't require animal farming, right? And with all the animal rights issues associated with that and all the environmental impacts, it's a supply chain you'd like to fix. And I think, again, biotechnology is stepping up to potentially be the solution. Okay. And one more, not to toot our horn too much here at Ginkgo, but we just announced a couple weeks ago a partnership with Bayer Crop Sciences, a $100 million partnership. And this is to work on the problem of nitrogen fertilizer. So the way that we get our nitrogen fertilizer today is there's a lot of nitrogen in the atmosphere, about 78% of the atmosphere. You pull that through a big chemical plant, you add a lot of energy and hydrogen, and out comes ammonia. And that's about 3% of worldwide natural gas goes to that process, $80 billion a year of nitrogen fertilizer. There's certain plants, like peanuts, that you don't need to fertilize. And the reason is they have microbes that live on their crops, you can see one stylized here, that live in their roots and do that same chemical reaction of taking nitrogen out of the air and making fertilizer for the plant. So you don't need to fertilize them. Trouble is we don't only eat peanuts. We actually largely eat rice and wheat and corn. And those crops do not have these similar microbes. So we have to add fertilizer to grow them at scale to feed the planet. And so the project we're doing with Bayer is, could we take the microbes that live on those big food crops or on vegetables or other places that don't have these nitrogen fixers, take the genetic code from those peanut microbes, move it in, so that over time you could have crops that were our big food crops, but we didn't need to add at least as much fertilizer. And so it's a cool project, just got started, but again, points to totally new area being opened up by the technology improving. Just to point out on the fundraising side, in 2016, about $1 billion in venture capital coming into non-pharmaceutical biotechnology applications. So about 150 of that to Ginkgo, but you're seeing increasingly investors betting on this, which means you have funding to keep driving the technology. That's sort of why I bring it up, okay? All right, so that's sort of a vignette on a few of the markets. There's other things going on too that are starting to pull this, but again, sort of not your historical biotechnology, a lot more capital coming in, a lot bigger markets available to biotech coming up. The next thing I want to talk about is how does this work, right? So why is it getting better and faster? And so the first thing I'll mention is sort of some things that have been getting developed over the last 17 years now. The first is reading DNA. And so that's this blue line here, okay? And this is the cost to sequence a human genome, going from about $100 million for that first human genome back in 2000 to, at the beginning of this year, Illumina, which is the main DNA, the company that makes the machines that read DNA, announced a machine that could do a $100 human genome coming down the pipeline. So that's a million-fold cost reduction in reading DNA. So to give you context, that dotted line at the top, that's Moore's law. That's how fast computers get year over year. Okay? So the pace of reading DNA, particularly over the last 5 years or so, it's like going from desktop computers to mobile phones in 5 years. Unbelievable. And you're starting to see all kinds of new applications for that. You heard about some this morning, using all that sequencing to resurrect extinct proteins and things like that. The amount of applications for sequencing is just scratched because it's getting good too fast, basically. We can't keep up. And then the red line is that DNA printing. Right, so that has not, back when I was in grad school, we were paying, call it $5 a letter. Today, you're paying about 3 cents a letter. So now think again, as like a computer programmer. Imagine you write a piece of code, and every 0 and 1 that goes into the computer, someone charges you $5. Okay, so it makes it a little bit difficult to program in that environment. So this is a key driver for us to be able to do more engineering of biology, is bringing down this cost to write DNA. And so it's made progress, about a 100-fold drop. Ginkgo's done a lot on that front. So sequencing, we just kind of come along for the ride. For printing DNA, we're the largest consumer of DNA in the world, printed DNA. Ordered about 600 million base pairs last year, or for this year. It's about 30% of worldwide gene synthesis. There's 2 companies that do this sort of at scale. We acquired one of them back in January, so we brought that technology in-house at Ginkgo. So we're definitely big believers in this as the technology that's enabling the design of biology. The other technology that's worth understanding is what happens after you get that tube of DNA. Now you have to take that DNA and put it into a cell, grow the cell up, and essentially debug it, test. How does it work based on the changes you made? Well, the way you do that right now is you essentially hire a bunch of people with my background, PhD in biological engineering, and you throw them at a bench, and you give them a pipette, which is the wonderful tool of the molecular biologist, and you stand there and you do by-hand lab work year over year over year. And we're replacing that at Ginkgo with essentially highly automated robotic facilities. This is our second generation facility. It's called BioWorks 2, where we take those lab protocols that a bench scientist would do and we move them all onto robotics. Okay? And so to give you a sense of what that looks like, these are lab protocols per month in the facility, going from about 50,000 back in January of 16, now close to 500,000 operations a month. And importantly, over that same period, the operations per person went from 1,000 to 7,000. So as we're building bigger facilities, it's getting cheaper and more efficient. And that's normal, right? Like that's normal manufacturing, right? And so as we're shifting from kind of that bespoke scientist working at the bench doing kind of custom lab protocols to more of an automated factory-like environment, we're starting to see what you would expect, which is bigger factories make for lower cost products and operations. And we're opening up our third one, BioWorks 3. This is an out-of-date photo because this is going to open in about 2 months. And it'll be in Boston, the rest of our facility. This is what we think the end game looks like for these types of facilities. This is Intel and Samsung's semiconductor fabs, big, highly automated facilities, I think is where this work will get done in the future. The way Ginkgo works is we essentially make this platform available to other companies. So I mentioned the deal with Bayer, but we've also worked with Cargill and ADM and Kerry Foods and Robertet and Fragrances and about 50 different organisms now that we're doing designs. So we have a design contract to build a microbe for that company with 25 different companies. So we really are the developers of the platform, and we're happy to keep working with more and more people to enable them to design biology to do new things. Okay, so I want to just touch on one last thing here at the end and tie it also to a little bit about astrobiology. So this is a photo I like to show because I give a lot of talks in Silicon Valley, and so I like to ask, what's the most complicated device on this table, right? These are some of the greatest things we've engineered as human beings, and the answer is the houseplant, right? And the answer is sort of like obviously the houseplant, right? So the houseplant, for example, is self-repairing, right? You break that leaf, it'll fix itself, right? Imagine you crack your iPhone screen and this thing repairs itself over the next day, right? We'd all be hailing, Tim Cook as a god, right? But that's a basic functionality of biology, right? It's self-assembling, right? So you plant a seed, you add air, water, and sunlight, and this thing manufactures itself with no factory around it, right? Again, a concept that's totally foreign to us. No product that we make today is manufactured by itself, okay, right? It all has to be done with a manufacturing facility. It's self-replicating, right? It makes copies of itself, right? So again, imagine you make that first iPhone, it went off and made copies, right? What does that do for margins? And then it's renewable, right? When you're done with it, you throw it on the ground and it can just diffuse into the environment, right? So this is a much more powerful manufacturing platform than any of those other things. Biology is, at a macro level, is amazing. But what about at a micro level, right? So you probably saw something like this if you did like a high school biology class, looked at some pond water. There's a little protist, and I'll draw your attention to that little hair sticking out of the back. So you probably saw these little guys swimming around. Well, how do they swim around? On the back is what's called a flagella, okay? Here's a close-up. Okay, it's basically a tail that spins, okay? Whoops. It's a tail that spins. And how does that work? Well, at the junction between the tail and the body of the microbe is that device on the far right. It's called a flagellar motor. All right, the red disc spins inside the blue disc at about 20,000 RPMs. All right? To give you a sense of scale, on the lower left, that's a electron micrograph of a current generation Intel semiconductor chip. All right, so those two fins you see there, together they're a transistor. All right, and the scale bar that you see is 40 nanometers wide. All right, same scale bar, an electron micrograph of that flagellar motor. Same size. So in the same area, right, that Intel, with a $4 billion manufacturing facility, is able to make those two dumb hunks of silicon right there, biology is assembling a 30,000 RPM rotary motor with thousands of atoms being placed in 3 dimensions for free in one of the puddles outside right now, right? So like at a molecular manufacturing level, biology is also better than our traditional manufacturing, right? So we really see this as a platform to basically make most anything. And I think today, we to me, this sort of, as this technology is improving, there's sort of this very obvious application as we sort of bear down on trying to be more of a multi-planetary species, which I think you're seeing more and more of this coming out of NASA. This is a poster from I think 3 or 4 years ago at a SynBio for space conference. And it touches on a few things, but to me, the key idea is we're spending a lot of money on developing new rocket technology, and how are we going to get there? How are we going to get to Mars? All these things, and we're going to get there, and if you tried to live there, you're just going to die, right? And the reason is, fundamentally, here on planet Earth, the core infrastructure is provided by biology, right? You heard earlier, our atmosphere is provided by biology. Our water gets cleaned by biology. Our food is produced by biology. The basic functions of this planet are biological. That's what keeps us here. And so the idea that we're going to go somewhere else and not bring biology with us if we plan to live there is absurd. And so there should be a program of equal, if not more, attention being given to the engineering of biology that's going to support our efforts if we're actually going to get to Mars, as we're putting against the sort of rocketry stuff, which is certainly fun, but at the end of the day, we got to be able to live. So tons of applications there, and happy to talk more on the panel of things you might imagine doing. And so I'll sort of end here, which is, I mentioned that at a nanoscale level, biology is better than Intel at placing atoms. But thanks to self-assembly and self-replication, biology also just makes more physical stuff than anything. Think of jungles, think of industrial agriculture. Biology just makes more stuff cheaply than any other technology. So it's better at both the small stuff and the large stuff, right? So why don't we use it to make everything, right? And that's really the goal of synthetic biology. We think we should. We think essentially we should move over to a biological manufacturing paradigm for everything. And the only missing piece really is our ability to program it, and that's getting better every year, faster and faster. And so I think we can get there. So thank you very much.
Okay, excellent talk. Thank you. We're going to have a Q&A, and we have not that much time. So do we have questions from the floor? Should we just jump straight to the questions from the floor? What?
We have lots of time.
Oh, we have lots of time. How much time do we have?
We have our microphones here as well.
Louis, how much time do we have?
Half hour.
Oh, we have a half hour. Okay, I'll relax then. I'll call on you soon. I do have my own questions. Jennifer started us with a spoken word about the beginning. It was a virgin birth in her retelling. I know another myth, Greek myth, and it's Uranus, the heavens looking down on Mother Earth and smiling upon her, and so life is transmitted that way. So I just wonder, which of you, first of all, which of you think that life on Earth came here from elsewhere? And then, as a second question, I wonder if you, Giada, if you find a planet, whether you, Betu, can design an organism that Jason can build and we can send there, and what are we waiting for? Should we, this plan to seed life in whatever form, how near term is that? So maybe start with the, are we extraterrestrials question or not? Yeah.
Well, there is this idea called panspermia, which is the idea that life on Earth came from space, came from comets, asteroids, whatever, arose somewhere in space and was brought here to Earth. I don't know that we need to invoke that to understand life on our own planet. But I do think we need to keep this in mind when we're going to other planets. For example, when we go to Mars, we are bringing stuff there from Earth that could potentially survive that journey to Mars. And so we don't want to ourselves inadvertently contaminate these other planets by accidentally bringing microbes there, bringing spores there that might be able to survive the journey in a dormant state, and then once they find some happy place on Mars, revive themselves and maybe start colonizing that planet. So this is this idea of planetary protection that we need to be careful about when we try to go elsewhere in our solar system, because we're sending things out there. So we don't want to pollute the signals that we're looking for with Earth life. If we want to find life on, say, Mars or Europa, a moon of Jupiter, we don't actually want to be accidentally detecting what we brought. Don't know if Batul will.
But wait a second, you're saying
Yeah.
so scientists can satisfy their curiosity about these pure ecosystems. You don't want to send, you would be against sending life elsewhere now, send the spores in the capsule for fear of contaminating it?
I think if our goal is to detect life that arose on Mars or arose on Europa, we have to be careful about what we're detecting in the future. So we don't want to be accidentally detecting Earth microbes if that's not our goal.
Are we extraterrestrials?
Well, we are aliens, right? We all are. We live on a planet. We are life forms living in the universe, so we are aliens. But of course, to answer your question more scientifically, I do think that understanding, I think Jada raised a very important point. I think it is important that we study whether life could be carried out on meteorites, for example, on our planet, in order to understand whether we can carry our own life to another planet the same way.
How far back can we get with the techniques you use to sort of go back to common ancestors with if imagine you had everything on the planet sequenced, just a wave a wand, you know, like was the math going to break at some point?
So it depends on if you're studying it, trying to answer this question at the gene level, it depends on the process that you're interested in. So if you're studying a universal process, and it will be as universal as is shared on our own planet, by accessing the ancestral state, you're presumably reaching out to the root, and that will be as old as life itself. So if you are resurrecting a molecule that life depends on,
I see.
that will be as old as life itself. So ribosome, for example, is a good example of this.
You can go all the way back.
You can go all the way back.
That's cool.
Batul, do you think that if, say, a meteorite from Earth hit Mars in the distant past and seeded life somewhere on Mars in the distant past, that you would be able to, first of all, recognize that life originally came from Earth, or I suppose vice versa, and also reconstruct when that event occurred?
Well, again, that will depend on the biological phenomenon that we will be able to assess. So, because all life we know is what we have here, the first test, of course, to do will be to understand whether what we found on Mars came from here. That will be the first experiment to do.
So someone showed a slide earlier of Craig Venter, who did an experiment in the desert, and he had this idea of sending the biological converter, he calls it, to Mars. It's just a gene sequencer. You could send it to Mars. It would sequence the DNA, and then you can transmit it back to a secure laboratory. So it's like kind of a biological fax machine. But if you know anything about Craig Venter, the joke is that he really wants to point it the other way, right? Take his genome and send it out to the planets. So, but the serious question is, if we do want to do technological panspermia, is it possible? Is it literally possible? Should we do it? And what are we going to be sending? Are we going to be sending a bacterium, a spore, or are we somehow sending what you said, just the information? So let me know each, let's have a game plan for actually doing it. How would we do it?
Yes, I can comment a little bit on this. I actually think it's also a way to think about the previous question you asked, which is like, did life here originate that way? So one way to ask that question is, is it impossible for us to send life to one? We know where these exoplanets are now. if we wanted to send something in that direction and put something on it that would survive the trip and when it got there would replicate, like, is that impossible or not, right? And if it's possible, then you can, assuming you assume life at least was able to start somewhere on the universe, then you could decide that that might be how everybody got around, right? If it was just easy, then maybe that's how it happened. But I bet it's hard. It's kind of cold out there and there's a lot of radiation, so you got to figure out how to get it over there. So yeah, I don't know. I mean, I'm not sure people have thought too hard about how to design it to make that trip, but it would be an interesting design problem.
Do you think that life can make the trip to an exoplanet as a living thing or as information?
I'm not sure if the distance itself would support life. It depends on how we are going to transform this actual life form on an exoplanet. But I think it is important to keep in mind that when we talk about living or bringing life to another planet, that our priority should always be to protect what we have on our own planet. And in some ways, it is a bigger challenge. So when we are trying to understand Mars or whether life would exist on Mars, I think ultimately we learn so much about what we have here and also learn lessons on how to better protect what we have so that we maintain a future for ourselves. And I think majority of the findings that come out of astrobiology research helps us understand our own planet at first place, because there's so much that we don't know about it, what we have here.
I guess maybe just a quick comment on that. I do think though that there's like establishing ourselves on another planet. I don't think there's I don't believe there's a lot of great reason to do it. I don't think it's like a great the whole backup for the Earth, all that stuff's a little bit ridiculous. But it certainly is a thing that like the human species can rally behind, right? Like it's a big project. It has a clear goal. It's, right? It's these kinds of things that they're hard to find, that could kind of unify a lot of people behind, trying to accomplish a big thing. And in that sense, I think like there is reason to do this beyond, the bang-on effects of what we learn about how we keep ourselves, fat and happy here. There can be an inspirational, unifying vision there too. So I think there's reason to send life to Mars. Like, let's get it ready. Yeah.
Okay, so more of a colonist.
Yeah, I mean, give us something to believe in.
A question right in front. Could you just say your name and your question?
I'm Nikki Twilley, and I'm moderating the third panel. And you had my piece about spider silk on the screen before, which is cool.
Didn't know you were there.
Great
piece.
So one thing I'm interested in is sort of contact and whether that might leave a biological trace, like either because we have had contact with extraterrestrial life in some form before, like life here has had that, or whether you might be able to see some sort of trace of interaction or contact. I mean, at the moment we're talking about looking for life that we haven't touched, but if lives are touching, as it were, would that leave a different sort of trace? Either in fossil or in, planetary sort of spectra, if that makes any sense?
Or in DNA. What if it was like, I don't know, like the kind of old viruses that are in the human genome and stuff?
Like in the X-Files, that to have a part of our DNA sequence that doesn't. Well, at the DNA level, it will be, I think, probably not even at the process, but at the chemical level, it's possible that that will be one way of defining that we have an extraordinary life form. That if we see that an organism that may have a different chemical in its own DNA backbone, or depends on a different process in order to process its own information. So the way I would define it would be heavily molecular, and not even at the biological level, but at the chemical level. And I think the origins of life studies is an excellent venue to answer this question, because we haven't made life from zero yet. We are, and this is also a very new field, and life had definitely more time than we had, which is a few decades. So I think when we understand how we can make life, we can then answer these questions of whether we can make different life and what are the limits of life.
Let me reframe the same question for you, Giada. In your bio, you say you hope to be alive when contact is made. But now from your talk, I know by contact, you may mean someone seeing through a telescope at great distance a biosignature on another planet. So it's not the kind of contact where the spaceship arrives and hovers over Washington.
No, not at all.
When that contact is made, I mean, what is it going to mean to scientists, and then what do you think it'll mean to society here? It's not the kind of contact that we're used to thinking about.
Well, I think it would be a profound moment, right? In the history of our whole species, we've found life on another planet. That's an amazing piece of knowledge. We've never known of another planet in the whole universe with life until we find those biosignatures that are out there. And I think that I'm hoping that this might actually happen in our lifetime because we're starting to develop the technology that would enable these discoveries to be made. I find it incredibly exciting, and I get excited about the possibility of microbes on Mars or microbes in the oceans of Europa, metabolisms that we detect on exoplanets. We don't know how common life is in the universe, right? This is this N of 1 problem that we were alluding to earlier. We have one example, and it's sitting here on our planet. So in order to actually start to begin to understand our place in the universe and how common or rare life is, we have to start getting statistically significant samples of planets so that we can start to say, okay, life might occur on some percentage of these worlds. We're common, there's lots of life out there. Or we're rare, we're really, really special here. I think that when we talk about intelligent life versus microbial life, it's interesting to look at the history of our planet, like Batul was talking about earlier. If you look at the history of Earth for a huge chunk of Earth history, for most of Earth's history, life was microbial. You'd have to go back in time with a microscope to see that there was anything going on our planet that was alive. I think just based on that, for the N of 1 problem, it's probably a safe assumption to assume that a lot of the life out there that we could detect is microbial. So that's why we're going after metabolisms, because that's something that all life produces. So it doesn't have to be intelligent to detect it. You can detect microbes across interstellar distances by looking for their metabolic byproducts.
Any other questions? Gentleman there.
Hi, David Grinspoon, Planetary Science Institute, former astrobiology chair. This is a question for Jason. You've described some exciting and very powerful new capabilities, and you've convinced us, I think, that it's getting cheaper and easier to do this kind of work, and that there's incredible promise to remake our economy and our the world we live in ways that are very hard to foresee. What could possibly go wrong? I mean, you know, the history of new technologies shows that, you know, to some degree they all have mixed legacies. There's the possibility of unintended consequences of people trying to do good things that accidentally do not so good things. And of course, there's the possibility of bad actors when, as you've described, things get cheaper and cheaper. You imagine people in their basements cooking up organisms and releasing them into the world. So I guess my question is, what keeps you up at night? What is the, what's your nightmare? What's the worst consequence you can see of this development? And then how do we prevent that?
And be as specific
Yeah,
as possible.
absolutely, absolutely. So it's an important question, you know, and it's the right question to ask. Well, I'll start a little bit with why we're sort of excited about it, and then speak to your point. You know, I think we have a current production system of how we make our stuff today that just, if you scope up 10,000 feet, looks like dig a hole in the ground, take stuff out of the ground, process it, manufacture it, throw it in the trash. And we're just like hoping the hole in the ground just never ends, you know, right? Like that's our strategy, and we're just gonna stick to it until the place burns, you know? And so, whereas biology has a manufacturing platform that we know has played well with the planet, for 3 billion years, right? So like our core belief, the reason we like sleep, I can sleep, I don't sleep a lot, but sleep enough, well enough at night, is that moving to more biological, it's kind of like, we would just like to replace our current technology with biology. We want to go in that direction, right? And we think that's going to be an inherent good because biology grew up with this place, whereas technology we developed totally orthogonally over the last 300 years and slapped it on, right? We should move back to what actually plays well. So that's the reason, okay. Now that said, obviously it's powerful technology and all your points are correct that it can be misused. I think the key thing really is the thing that keeps me up at night is like, look, like we still haven't solved pathogens for us that pop up in the wild all the time. We deal with the flu every year, right? Like the big issue is really just like the biology that interacts with humans in a pathological way. That's a lot of the other stuff. Obviously, you can always come up with scenarios, but that one to me is the one that's 1,000 times more important than all the rest to worry about. I think that the upshot is we have to deal with that anyway. Right? Like we have, there's people dying all over the world from pathogenic diseases all the time, every day. So that's a problem we need to solve. We're sort of hopeful that the kind of technologies that are going to be developed here are going to be a key part of solving that problem. But like that's the thing to pay attention to. That's the research to look after carefully. That's where this is. I think the rubber's going to meet the road. Yeah.
So I actually ordered some of my ancient genes from your company.
From Gen 9?
Yeah. And when the ancient genes arrived in the FedEx form,
yeah,
it came with a T-shirt.
Oh, nice.
Thank you very much.
And this is before we acquired them. No more T-shirts.
It was an organic T-shirt. It said, bacteria is the new black. And on the back it said, you should wear it every day. So I was intrigued by this and I wear the T-shirt.
Oh, nice.
And I then learned that the perfumes that you produce, you use bacteria.
Yeah.
So I'm not sure how I feel about, even though I wear my bacteria, and I know that you guys even made cheese out of my bacteria from the belly button, but that's another topic.
Yeah.
How should we be, or to what degree are you concerned that, back to your nightmares, the evolution of this bacteria on our skin, and then to what degree do you assess the controllability of this organism once you engineer them and then give it to humans to use as perfumes?
Yeah, so today we use brewer's yeast to make fragrances in a tank, right? It doesn't end up on a person, but I actually think that's a really good idea in general. Because not to go too far in the weeds on this off topic, but like a lot of your malodor is really caused by bacteria, right? It's like the bacteria that live in your armpit, the bacteria that live in your mouth. That's like what causes it. So there's sort of like, I think, interesting opportunities to make stuff that grows there that smells great, right? Like, or is better for you. And a lot of your skin diseases and things are also bacterially caused, right? Your acne or eczema. So I think there's a big opportunity around that stuff. I think, look, to me, biology, like fundamentally is something that we interact with. It's going to be honest, like the idea that it's going to be like a clock, that it's like, oh, we're just going to control, it's going to operate exactly. That's not the right paradigm. The right way to think about it is sort of like, I have a dog in my living room, right? It's essentially a genetically engineered wolf that plays with my children, right? And we don't expect that thing is operating like a robot, but we've developed a relationship with that biology over time that is a good and a healthy interaction. I think that's kind of going to be the framework for how we deal with the biology that we ultimately kind of put on ourselves and interact with.
We have a question in the back there, in the back row.
Sean Domical, Goldman, NASA Goddard Space Flight Center. So I want to take something the first two speakers were talking about as another potential example of a place this could go wrong. So if you listen to what Jada and Beitul were saying, we know that life has had a history of altering its environment on a global scale and the climate of the planet on a global scale. And that's borne out in both our understanding of geological history, and it's why we think we can detect bacteria, which are microbial in size, across interstellar space. So my question is, if we know that biology is really good at altering its environment, we know we have major global scale environmental problems today, that are tied inherently to our economy in terms of production of fuel, and that fuel is inherently an organic molecule. I could see the economic incentive, especially with the way that cost curve that you showed, for someone to try to invent a microbe or engineer or design a microbe that would be really good at pulling carbon from the atmosphere and turning it into fuel that we could then burn for the sake of energy to put that carbon back in the atmosphere, probably using light as the energy source to fix that carbon. The problem is, this is where the Dr. Grinspoon or David's Frankenstein monster becomes really troubling, because if that thing goes awry, it could essentially be the ultimate invasive species, and we'd have a major problem controlling how much carbon it draws down. So I guess these are the I don't worry about having too much spider silk.
May we have the question?
Yeah, sorry. So that's the question is when it gets at that scale, do we need regulation? Do we need legislation? How do we control it once it gets outside a lab scale thing and an environmental or global scale thing?
I'd maybe recast it to ask, I can get to it, but I'm kind of curious, as you think about us interacting with other planets, like we do need to send things over even to explore. How do you approach, how does it work now? Is it sort of like, I know they try to sterilize everything before you send it. Is there some step beyond that where you try to contain it to a certain region of Mars or something, right? Like, have people given thought to that at all?
For example, the Cassini spacecraft, which was recently orbiting around Saturn, was recently crashed into Saturn, both to collect data as it goes through the atmosphere, but also to avoid contaminating some of the icy moons that are around Saturn. This is, again, this planetary protection idea. You don't want to contaminate these places that might have a small chance of having extant life of their own. And I think, it is a really interesting and good point to consider how life on our planet has changed our entire planet over geological time in fundamental ways. The state of the atmosphere has changed because of life. And so I think it is interesting to consider how, as we begin to engineer these new life forms, going back to Sean's question, could they do the same to our planet or other planets?
Yeah, and I think what the scale that you work on is very different than, right? You have robots, we have 2 grad students.
Yeah.
And so I am curious to know, like, what kind of regulations or is it something that you guys also think about?
Sure.
To follow up with Sean's question.
Yeah, so the answer on the regulatory side is it's going to be like every other product that goes out there, and it's going to have to go through all kinds of, all the usual checks on that sort of thing. But I do think I would say it's unexplored, and there were a few companies back, there was sort of a heyday when oil was expensive of companies working on exactly what you're talking about. Could I have an algae that photosynthesized? And they literally went through this, exact whole process, and could we do outdoor field trials, or does it have to be in a greenhouse? And so a lot of thinking there. I think it is something that's a new idea, right? There hasn't been a product like that, and so you're going to have to ask those questions. On the other hand, again, if we just keep pumping oil and burning it, we also know it doesn't end well. So there's going to be a sort of risk trade-off there to consider. But yeah, it's a good question, and I think there will be new things, and we'll have to approach them carefully and have regulation. Yeah.
How challenging do you think it would be? So let's say we want to send humans to Mars and we want to make Mars a nice place to live. What kinds of what kinds of microbes will we bring with us? Like,
you guys tell us. I need you to tell me that, actually. You guys are the ones who understand how to, right? You're the exoplanet expert. What do you want to find on one of these things to know that it's
Your presentation had a nice Mars city. Mm-hmm. And in one of the buildings, I think, was a place for microbes to live.
Yeah.
And I think the choice of microbes for that illustration was cyanobacteria.
That's a good one.
And this is how I think engineering organisms can help, because if we can not only design organisms that can live on another planet, but also we can simulate evolution experiments here on Earth. And I know that there are various studies, especially maybe someone in the room is studying on these things. When we send microbes to the space station, we can precondition them to the conditions before we send them off.
Yeah.
So we can use the tools from evolution experiments and then teach these organisms to survive before we send them off to this journey.
Yeah.
Well, every genetically modified organism that I know of today is inferior to the real thing, right? There's always a burden of its change, and what the question was about, and what the answer leads to is, on this planet or another, we're going to create organisms that are superior at breeding or metabolism, which is a new concern. Question here.
Hi, Allison McGraw, planetary geologist, astrobiologist, National Academies of Sciences, and University of Arizona. So glad to see a U of A representative up here. And I'd like to echo Jason Kelly's thought of, it's crazy not to bring biology to Mars. And NASA's got this huge demonstration of capabilities coming up. We'd like to get into lunar orbit space and get people to Mars. And there's a large gap in the funding and being able to supply the biology that you would need there. And with NASA looking for large partnerships and divestments, do you see something like Ginkgo being a potential? Because I think we need people like yourself and these representatives on these committees and actually thinking about this, because once we get the people there, how do we keep them there, right? Yeah. And a lot of the architecture that they're releasing for this Mars timeline, getting to people by 2033, there's no afterthought. So the biology is a big afterthought, and they're looking for big partnerships. Do you see a partnership there?
I mean, yeah. The answer is yes. Would love to, both personally, but I do think biology has a lot to bring to the table. So there's obviously the big stuff, like I already mentioned earlier, like if you want to have planetary support systems, they're going to be biological, nothing else. We have no other technology that's close to self-replication and scale like biology. That's it, period. But that's going to take a while. But in the near term, you still have to, keep a bunch of people living there. And that means they need building materials, which also helps to have self-assembly to produce, right? They're going to need food, which we don't have a lot of other non-biological options, right? And they're going to need stuff that they don't know they need to bring, right? And this was this point, this kind of digital to biological converter. It's actually a really interesting idea, right? So think of why a 3D printer, right, is a useful object on Mars, because you might have forgot to bring the right size wrench or something, and it's not going to be fun to wait for the shipment, okay? And so what you can do is information can be sent quickly. The printer can then print the object you need, so you can be responsive to a problem you didn't know you were going to have, and then back on Earth, a whole team of engineers can solve your problems and just send you information, all right? So that's great. 3D printers are going to be important, and that gives you kind of big objects. But what happens when you get sick and you didn't bring the right medicine, right? Or a million and one other things that can go wrong and could use a more sophisticated solution? Well, I could also send you DNA code, okay, through, just informatically, and then you can print that DNA on Mars and put it into the genome of a microbe, brew that up, and out comes some medicine you need, right? So there's a whole other set of things you're going to need that are also going to be supported through the ability to informatically send support, right? Biology, I think, is going to be a powerful tool in that regard, also for a mission like this. But I think it's this distinction between sort of camping and settling, right? Like if we're just going there and coming back, then you just bring what you need, and but if you're going to stay, it's a lot, and I think it's got to have biology, and I think it is completely ignored right now. Yeah, we'd love to support it.
Great answer.
I'm Marcus Schmidt. I'm going to speak at the second panel. I have a question to the first speaker about detecting or trying to detecting biosignatures on exoplanets. It's a question about the epistemic kind of quality of the findings that you might produce when the telescope is up there and running. So imagine you find planets where there are signatures that look similar to what you have created in the laboratory. What do you think that life could be the source of it? Can you then say there's life there? Where is the benchmark where you can say beyond any reasonable doubt that you have found life when you only use passive analytical methods? Especially if it's such an important question. So how you can deal with it, that we can't go there and actually take a probe or do some active testing or something? How you come up with this epistemic challenge?
I love that question. And it's a question that we are grappling with right now in astrobiology, because it is going to be so hard to interpret these planetary spectra. It's going to be so exciting and so great to get these spectra, but it is going to be so hard to interpret them. I mean, really what it comes down to is that unless we send something there, or unless we get signals from intelligent civilization that's there, we're not going to be able to say with 100% certainty that there is life there, because there are non-biological processes that can mimic these biological signals. We call these biosignature false positives. There are situations where you could get an oxygenated atmosphere that doesn't involve biology whatsoever. These non-biological processes do tend to leave tells in the planet spectra that could distinguish them from biological processes, but there's a humongous parameter space out there of things that we haven't thought about yet. And so this is why we're trying to anticipate these challenges by running these computer simulations of both biosignatures and also false positives, which I didn't even touch on, but are also fundamentally important to understanding biology on exoplanets. But yeah, it's going to be hard. It's going to be really hard. We're doing our best to understand the challenges. And I think if we have enough contextual information about the planet, we could be pretty sure that there's life there, especially if we see things like maybe seasonal variations in the abundance of something that could be caused by seasonal emissions of something that might be biological. There might be a suite of biological gases that we think are present from a complicated biosphere. But I completely agree with you. It's going to be hard, but.
Just a quick follow-up question to that. If you are convinced, what is the next step? Do you then beam a radio signal directly at that planet, or do you put up a shield so they don't see us? I mean, what is the next? What is the?
They've already seen us if they want to, right? We've been sending radio signals into space, and our cyanobacteria and methanogens have been sending their own signals into space. So if you're a technological civilization looking for life elsewhere, we've been broadcasting the presence of life on our planet for billions of years before humans even got here. So I think it's kind of hopeless to try to hide the fact that we're here. And I think once we find that planet that we think is alive, we're going to try to want to study it as much as possible, because it's going to be so interesting and teach us so much.
If you were able to see, like, say, 10,000 exoplanet spectrum.
I wish. That'd be amazing.
But like, once you have, like, some of these scopes up and running, I mean, that could be the case, right? Can you derive something from like being able to look across them all? Well, then it'd be like, oh, well, these ones are like,
Oh my God, that would be amazing. So that's what we would be like doing comparative exoplanetology. Right. So we already do comparative planetology in the solar system. Comparative exoplanetology, where we're like looking at populations of planets and understanding like how different things occur on different numbers of planets with different frequencies.
Do you think it could make something look obviously alive in that case? Would something jump out?
Possibly, yeah. So people have thought about detecting like statistical signatures of life. Like maybe you can't detect it on one planet. Maybe that's too hard. But maybe if you have a ton of planets, maybe it becomes more and more unlikely that that signal is non-biological when you've detected it on like 100 different planets. Yeah. Let
me just wrap up here by turning the conversation to my favorite topic, which is germline engineering and.
That's one more.
Okay, I will give you the last question.
Okay, I'm the last one. First of all, thank you for a very interesting presentation this morning. Sorry? Oh, Victoria. I'm a nobody. I have no big letters after my name. I'm a retired school teacher, just an interested bystander. One point, if we go to Mars, please, somebody figure out how to deal with waste products, because we have not figured out how to deal with it on our own planet. And being retired, I'm a practical person, so I have a question for Ginkgo. Are you a publicly traded company? And if so, what is your symbol?
We are not, no. Still a private company, but sometime in the next, not too distant future, it'll probably happen.
Thank you.
Unless it crashes and burns.
Well, let us know when it happens, because I think after this conversation, we're all going to want to buy shares in the future of this whole concept. I think we're probably out of time, Luis. I think I can leave a closing question and not even get the answer. My favorite subject, I was saying, is perfect babies, the possibility of using these techniques in the human genome. and what are we going to turn ourselves into as a species. And so maybe at coffee or later on, we can think about how guiding the evolution of ourselves is going to somehow intersect with these other concepts, and how we might populate Mars and survive there as well. So great. Let's have a round of applause for everybody.
Welcome back, everyone. Good afternoon. We're about to do a piece for you here called I Am Life, but first I wanted to thank Luis very much for inviting us, and the Library of Congress for having us. This has been really exciting to be a performer at the Library of Congress, relatively unusual in a science humanities conference especially, so thank you. And I wanted to introduce my colleague and collaborator here, Banu Mohammed. Banu is a rapper, a vocal sound effects artist, a beatbox artist, and a performer. And we've had a lot of fun together. Originally, I wrote and we worked on this piece and the next piece we'll do for you later as a podcast. And Luis heard it, loved it, said, let's do it here. So we have staged it for you. And so this piece is called I Am Life. I am life. I am creativity. I am cooperation. I am intelligence. I am bacteria. I am fungi. I am plants. I am palm trees and potatoes. I am white clover and the honeybee pollinating it. I am silverback gorillas, spitting cobras, and house cats. I am humpback whales and horseshoe crabs. I am a peregrine falcon soaring through the air at 240 miles an hour. And an eastern chipmunk quietly burrowing into the ground. I am a cheetah racing across the Namibian savanna. And a snail ambling through your garden. I am human.
Oh, hello.
And I am the animals that humans eat. I am life. I have countless ways of being on this planet. I love my beautiful earth, my home, my partner. We move together. I have changed her rocks, her water, and more.
And she, in turn, has shaped me.
On this perfect planet, now so beautiful, I have developed cognition and consciousness.
I think, therefore I am. Cogito, ergo sum.
Which makes me want to know everything.
Everything. I want to understand my earth and all she is.
I want to understand myself, life, and all the different forms I take. Out of this desire to know, I created eyes.
Oh, look.
And even higher forms of consciousness.
Look, I said look.
Now, I explore the stars, other planets, space. That which is beyond my home, that which is the greater home, the dazzling, beckoning universe out of which both my earth home and I emerged. There is such joy.
There is such joy.
Such thrilling ecstasy.
Such thrilling ecstasy.
From knowing. And consciousness.
And consciousness. Consciousness.
I have faced many obstacles. Volcanoes that last 1 million years. Ice ages. Global warming. Asteroid impacts. I have gone through many major extinction periods, losing countless brothers and sisters. I grieve them, but I go on. To be life is to be both heartbroken and hopeful. The question at the beginning of my time remains unanswered today.
Will I make it?
Will I make it?
In the end?
In the end?
In the end? In the end?
I began when the Earth was just a few 100 million years old. A baby. A cranky baby. Oh, you wouldn't recognize that Earth. It was a tumultuous place of boiling magma, meteor showers, chemical oceans fizzing furiously in the acid rain. I don't remember much about that time, except that in certain places, I had exactly what I needed to get started. Bubbles! In warm places, carbon and hydrogen, sulfur and phosphorus and oxygen mixed and mingled, joining together, gurgling and surging until bubbles formed over and over again. For 100 million years, these bubbles arose and popped and reformed and developed. Well, some of them must have had stronger membranes. I think that's where my first cell membranes came from. Well, all life has cells, after all, and a membrane is the first requirement for a cell. What came next? I don't really know. Well, I can't really remember how I went from bubbles to full-fledged life in that wild chemical soup. Do you know? Do you remember how you went from embryo to baby? All I know is that because of bubbles, I had what I needed to get started. A membrane enclosing a space within which metabolic activities could take place. From there, I started creating.
Eureka!
First, I am a big fan of repetition.
Repetition.
I really, really like to repeat myself.
Repeat myself. Repeat myself. Repeat myself.
Once I had a design that worked, I wanted to be able to make it again.
And again!
And again!
And again!
And again!
And again!
So, I made RNA, which encoded the information of who and what I was, and downloaded it.
Download complete.
I was now able to make other cells that look like me.
A clone she is.
Which is great, because I am really, really good looking. Also, I made sure that my membrane was able to receive matter and energy that I needed, and release matter and energy that I didn't need. In other words, I invented eating and pooping. You're welcome. Lastly, I invented autopoiesis, the ability to make and remake myself. I was my own personal handyman.
I got my wrench. I got my tool belt.
Well, handy woman.
I got my Bunsen burner too, baby.
Well, handy cell.
Hello. Hello.
It took me a few 100 million years to accomplish all this. But on this tumultuous and unstable planet, I had to have a structure. That could be maintained. And it's worked. So far, I have survived. But it hasn't been easy. And it's unclear whether I'll make it in the end. I will survive. Let's take a bow.
And if I could have the panelists join me on the stage. Great. So my name is Valerie Thompson, and I am the book review editor at Science Magazine, which is the flagship journal of the American Association for the Advancement of Science. And it's my pleasure to welcome you to this afternoon session of Life as It Could Be. Whereas this morning's session kind of highlighted the perspectives of scientists and engineers who are working at the forefront of astrobiology and synthetic biology, this afternoon's session, which is entitled From the Library, will offer a humanities perspective on the future of life. So just a reminder, please hold your comments until the end of the session when we will have a short question and answer period. So without further ado, I will introduce our first speaker. Luis Campos is a historian of science at the University of New Mexico, although he's currently wrapping up his year-long residence at the Library of Congress as the Blumberg NASA Chair in Astrobiology. Dr. Campos has written widely on the history of genetics and synthetic biology. For more details, you can see the program. And for those who missed the earlier session, Dr. Campos is also the organizer of the symposium. Dr. Campos.
Hello again, everyone, and welcome back. And welcome back to these afternoon sessions where we turn from the wonderful scientific perspectives we had to other ways of considering how we might frame astrobiology. So since this session is called From the Library, I thought I'd use my time now to talk about the place of astrobiology in the library and what a habitable world this has been this year. So the first question that might come to mind is, what does astrobiology have to do with the Library of Congress? So I'd say, let's take the astronomer's advice and look up. Right? Wherever I look around this glorious palace of wisdom, there are astronomical references. The hallway that you entered through just now on the way in is decorated with the muses that preside over the arts and the sciences. And just the last one on the way in is Urania, right before you enter the room here. Out there in the atrium, there are other personifications of astronomy and of botany that are there. In his book, The Living Universe of 1933, Francis Younghusband said, "We can get planets from astronomers, but are any of them fit abodes for life? Is a single one? The question falls in between the astronomer and the biologist. Each says it's outside his province. It arises in a kind of no man's land of science. Science will, however, sooner or later have to occupy that land." By 1962, Francis Jackson and Patrick Moore wrote in Life in the Universe, that's a common title as you'll see, that it is time then to bring astronomy together on the one hand and biology and botany on the other. 1933, 1962, I was intrigued to see, of course, that the library's been doing this since 1897 at least, right? And I'd like to think that astrobiology is the space between these two fields that we see personified here, and that it's placed there in the atrium at the very heart of this library. So, what does astrobiology have to do with the Library of Congress? Or another way of phrasing that question, what might we see when we look at astrobiology with a humanistic lens, using the tools and the insights from the rich set of disciplines known as the humanities? Neil deGrasse Tyson tells a story in his autobiography that after weeks of drawing thousands of pumpkins in his studio art class, his instructor suddenly said, "No longer draw the pumpkins, draw the space between the pumpkins." Neil describes beautifully what happened next inside his head, and he concluded, "I broke free of the logic box that I did not know had contained me." So my goal for the second half of our day today is to break free from these logic boxes that we don't know contain us. I had a similar revelatory moment this past winter when I was struck by the commonalities between current artists' interpretations of some newly discovered exoplanets and the visions of an artist from a century ago that I had just seen on display at the National Gallery. So these newest of discoveries are, of course, incredibly exciting, but the historian in me wants to say that in some ways the narratives that surround these strange new worlds are enduring. Now, since I know that for many of you, history is a foreign planet, and since this is an after-lunch talk, I will spare you the arcane technical finds that I've discovered this year that are so delightful to me as a scholar. This is actually a population census of the solar system. That was a fun thing to run across. I'll spare you the wonderful little jokes that appear in all sorts of places, that there will be dragons. Remember, here be dragons on the edges of the maps. And, of course, there's E.T. there. Instead, I wanted to share some remarkable things from what I've found here in the library's collections as I've chased my footnotes, reading my way through over 1,000 books, most of which I discovered seemed to have almost identical titles. So every time I completed a shelf of books I read, I took a picture of it, and that's what you're seeing here, is what I did over the year. Here are the identical titles, and so what I ended up doing is I saved them, and I kept them on the shelf so I wouldn't accidentally order the book with the same title again. And so you can see there's quite a number here. That led me to discover that there's a 250 book limit in the library. Who knew there was a limit to the number of books you could have out? So let's be even more specific then. If cosmos means an orderly, harmonious whole, that's what it means, then the library is a vast cosmos unto itself, replete not only with books full of knowledge waiting to be gleaned, but also elevators that skip hidden floors, hidden passageways to exoshelves you never imagined existed. So even as I have lost myself in the blooming, buzzing confusion of the history of astrobiology, I found the library a really wonderful place to get physically lost in. And since this is a session entitled From the Library, I thought I would share with you a story that I began researching years ago, but which I followed up and pursued to a dramatic conclusion here this past year. So the story begins in the early days of astrobiology, in the middle of the century, when one of the foremost scientists of his generation, a visionary for the future of evolution under human control, had just won the Nobel Prize and received a letter from a young student who would one day become one of the most famous astrobiologists of all time. I'm referring, as some of you may know, to Hermann J. Muller and Carl Sagan. Astrobiology is coming of age, Sagan wrote to Muller on September 15, 1958. Sagan was 23. Muller was 68. Sagan, of course, needs no introduction to this audience, the famous and charismatic astrobiologist who inspired a generation of young people, including some of our speakers here today. And Muller, in the history of biology, is a legendarily significant and fascinating figure, and I see that we are very fortunate to have his daughter and my colleague here visiting with us today. In the early years of the 20th century, Muller worked in the Fly Laboratory at Columbia University, one of the epicenters of so-called classical genetics, and he made a number of fundamental discoveries. The field of genetics is so indebted to his work that some of his friends even joked it should be renamed Joe-ology, Hermann Joseph Muller. Now, I've written at length about Muller in my book, Radium and the Secret of Life, and I've in particular traced his use of the radioactive element radium and later X-rays to produce mutations in the hereditary substance. He succeeded beyond his wildest dreams in 1926, and he essentially inaugurated the modern study of radiation genetics. With the development and the use of the atomic bomb, his work seemed even more important and urgent. Muller's insights into the effects of ionizing radiation on the hereditary material were regularly sought out, and he received the Nobel Prize in 1946 for his work. His fame only increased in a Cold War world that was gaining new awareness of the dangers from nuclear testing. For how long would our world remain habitable, is what Muller was wondering. So it was during a celebratory lecture at Indiana University in 1947, one attended by over 4,000 people, that he publicly tied together his life's work and indicated an interest not only in the possibilities open to humanity for remaking the Earth, he said, but also we can now be sure, adventuring on other planets, the biological ones of remolding the life forms around us and in the end, even our own inner nature. So Muller was not simply an expert geneticist who sought to understand how nature had made things. He actively sought to explore what he called the wondrous potentialities of development thus disclosed in life forms, the basic possibilities of living things and the prospects of life as it could be, in other words. If we find a way to put our wishes, he said, in harmony with biological possibilities, the world of plants and animals should be increasingly ours to remold as we choose. biology held nothing but potential. At a symposium on the origin of life held at Yeshiva University in 1959, but which was really all about exobiology, Muller said, "Surely we will get to Mars within the next generation, to be conservative, if we do not have nuclear war. Surely, considering the inhospitability of the Martian conditions, we will find that life has not evolved nearly as far or become nearly as abundant or diverse as on the Earth. Will it too have its basis in some kind of genetic nucleic acids? If I could be put into a deep freeze and thereby live until that day, I would wager being dropped in boiling oil if it were not composed of nucleic acid. Beyond that, anyone would be rash to predict the chemistry, morphology, and physiology of that life on its upper levels." And he concluded, "This will be one of the most fascinating fields for the biologist, the biochemist, and geneticists of 1 or 2 generations hence, if not some of you." 1959, he's saying this. "Let us not forget here that our solar system is one speck in many billions in our galaxy, and there are billions of other galaxies." So Muller was fascinated by the biological possibilities of these billions and billions of potential abodes for life. But why stop with billions? Our own sun, he said, is not the only pebble on the beach. It is probably a very gross underestimate to say that many trillions of planets besides our own are at this moment serving as the abode of life. In many other worlds besides ours, living matter must coexist, he wrote, and in the star-bespattered abysses of the cosmos, there must surely dwell a myriad exotic forms. How much may we someday be able to learn of this life through super telescopes and other scientific telesenses without coming into direct contact with it is at present an unanswerable question. I think we heard some answers this morning. In a piece entitled "Life Forms to be Expected Elsewhere Than on Earth," Muller even wondered if our Earth in turn may be poor in life compared with what exists on some of the even more favored planets of other stars. I'd never heard of such a thought before, but talk about life as it could be. What a comparative study, he says, all this would make, and how enlightening. The outer possibilities were mirrored by inner ones for Muller. We may bear in mind that we too contain, within each one of us, he said, a veritable universe. In addition to the outer world, we have an inner world to understand and to administer, a world no less intricate and no less directly important to us all. There were new opportunities, he said, in the strange winds that are now blowing in from the atom, the gene, and outer space. We, transitional creatures, he wrote, must not shrink from our destiny or fear it, but must work in functional alliance with our genes, so that evolution will become, he said, for the first time, a conscious process. That will be the highest form of freedom that man or life can have. Life unlimited. Muller envisioned many kinds of futures. Someday, he envisioned, we will have automatic signaling systems, of which today's dial telephone is but a crude forerunner. And he asked, will the false pretensions of a few be better or less able to dominate us then, when the organic connections of all with all have become so complete? And what will become of our universities then, our legislative halls, our theaters, and our other meeting places? He envisioned that temperature regulation and ventilation will, in effect, bring us all to California when transportation does not. And he even predicted that the time would come when our machines will be more like living organisms, he said. But all of this was but a prelude. Even those, however, seem but tame beside the problem that challenges all the daring of the race, the problem of extraterrestrial projection. By this, he meant that man as a species is at the beginning of an adventure whose epic strains may ring down the corridors of the universe, he said. That man need no longer remain confined to his planet of origin, and that soon he will venture off into the cosmos, and his jobs of external creation will have begun in earnest. It is up to us to do our bit, he said, and to use what we know constructively. Our reward will be that of helping man to gain the highest freedom possible, the finding of endless worlds, both outside and inside himself, and the privilege of engaging in endless creation. Apart from reaching Mars and probing into the nature of its organisms, he said, and what would otherwise be the most exciting story in the history of the exploration of life that has ever happened to man, he said the most exciting development was what was going on right now in the laboratories of ourselves, where biochemists and geneticists, he said, are disentangling the warp and woof of which our earthly life is composed. Lecturing under the title Man's Responsibility for His Genetic Heritage, and just a month after the launch of the first Soviet satellite, Muller cautioned of the need for international cooperation, and he argued that the world cannot afford to allow individual countries their separate genetic Sputniks. I love that phrase. It was in the heart of this period of Muller's greatest thinking about the cosmos and life as it could be, the prospects for astrobiology and synthetic biology, that he encountered the young Carl Sagan. The record appears to begin around 1952, when a 17-year-old Carl Sagan wrote to Muller describing a radio program at Astra that he was hosting on the local station, WUCB Chicago. Muller replied to Sagan's questions and concluded his letter with a generous flourish. You are welcome to take anything you wish from the above and to condense or paraphrase. So a warm mentoring relationship was quickly established, and the two even went together to a science fiction congress in Chicago in 1952. 2 years later, Sagan sent Muller a science fiction story of his own, which Muller found very amusing. Not only did Sagan end up sending Muller a copy of his undergraduate thesis or a paper he'd written that year, but he even had dedicated a copy to him, saying, "To H.J. Muller, with sincere appreciation." The following year, Muller went to an American Institute of Biological Sciences meeting in East Lansing in 1955 to hear Sagan present. And that same year, Sagan followed up on a conversation and wrote to Muller to say that it is sad to think that there may be no biologists on the first expedition to Mars. Sagan, the planetary scientist and astrobiologist, and Muller, the geneticist and biological engineer, the two men talked over the course of years about the limits of life, what life forms might be and what they might be found elsewhere, and how life might adapt to new environments beyond the Earth. Mars was a common theme of Muller and Sagan's conversations. On April 26, 1956, one of them even signed off, "Take my greetings along to the Martians." The geneticist Jim Crow would later remark that if it were possible to send a man to Mars and bring him back safely and quickly, my candidate would be H.J. Muller. So, it was a special delight when at the start of my time here last October, Helen Muller drew my attention to a particular item in her father's papers. And it is, I think, a wonderful symbol of their relationship and the perfect visual encapsulation of my research project this year. It's a birthday card from Carl to Joe, and it's an image of Mars, the best available, I understand, at the time in 1955, 2 years even before Sputnik. Mars is crossed, and it may be hard to see here, but look carefully, by a red diagonal thread. And above this image, Sagan has written, "The red thread slowly weaves its way upwards." I knew that Muller often referred to the chromosomes as threads, but why red? And why was this reference so significant to the two of them? It was only last week, the morning after a marvelous performance of the Sun Ra Orchestra, which I attended with my predecessor, Nathaniel Comfort, that we began to put this story together. Nathaniel happened to have a copy of Muller's Out of the Night sitting on the kitchen table. I had read this book years ago, and the library's copy was literally sitting on my desk that weekend, but this time I picked it up. And Nathaniel's copy has the most interesting marginalia I've ever seen from the reader who bought and read this book. And within moments, flipping through, we had our answer. In that book, Muller uses a metaphor of a cord or thread to describe the history of organic evolution on Earth, where each yard stands for 10,000 years. He proceeded to describe a human generation as occupying less than 1/8 of an inch, and a cross-section of the cord the size of an aspirin tablet as representing the portion of one generation. Now this, he said, is just equal to the volume of the hereditary material which is actually contained in one generation of mankind, and which is to be passed on to the next generation. Hence, he said, our cord now acquires a further symbolic significance, and in that it may be taken as representing, in a certain real physical sense, the evolving germplasm of ourselves and our ancestors. Within this cord, he says, the fine fibers represent the chromosomes themselves, which are, in fact, filamentous bodies that intertwine, separate, and reunite in diverse ways as they pass along from generation to generation in the varying combinations resulting from sexual reproduction. At any given place, he said, there's but a single one out of all the masses of cords which has led on as finally to issue in our branch. This may be distinguished in our figurative representation by giving it a red color. It is this red cord which may be regarded as the red thread of destiny in a rather literal sense, he says. Its free end is even now being spun further, being transfigured by mutation, being twined and interwoven to give a new sort of living world dependent on its new properties. Muller's dreaming of biological possibilities, of life as it could be, spanned the astrobiological and the synthetic biological. They were one and the same project. Man could turn life to his own advantage, Muller said. He can take the red cord of life, the thread of destiny, from the hands of Clotho and spin it for himself, but not without care for the rest of life. There are more than 1 million species on this earth, he said, not counting the races and varieties of each, and every one of them is connected in devious and complicated ways with many of the other species, so that the pulling of a thread here may cause unexpected warpings and tearings of the fabric elsewhere. Each type of organism, moreover, is a veritable world within itself. So, the answer was staring me in the face all along. When we talk about the future of life, we are talking about fate. Or perhaps we now know the fates and what we might take from their hands. Now, unlike the muses that we have in the hallway here, I don't think Clotho the Fate is depicted anywhere in the library, though I'm keen to be corrected, and I'm checking with the historian of the library right now. But I love how this story shows the ways in which programmed research and happenstance serendipity, what has been called programmed serendipity, are part of the detective work of doing history and seeing how ideas are generated, shared, transformed, hybridized, or remade. But I also think this particular case illustrates the ways that a broad humanistic outlook on the world, as Muller certainly had, is part and parcel of good scientific work. His thoughts about the future of life in astrobiology and synthetic biology were deeply informed. As we think about astrobiology and how best to explore its humanistic implications, which is the intent of the Blumberg Chair, I think we might also pay careful attention to astrobiology's many upstream roots and contexts as well. It's no surprise then why Sagan would write to Muller in 1958 that astrobiology is coming of age. By the next year, Sagan reported to Muller that interest in extraterrestrial biology appears to be blossoming at last. Sagan writes, "Perhaps you were right all along and my skepticism was unfounded. The prospects are utterly beautiful. Even in places where we had always rejected the possibility of life out of hand, we may be mistaken." Sagan described his plans to complete his PhD in the next year, and that his next steps would one way or another enable him to keep his fingers, he said, still firmly in the astrobiological pie. And it's in this letter where the most remarkable passage appears, where Sagan writes about the birth of his first son in 1959, using Muller's language to describe his own deeply personal experience. "It feels strange," he writes, "adding our fiber to the red thread. I've never before had so strong a feeling of being a transitional creature at some vague intermediary position between the primeval mud and the stars." After Muller himself passed away, returning to stardust, Sagan wrote to his widow, Thea, Helen's mother, saying, "As you know, Joe played a major role in giving me enough confidence to continue this exotic quest to explore the planets for life." Sagan picked up the weaving of that thread of astrobiology and synthetic biology together still further. When I came to the library, I looked forward to reading my way through the 1,700 boxes of items in the Sagan papers. But what I didn't expect to discover here was that part of his personal library had also been preserved here. And that's when I made one of the most delightful finds of the year. There, on the shelves of some small fraction of Carl's library, was a book stamped Compliments of the Author, Muller's Studies in Genetics. I opened it up and discovered one of those treasures that only exists on paper, in real life, in a book, somewhere on a shelf, on a hidden floor, down a long passageway, in a grand building of the most august library of the land. An inscription from Muller to Sagan. "Here," he says, "is a record of some explorations into the universe inside, explorations of a type now as common as a voyage across the Atlantic. But to us, these trips were as wondrous as in its day Hanno's circumnavigation of Africa. What a triumph it will be when we can coordinate and combine our outer and inner quests." Christmas and New Year's greetings to you and yours for 1962 and 63, and may we someday meet in spirit on the tundras of Mars. Joe and Thea. So what does astrobiology have to do with the Library of Congress? For the past year, the library has been my laboratory, the place where I go to be surprised by what we think we know about the histories of our science. But I've also been surprised by what I've seen on the streets of Washington. What does it mean that at this exact moment that we are searching for and actually discovering other Earth-sized planets, potentially inhabitable zones around other stars, that some citizens are marching the streets saying there is no planet B, or that Mars is not an option? Contestations over habitability continue apace, and that was as true in Muller and Sagan's time as it is in our own. Historians of the future will no doubt have their work cut out for them in analyzing the relations between science and society in our contemporary moment. And to help diversify the ways that we think of astrobiology and the perspectives that we bring on it, 2 weeks ago I hosted a gathering of leading and emerging scholars in the humanities for extended conversations. I called it Habitable Worlds, as you can see, in the firm belief that the more ways we have of thinking about astrobiology, the better off we'll be. And that's also the belief behind our symposium today. But thinking about astrobiology in these broader ways, ways that extend beyond the science but that reflect back on it, is not just my invention. It's been a part of the field from the very beginning. And from digging around in the papers of my chair's namesake, Barry Blumberg, I was delighted to discover that this humanistic approach to astrobiology is in fact coextensive with the very existence of the Kluge Center here and its scholarly efforts. Now, some of you may have had the good fortune of knowing Barry in person. I did not, though he did receive the Nobel Prize in the year that I was born. But Barry was the founding director of the Astrobiology Institute. He was invited to serve on the Scholars Council here of the library within a year of that first astrobiology meeting. He was excited by the prospects of astrobiology, and he held that it would have the advantage of being unencumbered by hypothesis. And so astrobiology came to be built into the structure of the Kluge Center from the very beginning. And both this chair and this center were deliberately designed to help Congress better understand the importance of unfettered research generally, to bring Greece to Rome, as one founding document of the center put it. Barry was all about unfettered ideas and even the aesthetic provocations that scientific work might engender. He was in correspondence, for example, with Libby Larson, a composer from my hometown of Milwaukee, who was researching the relationship between music and transportation. In a letter, Barry proposed various, he says, episodes from the history of science and medicine that might stimulate your rich imagination. I have often thought, he wrote, of a musical opera centered around the hepatitis B virus. It is sometimes evil and sometimes benign. Mothers transmit it to their children, and it's often transferred from one partner to another, and it only has 4 genes. I even have some ideas on casting, and an aria or so. Think of the challenge of an opera, Barry wrote, with a pathological virus as the hero or heroine. Well, that may be getting too close to the edge. Talk about life as it could be, right? There are many ways that we can ask what the limits of life are, and this afternoon is all about exploring these limits. The next 2 scholars who will address us are going to speak on weird life and xenobiologies, and a bit later this afternoon we'll learn about the fascinating interventions and experiments and provocations of the wonderful panel of artists and designers we have lined up. It has been a real pleasure to be able to incorporate art and design into our symposium today. C.S. Lewis once declared that one of the functions of art is to be present and to present what the narrow and desperately practical perspectives of real life exclude. And the philosopher Ludwig Wittgenstein wrote, "The people nowadays believe that the scientists are there to teach them, and the poets, musicians, and artists, etc., to please them. That the latter might have something to teach is not something they consider." I think Barry would agree. And my hope for us this afternoon is that we might be open to being taught about astrobiology and synthetic biology in new ways, and that we might not simply celebrate but interrogate the ways that we think about these fields and their common points of intersection, to know both their legacies and their limits. Thank you.
Thank you, Luis. Our next speaker is Stefan Helmrich. Dr. Helmrich is an anthropologist at MIT, where he studies how scientists conceptualize the limits of life. So Dr. Helmrich's books have examined everything from the study of microbial life in the deep seas and oceans to scientists who simulate and synthesize life in silico, and the shifting boundaries that define seemingly straightforward concepts like life, water, and sound. Dr. Helmrich?
Thank you for that kind introduction, and thanks to Luis for inviting me and for organizing this, and to the Kluge Center and the Library of Congress for hosting this polydisciplinary conversation. I am in need of Starship control. This is how this works? Yes. Well, it has become a commonplace in today's history and philosophy and sociology and anthropology of science, and in many corners of biology, that the theoretical object of biology, life, is today in transformation. Reproductive technologies, cloning, and the politics of genomic representation torque the facts of life, which are now made in labs as well as in bodies and ecologies. Biotechnology, biodiversity, bioprospecting, biosecurity, biotransfer, and novel biopolitics draw new lines of property and protection around organisms and their elements. Cultural theorists and historians of science tell us that life itself, as a concept, was consolidated around the year 1800 when the discipline of biology was named, and life itself has now morphed as material components of living things, cells and genes, rearrange and disperse, exist in distributed laboratory choreographies that have them frozen, amplified, exchanged, and sequenced. So where is life exactly in this picture? It's somewhere suspended between the DNA and the machines, the sequences that spell out its particulars, the work that's done to reveal it. Writers in rhetoric and media studies claim that as life has gone informatic, it has become malleable, mediated, virtual, partable, multiple. Even Nature, the magazine, has gotten in on the game, editorializing in a 2007 issue that, quote, "It would be a service to more than synthetic biology if we might now be permitted to dismiss the idea that life is a precise scientific concept," unquote. I'll repeat that. The editorial in Nature says, quote, "It would be a service to more than synthetic biology if we might now be permitted to dismiss the idea that life is a precise scientific concept," unquote. So, how did we get here? And what does this mean for thinking about life as it could be, here or elsewhere? In 1802, in his disciplined dubbing book, Biologie, Gottfried Treviranus asked, "What is life?" A question that has admitted various answers. Physicist Erwin Schrödinger's 1944 "What is life?" offered that life might issue from a hereditary code script, which conception became in subsequent years enlisted into models of DNA and into informatic and cybernetic visions of vitality more generally. 51 years after Schrödinger, microbiologist Lynn Margulis and writer Dorian Sagan offered a less unitary account in their book, What is Life?, in which they delivered a distinct answer to the question for each of life's 5 kingdoms: bacteria, protists, animals, fungi, and plants, emphasizing neither some underlying logic nor an overarching metaphysics, but rather the situated particulars of microbial, fungal, plant, and animal embodiment. Life could not be compressed into the linear logic of a code, but was a process ever overcoming itself in an assortment of bodied manifestations. If Schrödinger's model fit into forms of life calibrated to Cold War practices of coding, secrecy, and cryptography, Margulis and Sagan's view speaks to a world in which environmentalism and biodiversity and their unknown futures organize contemporary forms of hope and worry. If it is now possible to think of life as having a variety of proliferating futures, as a 2007 conference at Cornell had it, it is also possible to inquire, as a 2007 conference at Berkeley did, what's left of life? This question asks whether life itself as a metaphysical object still exists. It is an extreme question, not what is life, but what was life? How has it become possible for scholars in the sciences and humanities to declare the possible end of life as a coherent concept? In my own work as an anthropologist, I've sought to answer this question by doing ethnographic fieldwork among biologists who worry very explicitly about the limits and extremes of the category of life. So in early work in this book, Silicon Second Nature, I told the tale of scientists in a field called artificial life, a field dedicated to modeling biological systems in computers. Artificial life, which saw its heyday in the 1990s, claimed that it was not just simulating life in silico, but synthesizing life in cyberspace. Remember that word? so that life might escape from its carbon instantiations. Artificial life would subsume and supersede organic life. In a next project, in the book Alien Ocean, I studied marine biologists studying microbes in extreme ecologies, like deep-sea hydrothermal vents. These scientists rewired grounding assumptions about what living systems could enact. And their encounter with extremes also pressed them up against questions of how to classify the objects of their study. And in work about astrobiology, some of which is reported in this book of essays, I tracked scientists struggling with how to define biosignatures, signs of extraterrestrial life. So I've hoped to know how the category of life is coming under stress in these limit biologies, as I call them. So in what follows, I contextualize my own ethnographic attention to extreme biology, and I suggest, as I compare my three cases, that these extreme biologies point to instabilities not only in the concept of life, but also instabilities in the concept of nature, organic, human, and earthly, and cosmic. And such instabilities, I suggest, can be mapped by attending to how scientists of extreme biologies test the limits of form in life forms. If, as Treviranus wrote in Biologie, quote, "the objects of our research will be the different forms and manifestations of life," unquote, those forms are being torqued by the object and form of biological inquiry, by changes, that is, in both the substance and the discipline of biology. So let me give you a snapshot then of these three cases, starting with artificial life. Artificial life was a field devoted to mimicking the logic of biology in computers. It was meant as a kind of rebooted theoretical biology. My 1990s fieldwork among artificial life scientists was centered at the Santa Fe Institute for the Sciences of Complexity in New Mexico, where researchers claimed that life would be, quote, "a property of the organization of matter rather than a property of matter itself," unquote. Some found this claim so persuasive that they held that life forms could exist in computers, and indeed they often compared computer viruses to biological viruses and said that they were both viral. Some hoped that the creation of such life could expand biology's purview to include not just life as we know it, but also what they called life as it could be, life as it might exist in other materials or elsewhere in the universe. Physicist founder Chris Langton summarized the ethos behind artificial life, saying that it was about, quote, "the attempt to abstract the logical form of life in different material forms," unquote. This definition of life holds that formal and material processes can be partitioned, and that what matters is form. So what was form for artificial life scientists? I'm going to say that it was two things, information and performance. So, information. Artificial life was known for its computational models of evolutionary dynamics, which were celebrated in the glorious 1990s computer graphics available at the time, like this. In systems like Tom Ray's Tierra, pictured here, assembly language programs' resident and random access memory could self-replicate based on how efficiently they made use of central processing unit time and memory space, with the idea that only the fittest bits of code could survive. So according to Tom Ray, the creator of this system, Tierra was, quote, "an instantiation of evolution by natural selection in the computational medium," unquote. So Ray understood life to be information replication, analogizing computer code and genetic code, and arguing that his computer code was a kind of genetic code with respect to this artificial universe. Ray's informatic principle, which was emergent from what he called a seed program inoculated into a computational medium, was, I think, a kind of descendant of Aristotelian form. And if you've revisited Aristotle's Generation of the Animals lately, you may recall that Aristotle named form as a kind of abstract, masculinized force impressed upon, but also transcending the material, feminized world or matrix. So in artificial life, matter became immaterial. As Langton wrote, quote, "The dynamic processes that constitute life, in whatever material bases they might occur, must share universal features, features that will allow us to recognize life by its dynamic form alone, without reference to its matter," unquote. This is a kind of extreme Platonism. The other side of form for artificial life was performance. Computer programs generated using genetic programming, in which programs are treated analogically as genetic information, were often given phenotypic representations on a computer screen so that users might make judgments about their performance. So the image I have here pictures two boxy, articulated, two kind of this model represents organisms as collections of articulated boxes that inhabit a three-dimensional world. And the image here, there's one that's kind of has the point of view of you, the viewer, and one that's sort of coming at you with these two arms. And the idea is that they're battling over possession of this little cube. And that's kind of a proxy for a selective process. So they were inhabiting this virtual three-dimensional world. And what enlivened these organisms for, I think, many of the people who looked at this in real time was a kind of pleasure in their performance. Life became, in effect, something that could be simulated. If it could be made lifelike, it became persuasive. And the lesson that scientists took from this was not that simulation reveals that there was never an original, the kind of claim that post-structuralists like Jean Baudrillard once upon a time made. They did not take the lesson that simulation reveals that there was never an original, but rather that the field of biology does not yet know about all the forms across which life might exist. Life becomes abstractable, metaphysical, something that can be ported across substrates. What is a life form in artificial life? Pure form. And the ends of life are in its endless forms. So, on the initiate's view, artificial life's extreme formalism leverages biology into the realm of universal science, like physics, with a formalism applicable anywhere in the cosmos. But, as anthropologist Marilyn Strathern saw as early as 1992 in this book, After Nature, the putatively oxymoronic artificial life suggests an undoing of the self-evidence of life as a natural kind. Artificial life signals that, as Strathern put it, many of us now live after nature, by which she meant in pursuit of nature, in the wake of nature, post-nature, all of those things all at the same time. Artificial life was supposed to take after the natural world, at the same time that it was meant to be something that would supersede and come after the natural world. But not so fast. I want to suggest that extreme is the new after. When I completed my study of artificial life, my attention was captured by research into microbes living at deep-sea vents, at extremes of temperature and pressure. Here were life forms pressing against the boundaries of what biologists believed living things capable of enacting. These creatures made their living not through photosynthesis, but through chemosynthesis, the production of organic materials using energy from chemicals such as hydrogen sulfide. Pictured here spewing out of a deep-sea hydrothermal vent. And there were many other kinds of extremophiles too. Methanotrophs use methane as an energy source. Barophiles live at pressures greater than 400 atmospheres. Halophiles withstand high salinity. Psychrophiles are cold-loving microbes with a minimum growth temperature of 0 degrees Celsius or less. If artificial life scripted life as detachable from particular substrates, the marine biology of extremophilic microbes also construed life as possessed of an as yet unmapped elasticity, but one that was always anchored in organic chemistry. So not abstract form, then, but form plastic to extreme conditions. Marine microbiologists think a lot about scale, about microbes with respect to their genes, their metabolisms, and their interaction with one another in ecologies and in global biogeochemical processes. Many of these scientists' theoretical conundrums are about how to link, as they phrase it, genomes to biomes. This is the webpage of the Center for Microbial Oceanography, Research and Education at the University of Hawaii, dedicated to linking genomes to biomes. The form in life forms changes with scale and context. Categories, genetic and metabolic, proliferate. Microbiologist Penny Chisholm has emphasized links between the genomic and the global, and giving her take on the recent sequencing of Prochlorococcus, the world's most abundant photosynthetic marine bacteria, has said, quote, "I consider this the minimal life form, having the smallest number of genes that can make life from light and only inorganic compounds. It is the essence of life," unquote. Although, after explaining Prochlorococcus's place in Earth's biological pump, she concluded by offering that this microbe should also guide biologists to think of life as something with properties similar at all scales, a system of self-stabilizing networks. Life is a hierarchy of living systems, unquote. So here, the metabolic, energetic aspect of vitality provides the lattice through which one can link genes to globe, with life becoming this kind of sliding signifier, taking you from the very, very tiny to the very, very big. And somewhere in this shuffle, even the founding biological category of the species is lost, which is particularly blurry in microbes anyway because of lateral gene transfer. Gene transfer interrupts what Darwin called the natural classification that would follow from following lines of genealogical descent, which he offered in the final chart, or the only chart, in Origin of Species, this very famous structure for thinking about life as arrayed on a kind of great tree. In a microbiological idiom in which we pay attention to lateral gene transfer, in other words, not just lines that are kind of cascading, in this case, up from the bottom of the root to the top, the tree of life becomes more like a web or a bush or a rhizome. So in a microbiological restaging of those personalized family genetic history algorithms that are advertised for people curious about whether they have a haplotype that links them to a lineage of biblical priests, species, like the pure racial type, falls apart under the genetic informatic lens. Nature no longer grounds a linear classification. It becomes very difficult to make a claim that there is such a thing as a natural classification just given by nature. So what are life forms here? They're abstractions, even as most microbiologists would also claim, contra artificial life, that such forms cannot be abstracted from living things. Life becomes a plastic property that scales from gene to globe. So what kind of extreme have I detected here? Something like extreme materialist relativism. Well, the word extremophile, lover of extremes, coined in 1974 as a scientific-sounding hybrid of Latin and Greek, has usually been taken to refer to microbial life forms. Biologists will point out that the term can apply to metazoans as well, and more, that extremophily is a relative term. Humans might be imagined as aerophiles, air lovers, in extreme from the vantage point of anaerobes. The extreme functions as a relativist rather than totalizing operator. What this accomplishes is attention to environment. The ends of this kind of biology are all about ecological context and about displacing humans as the only ends of evolution. As I discuss in Alien Ocean, such a view means to awaken humans to their ultimate superfluity to earthly life writ large, and therefore to their responsibility for the effects of their anthropocentrism. Astrobiology. So I'm riveted by the work of analogy and metaphor in astrobiology. So think about analogy. Astrobiologists spend a lot of time in wet labs experimenting with extremophiles as extraterrestrial proxies or analogs, or time in the field thinking about contemporary earthly ecologies as possibly analogous to ecologies of the early Earth or ecologies on other planets. And think, too, about the metaphor of the biosignature, about which we heard such fascinating things this morning. Defined, and I'm going to take an older definition, defined by McKay, Clement, Thomas Keptra, and Gibson in 2002 as, quote, any measurable property of a planetary object, its atmosphere, its oceans, its geologic formations, or its samples that suggests that life was or is present. A short definition is a fingerprint of life, unquote. There are both direct and remote signatures of extraterrestrial life. Direct signatures might be measurements that show evidence, in rock samples, for example, of the production of organic molecules. Remote signatures include such items as the spectral signature of the atmosphere, which can point toward such bioproducts as ozone or methane. Astrobiologists often zero in on the spectral trace of water as an indication of the possibility of vitality. And a founding challenge presents itself here, as astrobiologists know better than anyone, and that is that, according to astrobiologist David Desmarais, that, quote, our definitions are based upon life on Earth, and accordingly, we must distinguish between attributes of life that are truly universal versus those that solely reflect the particular history of our own biosphere, unquote. And this is no simple task, since knowing what is universal is precisely what is to be discovered. If the search for extraterrestrial intelligence, or SETI, sought signals in an ocean of noise, looking for the arbitrary and organized surprise, what scientists call information, astrobiology searches in a less structuralist mode, scouting for what semiotician Charles Sanders Peirce called indices, indirect representations, traces of its object, life. What are life forms? Things that leave traces of their form. And in my conversations with scientists in astrobiology, in many instances, institutionally enough of a limit biology that most researchers in the field remain based in traditional disciplines like microbiology and geochemistry, the question of what could count as a proper trace was a live one, and several scientists referred me, by way of highlighting the complexities here, to this historically controversial image, which is an electron microscope image of the inside of a Martian meteorite discovered in Antarctica in 1984. Mars meteorite ALH 84001, which had been blasted off Mars 16 million years previous by the impact of another meteorite and had arrived at Earth 13,000 years ago, harbored elliptical shapes that some believe to be the outlines of ancient alien microbial life. David McKay and colleagues, in a 1996 Science article entitled, quote, Search for Past Life on Mars, Possible Relic Biogenic Activity in Martian Meteorite ALH 84001, unquote, wrote, quote, ovoid features are similar in size and shape to nanobacteria in travertine and limestone. The elongate forms resemble some forms of fossilized filamentous bacteria in the terrestrial fossil record, unquote. They concluded that they had found, quote, evidence for primitive life on early Mars, unquote, with an age of about 1.3 to 3.6 billion years. Their evidence, arrived at through the amplification of electron microscopy, offered a similitude that ended up leaving the object quite open to criticism. Other astrobiologists balked at the pattern-matching recipes for seeing traces of life in this image. Still, advocates' arguments went beyond visual pattern matching. There were also traces of organic carbon compounds, though either biotic or abiotic processes could have produced these, as well as magnetite and sulfides similar to those made by earthly bacteria. Objections persisted. Opponents pointed out that traces on ALH 84001 indicated formations 2 orders of magnitude smaller than any microbe, too cramped to fit within their boundaries all of the apparatus that cells need to function. Supporters of life signs in ALH 84001 countered that these were not microbes but nanobes, a reframing that recognizes a limit but then leaps over it by conjuring a new category for debate. And so a constant repositioning of what life could be and the form it might take is, it seems to me, a kind of constant companion to astrobiological discourse. In a 2007 report issued by the United States National Research Council's Committee on the Limits of Organic Life in Planetary Systems, biologists interested in life on other planets speculate that living systems might employ ammonia, sulfuric acid, and methane as a solvent in the way life on Earth uses water. Such claims open up the possibility of looking in as yet unexamined locales for life, such as the methane lakes of Saturn's moon Titan. Such a framing poses life as at once materially anchored and capable of taking a variety of forms not yet known. Indeed, the opening inscription of the 2007 report reads, dedicated to non-human-like life forms, wherever they are. Leaving aside the fact that non-human-like might better read unknown or extraterrestrial, the statement is, I think, an expression of faith in form, and in its way, life. So form is a kind of hybrid of the empirical and the conceptual, a hybrid of the real, or a hybrid of reality and the formalisms that we use to apprehend it. So what are life forms for astrobiology then? They are things that leave traces of their form. And I'll just do a brief riff on Jacques Derrida, the post-structuralist who theorized the signature as a sign of presence that is manifested by an absence, namely the absence of the person who signed it, right? Like when you write a check, it means that you were there, but you're not there when it's cashed. But the absence of the signer, of course, brings up all these questions of authenticity and, of course, forgery, right, or false positives. So what are life forms for astrobiology then? Things that leave traces of their form. And to be sure, astrobiologists want to be awake to the possibility that they do not yet know what all of these forms could be. Astrobiologists hold that there are many logics from which to reason about extraterrestrial life, from Earth as one planet among others, from organic chemistry, from optics. As Baruch Bloomberg suggested, quote, life has the characteristic, using philosophical terminology, of being and becoming. It exists in a particular form now, but has the potential, because of the diversity in its offspring, of becoming something related, but also different, unquote. Within this awareness, phrased though it is in terms of kinship and inheritance and descent, is a sense that astrobiology depends on what Charles Sanders Peirce, the logician, called abduction, the argument from the future, to be distinguished from induction and deduction. So if scrutiny of the signs of life reveals life to be one endpoint, not just of processes of induction and deduction, but also of abduction, of making a good guess, then astrobiologists and the rest of us in that world live not so much in Marilyn Strathern's after nature as in a deferred, objectively relativist future, extreme nature as founded on extreme natural relativism. So, getting toward the conclusion, what was life? What was life? Cultural theorist W.J.T. Mitchell has written in recent work that, quote, there is a new kind of vitalism and animism in the air, a new interest in nature with a capital N. The philosophy of life has returned in the age of biogenetic engineering and bioterrorism, unquote. But against this view, Eduardo Katch and Avital Ronell, in their book of bioart, Life Extreme, you can't really see the title there, but it's called Life Extreme, they suggest that, quote, the stability of life or of the living is thrown off course, unquote. And I must agree. One of the concomitants of such a throwing off course is a fascination with extremes and with limits. The three limit biologies that I've examined indicate instabilities in the nature that is supposed to ground life. And all of them do that through a wiggling of what is meant by the form that life takes, a loosening that suggests epistemic shifts in the biological sciences. In the age of synthetic biology, biologists know full well that their knowledge is, in addition to an attempt to describe the organic world, an epistemological construction, a trend that Sophia Roos has lately pointed out in her book, Synthetic, which is an ethnography of synthetic biology. And this puts biology as a universalizing science at risk, one reason that limit biologies often come with a promise to reboot the life sciences. And that promise may be read out of the argument of images that you may have detected accompanying this talk. All of the totem figures that I reproduce, the very simple computer virus kinds of things, the simple ovoid, some maybe nanobe Martian alien thing, the microbes, all of them are elementary forms of life, a limit of beginnings, right? The three biologies make explicit the instability of life in such other domains as reproductive technology, biodiversity, and biosecurity. The very appearance of the word life in quotation marks, like this, or I guess like this if you're British, in my talk, but also in many of the sources that I cite, indicates a social dissensus about its meaning. A fascination with limits operates not only among biologists, but also in my own decision to study limit biologies. What is a limit? It is the point at which an identity uncouples from itself, shades into something else. The limit is, in the words of anthropologist Rain Willerslev and Alberta Corsine Jimenez, quote, the place where a concept outgrows its shadow and becomes something else, unquote. In the examples I've offered, form becomes the shadow of life, the trace, only to outgrow it, at the same time as biologists try to recapture it. No surprise, then, that astrobiologists often explicitly discuss shadow life or a shadow biosphere. What is the shadow of life? The first draft answer would, of course, be death. And a good case could be made that today's biopolitics are ever more entangled with necropolitics, but that's not quite right. A better question might be, what can we see in the shadow of life's limit? The answer, the absence of a theory for biology. Reaching the limit of life reveals what was there all along, that there is no once-and-for-all theoretical grounding for life, that imagining life as it could be is a practice both conditioned by the particular history of the life sciences and one that posits an answer that is endlessly open. Thanks.
Thank you, Stefan. Our last speaker today is Markus Schmidt, who is the founder of Biofaction KG, a company based in Vienna, Austria, that assists in the assessment of new and emerging technologies, promotes collaborations between the arts and the sciences, and facilitates science communication across a variety of platforms. As he notes on his website, Schmidt is the most common surname in German-speaking countries, so if you Google him, you will find a lot of Markus Schmidts with a lot of different interests. So if we've succeeded in inviting the right person, then this Markus Schmidt has extensive experience working in the assessment of novel biotechnologies, including synthetic biology, genetic engineering, and gene therapy. Dr. Schmidt.
So right, thanks a lot, Luis, for inviting me to this very interesting symposium. I think it's a nice overview, and inviting scientists, social scientists, historians, and artists and designers is quite a special kind of symposium, but it's very rewarding. And I try to give you my 5 cents of wisdom, and hopefully this will be interesting for you. I'd like to take you to a journey into space, into a cosmos, but it's not the same cosmos as has been described in the morning session, not the one you can see when you look up into the night sky. It's a different cosmos of possibilities of life as it could be, an encounter with different forms of life that are possible outside. There has been many references to historic quotes and writings. I was very surprised to see a publication by James F. Danieli from the early 70s. This was a time before the Silma conference, where all the discussion about biosafety and implications of genetic engineering took place, which is one of my fields of work, biosafety. And so he actually predicted all the different societal ramifications, the ethical questions, and the safety and security implications, pretty much all of that that are discussed now in the early 21st century. And he pretty much laid out which questions we are dealing with right now with synthetic biology. And what he drew up, he found that there is a kind of a common denominator between different types of sciences, and they all go through 3 different phases. And there was the phase of observation, analysis, and then synthesis. So if you think about physics and Leonardo, and they observe how a bird is flying, and then try to find out what does make a bird fly, and they analyze the wings, and they try to copy a wing, and then make an airplane or a helicopter. And at first, of course, they fail, but later on, they manage and understand how to manage flight. And whereas the first synthetic flight machines, they look very much like the natural counterparts. If you go now, I came here with an Airbus, and it doesn't look like a bird, a little bit, but it's very different from a bird. So we have gone beyond copying what is out there in nature. We've understood concepts of flying and the principles of aerodynamic and so on, and produced something that is able to manage flying. So we have gone into the area of synthesis in physics, and the same goes through for chemistry. I mean, we entered synthetic chemistry in the last century, and so in biology, we're right now in a situation where we're just between analysis and synthesis. So we're starting to go into the third phase of being able to produce new forms of life that have not yet existed. The first steps are very similar to what we see in nature. So we had the presentation from ginkgo. They take a gene from a certain plant or bacteria, and then they put it into yeast, so it produces exactly the same product that is found in nature. So it's very close to nature, but we can go even beyond that. I show this these slides also at biology conferences, and people say, they're getting a bit frustrated, they're not yet there as physics or chemists, but then I tell them they shouldn't be frustrated, they should think of their colleagues in astronomy. So they're still far away from synthesis, but who knows? I found this astrobiology strategy from NASA. I think it's the current one, it's from 2015, and there are 6 points and 6 major kind of strategic goals that they want to achieve, and I was very surprised to find that the last one actually insinuates a synthetic element, so constructing habitable worlds. Actually, it means that they're not just doing description and analysis, but they want to go out and do something, kind of the great, and I'm a descendant of European people that did not immigrate to the United States, okay? So most of you, or many of you, are descendants of immigrants, and this frontier, this exploration of land in the West. I mean, this probably could go on now in outside the Earth. I think probably is a kind of a spirit that is continue to be active here. And actually, if you read in this astrobiology strategy, in this chapter about constructing habitable worlds, they don't talk about constructing habitable worlds. It's more about finding habitable worlds. So it's a kind of interesting. I guess they want to construct something, but they're well aware that they are not yet there in terms of capabilities. So it's kind of in between. Now, we heard a lot about astrobiology, and I think the term exobiology was also used today. And there's a third term called xenobiology, which has in the past been used as one of these 3 synonyms to describe possible life forms outside of planet Earth. I'd like to give you kind of a description of what this term xenos mean. Xenos is Greek and means stranger. And it doesn't necessarily have to be a negative connotation of a foreigner or stranger that wants to do you harm, but just a stranger. And it could be someone that you invite to your house when they pass by. So it's kind of a neutral denomination of someone you don't know. It's something unknown. Okay. So in terms, since we don't want to be xenophobic, we can look at what this term actually means. So before, I think NASA picked astrobiology as their main term, and sometimes it was exobiology. The term first showed up in a fictional book of Robert Heinlein, in a book called The Star Beast. It was mentioned in a science commentary where they were discussing which term would be the best to represent the search for life, extraterrestrial life. It actually has a fictional origin, and I think if you look into astrobiology and space travel, and so there's a lot of fictional origin and fictional kind of groundwork made before science and is going actually to go to do it. So I think that might be also a good reason to work with artists, designers, writers, filmmakers, and so on, in order to be to help to see what's coming up next. What is the next stage? I wrote an article a couple of years ago because we wanted to see if synthetic biology could offer some new strategies to help overcome upcoming and existing biosafety challenges. So in genetic engineering, with genetic engineered organisms, there is always either a perceived or real risk of horizontal gene transfer, of contamination, or of exchange of genetic material. Sometimes you don't want to have this exchange of genetic material. And so there were a couple of traditional ways to try to make sure that doesn't happen, but we will trying to find out some other ways. Okay? It's also now on Wikipedia, and there's a kind of a warning sign, a disclaimer that should not be mixed up with astrobiology, and has now its own life in a way. So the 3 main kind of fields or opportunities, or at least 3, are put up here. So it's about to help us have a deeper or better understanding about the origin of life. The other one is to produce efficient biotechnology products or production systems. And finally, biosafety engineering, to make biological systems that are safer than the one we have right now. I'd like to go through these 3 different steps in order to explain how xenobiology can help to deliver or to contribute to these goals. Looking at the origin of life, we have had some also the talk in the morning about making very old proteins or prehistoric metabolisms. Here's a quote from Sidney Fox that says, "The attempt to understand evolution in a reductionist way, starting from today's living organisms, equals an attempt to learn baking of a cake by disbaking an existing one." And obviously this doesn't work. So taking the life forms we have today and disassemble them and trying to put them together will not get us to not answer the questions about how life originated. This is from a scientist called Wong. He's from Hong Kong, and he was very instrumental in finding the way in which the genetic code must have evolved. And he has identified 8 different stages in the emergence of life, from prebiotic systems to the RNA world, peptided RNA world, and the last universal common ancestor, which we have heard also from today, where all the existing extant life forms came about. And then only after that, Darwinian evolution set in, so this is not from the very beginning. And then there is this 8th step, the synthetic life form. So if you look at Earth from an outside perspective, you can see that all of a sudden there are new life forms coming up that have not been created by natural evolution. But if you think that humans are part of the biosphere and it's just like one other organism, then these new so-called artificial life forms are also part of this kind of natural evolution, if you will. So there are a couple of nucleic acids that have been described by biochemistries, and life only uses 2 of them, so RNA and DNA. And the letter R and D corresponds to the chemical ring that forms kind of the backbone of the DNA. So information is stored in DNA, and RNA has kind of an information storing feature, but also a functional role in living cells. But there are so many other different types of nucleic acids, and we call them xenonucleic acids. And the question is, why did life use RNA and DNA and not one of the other nucleic acids that could have been used? Why was it chosen? What was the reason for that? Same is true for the genetic code. We heard about coding and genetic code today, but actually the genetic code is kind of the formula, if you will, to translate genetic information, so the information in the DNA, the basis ATGC, into 20 or 22 different amino acids that are then assembled into proteins or enzymes. So this is kind of the blueprint to make sense of the genetic information. And it's kind of the universal or standard genetic code that exists, and this is really almost used by all the organisms around the world. So it's quite unusual. It would be a monopoly. If you would like to do that in the economic world, you would never get it granted. It would mean that it's kind of the super company that is responsible for everything. So this is the case in nature. And the question is, in xenobiology, is it possible to create different forms of life or simulate different forms of life that use different genetic codes or have different biochemistries other than the one known from nature? So, and also this xenobiology can be used to make different types of biotechnological products. And there are many examples, and I only want to give you one example here. So talking about the genetic code, so it's now possible to alter the genetic code in a way that, that there are any 3 bases form a triplet, which is a codon, and this codon codes for one of 20 different amino acids. And so if you have 4 bases, it's 4 times 4 times 4, it's 64 different possibilities, different possible triplets that can be translated into amino acids. And there are only 20 amino acids, so that means that some amino acids are coded a couple of times, not just once. And this certain way how to translate it is the genetic code. By changing the genetic code, it's possible to say that, well, a certain triplet doesn't code for the amino acid like it is in nature, but it codes for something else. And this was a publication a couple of years ago. There's a certain technical routine and method how to do it. So it's possible to exchange 2 amino acids. That means that when there would be horizontal gene transfer between this new organism with this new genetic code and an old one, there would be kind of a communication problem. It's probably, because this is very close, it would be like the English version of English and the American version of English. So you would probably understand most of the time, but sometimes you would not, or the German, Austrian German, something like that. So, but you could go and extend this further on. On the right upper side, you see this is a diagram that depicts the success rate or the susceptibility of this genomically recorded organism for bacteriophages. Bacteriophages are like viruses that afflict bacteria. So, and that means that when normally this bacteria is affected by this bacteriophage, by this virus, and it can use it to reproduce, when it's genomically recoded, the same virus or bacteriophage can infect the bacteria, but it is not as successful. Okay, so it takes longer to reproduce itself, takes longer to inflict problems to the host, and so it seems that the genomically recorded organism is not totally immune to this virus, but is on the way of getting immune. If you look at this fish scale, you can see that the original one, the RFN1 plus, is kind of low down, and now we have mastered the first step away. And if we continue to do that, we come up to a very different genomically recorded organism where existing viruses are no longer able to infect this organism. Which means that if you, as Jason Kelly said in the morning, if you have a bacteria or any kind of organism and put them into a tank in order to brew a certain substance, the problem there is that sometimes they get infections by viruses or bacteriophages, and they have to clean up the tank and start from again, and that costs a lot of money and time and so on. So this is a way to make sure that this is not going to happen. But this is one example in order to make biotechnology more efficient, but there are many others that are waiting outside. And the third one, this is the one I'm interested in, is biosafety engineering. Can xenobiology be used to actually make bioengineering, biotechnology safer as it is from today? Right now, there are a couple of genetic safeguard strategies using regular DNA and the standard genetic code in order to avoid horizontal gene transfer. That is, the exchange of genetic material between bacteria from the laboratory with bacteria in nature. So as you just said in your talk, there is a kind of a constant interaction and sharing of genetic information, and it might be that for some reason we are not, don't want to have this exchange, and other ways to inhibit that. And there are ways to do it. However, these mechanisms are not very effective. So it means that for a real life application, it's not going to work. Okay, so that's why these genetic safeguard strategies are hardly used. Sorry. So the construction of our called genetic firewall is the idea that you have very different biochemistries in these xenobiological systems compared to natural biological systems, so that they cannot interact on the very biochemical, very basic levels of life. So on the DNA level or genetic level, they would not be able to interact or understand each other. So it's like the Chinese language or an English language or different operation system in computers. And I'm using metaphors here, and I know about the limitations of these metaphors. And the thing is when you're coming into an area of uncertainty, when new challenges abound, what are you going to do? Right? We celebrate diversity, and there are a total new number of different types of genetically modified organisms. Some of them are genomically designed, genomically edited, chemically modified, or even genomically recoded organisms. And the last two of them, I would say that they belong to xenobiology. So here's one example of xenobiology. In the top part, you see kind of depiction of DNA and RNA double helixes and how they more or less look like in one of the conformations they have. And the colored versions down there are different types of XNA double helices. There's TNA from threose nucleic acid, or HNA from hexose nucleic acids, and they all form different types of double helices. They don't look exactly the same as the DNA or the RNA, but they're doing the job. So that means it's possible to have an information storing biopolymer that is different from the one that is used on all living systems on Earth. Okay? That's an interesting fact. The same is true for base pairs. So not just the backbone of the DNA, but also the very chemistry, how the information is stored. And normally, you know about the AT and the GC, and they form these pairs. But it's possible also to introduce new pairs that do not exist in nature. And many of them have been tested, and recently it was possible to also even incorporate a third base pair, here it's denominated X and Y, into the genetic information of a living organism. So it's an in vivo reproduction of this third base pair. And this is just a proof of concept, and it shows that this can be worked upon, and the genetic information, the way how it is stored, can be expanded. That means that there are different parts in this modular biological system. The DNA, you have a base where the information is stored. Then there's the nucleic acid part that forms the backbone, and there's a kind of a link between the different elements. And all of these three elements have been successfully exchanged in the laboratory. So you can find there's a whole zoo of different nucleic acids with different features and different chemistries, but all of them are storing information. And now we're entering into a xenobiology space. So we look at the natural life forms that we know, and we open up a whole new cosmos of different forms of life that are in principle feasible, but we cannot find them on Earth or anywhere else. But they are possible in terms of artificial and man-made endeavor. And the current job is to find out what are the characteristics, what are the special features, what are the chemical, physical, biological, and so on, characteristics of these different, we can call it little planets of life out there in this space. And there's a lot of work to be done. I mean, there are already a couple of several dozens or even hundreds of different possibilities out there. But we can even extend this cosmos into code engineering. Now it becomes really interesting, and I would say that now the metric even approaches the observable universe. Looking at code engineering again, and I gave you this example of exchanging some of the triplets in the past. This has there's a huge potential in changing the genetic code. All right, and I told you that this could be used to make different types of languages, different types of organisms that cannot communicate with each other. But in order to have it as a real safety device, we need to have some kind of metric in order to measure the distance, between these different organisms, like to compare the differentness, if you will, between English and German or Hungarian and French. Now, how far are these different systems apart, and how large is this space altogether? So a couple of, it was 19 years ago, people started to think about this potential space, this potential space of genetic codes. It was a paper from 1989, and the title was "The Genetic Code is 1 in a Million." Now, in the English language and also in the German language, saying that something is 1 in a million means that it's extraordinarily good. It's a very outstanding, special thing. If you say that, if a Chinese is saying this to another Chinese, it means that exceptionally stupid. So it depends very much on the language, which kind of million is meant by that. So the title is "The Genetic Code is 1 in a Million." So which million is it? Is it the Chinese or the English version of the million, 1 in a million? Turns out they did some computer simulations and altered the genetic code to find out if it was more or less robust or resistant to mutations. So depending on how you change the genetic code, a mutation could either lead to the same amino acid or a very different amino acid. So in that case, it turned out that the genetic code is actually very good, comparing it to a million other genetic codes. But it turned out that there are better codes out there. So there are artificial genetic codes that exceed the natural code in a number of different parameters. So it depends on the molecular volume and other technical parameters. So it's possible to have genetic code that's even more robust, but it has never been invented by nature. And if you think about it, and here are some orders of magnitude, okay, so there are a lot of numbers, I'm sorry for that. If you think how many different genetic codes are actually possible, and you come to a staggering number, it's 1.5 times 10 to the power of 84. That is 1.5, and there are 83 more zeros behind it. It's such a huge number, it's beyond comprehension. I think the only number that is about that size is the number of elementary particles in the universe was estimated to be 10 to the power of 80 or something like that. So we're approaching the size of the cosmos here in the combinatorial possibilities. And if you take that, even if you put some extra constraints on it and say it should be very similar to the genetic codes that we know from nature, there's still 10 to the power of 71 codes possible. Yeah? And then we did an estimate of what's the total number of genomes that have ever existed on Earth in the last 4 billion years. Imagine that every cell would have a different genetic code, and every time it divides, it's different. No? You can only come up to 10 to the power of 45, which is not half 10 to the power of 80, but because it's with the zeros. So that life on Earth has never had the chance to try out all the different possibilities of the genetic code. It just, for some reason, came up with this one we have right now. It's pretty good. We are here, so I don't complain. But there are many more possibilities. And this opens up, as I said, a cosmos of possibilities. And maybe these other genetic codes have different characteristics that might be useful for one or the other thing. The other thing is there's a possible good reason why nature has not explored all these different landscapes. And this has to do with the fitness landscape, okay? So if there's a plant, a bacteria, or any other organism, and it exists at one point in time, and you know the environment changes, it has to adapt, it changes and evolves into something else. But along this way, in evolving into something else, it always has to be viable and be able to survive, has to be fit. At no point can it go extinct, because if it does, it's over, it's end of the line. It's also one of the figure you showed from Darwin. You know, it's kind of a tree, but there's always, from every extinct organism has a continuous line of fathers and mothers or predecessors down to the last common ancestor. You cannot break this chain of generations, okay? So that means that once you find something that works, you have to, and if you want to change it, the next stage also has to work. And now I'm using a metaphor from a semantic world, and this is a puzzle or a game, and you start with a certain word, with a 4-letter word, mole, and the challenge is to arrive at a different word that also has 4 letters, and you can only change 1 letter at a time, okay? So how would you do that? You start maybe with exchanging the first one, from mole to pole, pose. So there is a way that all of the words that come up there have a meaning, okay? There's no one word that has no meaning. So it's possible to change and morph from one thing to the other. But if you look at a word that is more complex, I mean, you can try it, but I would say that it's not possible to come from performance to macroscopic, but just changing 1 letter. That means that there are some islands of meaning in a sea of meaningless environment. Okay, so and the more complex a system is, the less likely it is that it's going to change to something other, something else that is complex. Or imagine if you take just 2 books out of the library, but trying to change 1 word at a time, you come from one book to the other, and all the books in between make sense. That would be kind of an interesting experiment. It take maybe more than a year for a scholarship to do that. So that means that actually this combinatorial space is comparable to the universe, where the majority of space is a vast nothingness, and there are a couple of spots, you know, the exoplanets. Maybe there is life out there. Okay, so and in between there's nothingness. And I'd like to quote Werner Herzog, who says that the common denominator of the universe is not harmony, but chaos, hostility, and murder. Which it's pretty dark. But on the other hand, there are these islands of meaning and these islands of happiness, or these islands of life where life can flourish, but they are quite rare. And to go to these other places is incredibly difficult. I mean, we can go to Mars and maybe to other planets in our solar system, but even go to the next exoplanet, Trappist or whatever, it's not conceivable to do it, as we know right now. And it could be that going to other functional xenobiological systems is just as difficult as to travel to an exoplanet, unless we find a very smart way to do it. I mean, there in science fiction, you read about wormholes. There's a kind of shortcuts through to this endless space, and we can find ways to make it smarter. Can we find a wormhole, something like this, to come up to these islands of life? And I give you another metaphor. It's another puzzle. And you know, this is a very famous puzzle. You need to find. This is the last 3 slides. Yeah. Thank you. So you can only use 4 lines to mark all the 9 dots, and you have to do it without stopping. And there is a solution to that, and the solution is that you have to go outside of kind of the box in order to be able to do it. But I guess that this will be not enough. Solutions like this will not be enough to find these new places. What if we just can draw 1 line? What do we have to do then? And so we have to be really creative and bend, you know, the space on which we're working, and then we can find a way. I don't know if it's possible, but I think in order to be successful and find different forms of life, we have to be very creative and very hardworking in order to do so. And I don't know if it's possible. Thank you.
All right, I think we're going to do a short question and answer period. We're running a little bit behind schedule, but I think that we'll be able to make that up in the next session. So just a reminder, as our former CEO of AAAS used to say, a question is something that is short and your voice goes up at the end. So let's try to keep that in mind. So I will take the liberty of the first question myself. Luis, you mentioned a quote from Mueller in your talk, for how long will our world remain habitable? And I feel like there's a sense of urgency there that we didn't really get into in the first session, where we were kind of like, oh, wouldn't it be fun if we, went to these other planets? So I wonder, given that there is this sense of urgency, does that change what kind of risk we're willing to take in terms of, how we think about synthetic biology and synthetic forms of life, putting stuff out there, I guess?
Yeah, I think, the question of habitability and how it applies to what we do in our science and what it means in larger cultural contexts is what I was trying to get at there. Mm-hmm. And that for some people to engage in the new possibilities of synthetic biology is to make our world more habitable than it is now. Whereas for other people, it would be the exact opposite, that they're concerned about what the potentials of that might be. And those sorts of discussions and debates are things that we find at any moment in time, and that's what makes that an important topic for us here on Capitol Hill, right? Because that's a political question of how society organizes itself. So I think rather than saying that I have the answer to that question, I think we should always be aware of the ways that it appears so obvious to us that this is the right way to think about a particular issue can be troubled by trying to understand how it might make sense in a different way to somebody living in a different place or a different time.
Sure. That kind of leads into my next question. So I feel like I was successfully convinced Stefan that the boundaries of life are kind of fuzzy and arbitrary, but if we can't even agree on what these boundaries are, like how do we hope to control that or regulate it moving forward?
I guess instead of the word arbitrary, I would say conventional. So not all hope is lost. I mean, we have definitions that do particular kinds of work at particular times. So the various kinds of hypotheses about what counts as a living system, assayed at a genetic or a cellular or an organismic level, have produced new kinds of things in the world and new kinds of scientific questions. It doesn't mean that's the only way it could have happened, right? So I guess I would not use the word arbitrary. I would use the word conventional and underdetermined.
Great. Are there questions from the audience? This one right here.
I just wanted to ask Professor Dr. Hare.
Can you introduce yourself?
What?
Can you introduce yourself?
Oh, I'm Helen Muller, not Mueller. Sorry. I wanted to ask you what the ethical implications. It seemed very mechanical what you were talking about, and I think there are tremendous ethical implications of what you're talking about. Could you just comment briefly on that?
So I think the reason why I'm interested in this is, apart from that it's, I think, find it a fascinating field, is the opportunity to produce a biosafety system that is very safe, okay? So there's a high level of safety and high level of probability that this is not going to fail. Because I see there that in synthetic biology there's a kind of very strong movement towards, developing new tools and new organisms and for all kinds of different applications. And many of them imply release of genetically modified organism. And I know that this has in Europe, but also I guess in the United States, there's a controversy about under which circumstances this is a good idea and under which circumstances it's not. So I think it's a responsibility to, at the same time as we see people developing these new tools, to provide new measures to make sure that this is not going to harm or to minimize the risks to the greatest possible extent. I think the ethical question itself is actually to work on different new biosafety engineering systems. But of course, this new system itself might also pose a threat or a risk or a safety problem to the natural world. And I'm not saying that this is not going to happen. I think it's very relevant and very important to be very careful in installing these new systems or testing these new systems and to describe them very well. And be also very careful in releasing anything like this into the environment before not enough information about their characteristics are known. So in that respect, I'm not arguing for like a large-scale release of these new kinds of life forms in the event when they're going to exist, but a very careful description and characterization of that. And that must go hand in hand with the development of these systems. The interesting thing is that because they don't exist out there, we do not find them in nature. It's a very painstaking process to actually produce these systems. And by producing them, I hope that there's enough time to learn about them, rather than to take something from nature and exchange it in one or other way. But yeah, there are also ethical issues involved that I think some of them are not yet have not yet been articulated, and they will come up in the future, and we will see what that will be. I also don't know all the answers to that. But from a safety perspective, I think this is very worthwhile to try to find out if this can work as a safety device or not. And this would be also a question to an ethical, to an ethical question, an answer to an ethical question.
It just reminds me of, Hitler wanted to create a superior race. So, I mean, the both laboratory and the women, that's what they were trying to do. So, I just wanted to.
So actually, the purpose is not to make something that is superior. You could also do something that is inferior, so that is not, challenging existing natural organisms. It doesn't have to be always better and stronger or faster. It can be worse.
Although I was very intrigued by your diagrams showing that there are other things that could be better, and I think we'll hear more about better in the next session, but that could be better depending on what your human criterion was of what you wanted to evaluate.
Exactly, yeah.
And so that nature has lots of possible
So
solutions for things.
There's a larger portfolio of things, and then it's our responsibility to choose wisely which characteristics we would like to deploy and which ones we don't want to deploy.
And there's another book that I saw on the shelf in the Sagan collection that strikes my mind now called Parallel Botany, which is a book about plant forms that don't exist anywhere in the world, but it's written as if they do. And it's fascinating to think that book itself is a very odd and unusual book with fake references, but it's constructed as a whole book. It's a wonderful intervention itself, but the fact that that was on Carl's bookshelf, I think, is another interesting thing. As part of his way of thinking through the possibilities was to think of parallel botanies that don't exist in the world but could, or that we analyze as if they did.
there's another question.
Hi, Nathaniel Comfort from Johns Hopkins. My question actually follows up on this discussion. Thinking about what those criteria are. Penny Boston, the director of the NAI, likes to say that life is not a cloak that clothes the earth, but is rather an integral part of the earth, that the earth is inherently a living thing. And if we start getting into synthetic forms of life, artificial forms of life, xeno life, based on other criteria than what the planet is made to support, we start getting into other criteria such as, for example, the criteria of capitalism, right? Where the criteria of life perhaps becomes profit and shareholder values. And this is really directed at any of the three of you who care, or four of you who care to answer. If you have any thoughts on the risks of designing life on economic criteria rather than ecological ones.
I could mention a book by someone called Melinda Cooper, which is a book called Life as Surplus, which is an examination of contemporary biotechnology from an economic point of view. And she examines kind of 1970s, 1980s limits to growth discourse and makes the argument that new kinds of biotechnology that target extremophiles are kinds of ways of she suggests that the idea of the natural resource is constantly to be reinvented, right? And so the production of knowledge about new kinds of life becomes a kind of a way of producing new kinds of resources, things to be imagined as resources. Now that can happen within a variety of economic systems, but she names market capitalism as producing some of the impetus for that, right? New kinds of property. You didn't know that not only can a plant be owned, but there's this thing in it called its sequence that could also be owned, right? So there's this kind of production of new kinds of knowledge that get naturalized, very much within a regime of economic accumulation. So I think that's a very good question.
Also to follow up on that, and you're right, and this is not something to come, it's something that's already here. Life forms are used in economic terms and they're part of capitalism, meat production or agriculture, and then that's widely existing right now. And one of the kind of consequences of this massive use of technology towards life, there is, for example, if you look at the phosphorus on nitrogen use, especially phosphorus, we're using up phosphorus resources. We take them up from the earth and they're used in agriculture, but then they go out to the rivers and to the ocean and they're dispersed. So there's no way right now to get them back, because in the natural world there is no metabolism that recovers phosphorus, from highly diluted oceans. So this is a problem that has come about with agriculture. And I'm not saying that all of the things that are done in synthetic biology are going to be done in order to help to make that problem go away, but there are some avenues and opportunities opening up with the help of artificial metabolism in order to try to recover or to undo the mistakes from the past. Okay? I'm not saying that this is going to be the majority of applications that we're going to see, but at least there's also the option to do that, not just, to also help to recover and become more sustainable and renewable. So long as one can make a profit doing it? It depends. If you look, for example, I'm from Austria, but in Germany, there are some rules that mandate that people who have contaminated the soil, they have to clean it up. And it's a law, but they have to do it. I guess the EPA has some similar laws. And this is not for profit, they just have to do it. And you have to have a strong government that is behind it in order to be able to do so. If you don't have a government, then of course then nobody will do it.
So the phrase that pops in my head on that real quick is new space, right? If we look at the history of space, it's something run by state governments and international treaties, and now we have it emerging and transforming in new ways. And one might think, what does that pattern look like for life space, right? And what is an interesting place for government investment or for industrial innovation, and how do those relate? And I think that question gets even more complicated as both of those spaces come together.
I think with that, we'll wrap up the panel. And if you have additional questions for the panelists, please find them after. So join me in thanking this lovely panel.
Okay, so as I mentioned earlier with the piece I Am Life, this piece also was originally, well, not originally, actually originally it was a stage piece, but we did make it into a podcast, at which time we really became appreciative of the soundscape that this piece can create. So we're going to start this piece from the back, so you can just listen to the sounds and the soundscape that we're creating. And we will come to the front, but at one point we'll kind of clear off the stage a little bit. Again, we won't leave the stage, but again, it's an invitation to focus on the sounds. Okay? This piece is called Life's Voicestra. 542 million years ago, the Cambrian explosion. Wow, it wasn't a literal explosion. Suddenly, animals, and all in the oceans, Opabinia, with 5 eyes and a grasping tentacle in the middle of its face. Anomalocaris. At 3 feet long, the terrifying giant of the ocean. Life diversified and proliferated wildly. Then, 440 million years ago, the Ordovician-Silurian extinction. 27% of all families and 57% of all genera gone. Maybe because of global cooling. But then a vast variety of fish developed. Insects appeared. And animals started to leave the oceans, the amphibians. But then, 375 million years ago, the late Devonian extinction. 19% of all families and 50% of all genera gone. Global cooling, oceanic volcanic activity, or comet impacts. Then, insects got big. Dragonflies with wingspans of 2.5 feet. And an even greater variety of reptiles and amphibians dominated the water and land. But then, worst of all, 251 million years ago, the Permian extinction, the Great Dying. 96% of all the species in the oceans and 70% of all the vertebrates on land, gone. Global warming, coal and gas fires, asteroid impacts, every type of organism was hit. It took life up to 10 million years to develop any kind of biodiversity. But then, smaller insects. And early dinosaurs, archosaurs, and crocodiles. But we know how that ended. 205 million years ago, the Triassic-Jurassic extinction. About half the life on the planet was wiped out, opening the way for the dinosaurs, who then diversified and dominated the Earth. Quetzalcoatlus, a 575-pound bird that was also a very fast ground runner. Coniophis precedens, a snake with a lizard head.
It's getting closer. It's getting closer. It's action roll.
Brachiosaurus, a 50-foot tall dinosaur. Well, 30 feet of it was just its neck.
Too much broccoli.
And early mammals, scurrying through the treetops or burrowing in the ground. But we know how that's going to end. 65 million years ago, a comet the size of a city
What is that? What?
came hurtling towards the Earth.
Get the kids! Get the kids! Get in the hole! Get in the house! Run! Run!
It was a very bad day for the dinosaurs, but it was a very good day for the mammals that burrowed in the ground.
We're good. We're good. Y'all all right? We good? We good? I ain't getting nothing in my claws either. We're good. We're good.
Okay.
All right. We're good.
Without the dinosaurs, these mammals thrived. 200,000 years ago, our earliest ancestor appeared, a squirrel-like mammal named Purgatorius. Slowly, the world we know today began to develop. Well, the only dinosaurs that remained were the birds. Now mammals dominate our water and land. Dolphins, whales, horses, and elephants, and humans.
Oh my God, Becky, look at her butt. It's so
Ew,
big.
So big.
Oh, wait. It's an elephant.
Oh.
It's an elephant.
It's an elephant.
Yeah.
99% of all the species that have ever existed on this planet have gone extinct. Where are we now? Today, the Holocene extinction and the beginning of the Anthropocene. Due to pollution, climate change, and the purposeful destruction of habitats, we humans have triggered the 6th major extinction period of life on this planet. The golden toad and many other amphibians are being lost. The entire Hawaiian Mahoidae songbird family is gone. The eastern cougar, a North American cougar, went extinct in 2011. Millions of bats have died in the US due to a fungal infection in the last few years. And the Yangtze dolphin, a freshwater dolphin that lived in the Yangtze River in China, went extinct in 2006. Where is life going now? And how do we know? Will synthetic biology help?
Should it?
What is life? What is life as it could be here on Earth and on other planets?
While we're getting the chairs set up here, I will introduce myself in this third panel, and then my panelists will join me. I am Nicola Twilley. I write about science and technology for The New Yorker. I co-host a podcast called Gastropod about more than just snails. And some audiences that works and some it doesn't. and I'm writing 2 books, one about the artificial cryosphere, which is how I refer to refrigeration, and one about quarantine. So the question of planetary protection comes up in that one. This final panel, which I have the joy of moderating, is from the studio. So we've come from the laboratory in the library, and now it's time for the artists and designers to have a say. So I'm going to invite my panelists up. We're having a party up here, apparently. So come on up, folks. I'm going to try. It's a studio if you're in it, right? Joe is concerned that he works in a laboratory, but I say his presence remakes it into a studio. How about that? Studio, laboratory, all the same in the end. Okay, so our first speaker is Daisy Ginsberg. She is an artist and a designer who explores these worlds of synthetic biology and brings a much-needed critical eye to visions of bioengineers. She's often the voice of reason in the room, I think. My favorite work of hers is a scatalogue, a collection of synthetic biology-colored poo. So you'll have to ask her about that later. But Daisy, up you go.
For once, I wasn't going to talk about the poo, so so it's brilliant to be here and to be thinking about my work actually from a very different perspective. So when I was talking with Luis about today and what how I could sort of frame what I do in terms of astrobiology, he started to tell me about the story from 1993 when Carl Sagan and a team of scientists announced that they'd identified signs of life on a planet in our galaxy. And they collected data with the Galileo Space Telescope from atmospheric methane to radio emissions to come to this hypothesis. And of course, this planet was Earth. And they were using Earth as a control for identifying existing terrestrial life, as debate raged around funding this kind of exploratory science. But when Luis told me this, I was really I had to look at it in a fresh way, because for the last 10 years I've been exploring a field of science and engineering which is less interested in justifying the importance of finding life, but in making life, designing new life forms for the benefit of humanity, or even the benefit of our planet, or so they promise. And we had a great introduction to this technology this morning, which was synthetic biology. So here's an example of what it actually looks like. This was made by undergraduates all the way back in 2004, a decade after Sagan's paper. And it's a plate of bacteria engineered to go dark when exposed to light, like photographic film. And these undergraduates made this for a student competition called iGEM, the International Genetically Engineered Machine Competition, which began at MIT around this time. And the statement, "Hello World," alludes to the first program that you learn to write when you learn to program computers. But of course, they were programming biology. I was a design student, and I was hearing kind of engineers going into biology and talking about trying to transform biology and life with it into what sounded to me like a 21st century design discipline. And I'm not a kind of regular designer. I come from a more critical practice, so I wanted to know, well, what would they design? What would be good design? And crucially, who would get to decide? And over the last decade, I've kind of hung around, and I've been watching this dream become increasingly real. So Jason showed the same article this morning from Wired magazine saying, "Watch out, Johnny Ive, biologists are the next rock star designers," and was very pleased about this. But what does life
as it could be mean when we talk about humans designing life. And the panels we've had have already started to raise problems with these kinds of questions. What life are synthetic biologists imagining? Is it just about making the planet greener, or is it about making kind of rose perfume? Do their dreams go further than that? And if they realize their dreams, will life still be recognizable? It will be recognizable to the Galileo Space Telescope as it looks for signs of life, but would it be recognizable to us? And these are the kinds of questions that I explore in my work, and I use the language of design to think about these problems of humans engaged in the design of life. So I'm going to share a few projects with you, and hopefully this will become more clear. So I'm going to begin about sort of the idea of making life elsewhere, and then sort of touch on this idea of bettering the Earth and bettering our own human-made systems, however poor they may be. But then we get into the strange space which has been alluded to of remaking nature, the potential to transform life beyond what we already know. So I first encountered this kind of question of making life elsewhere in the context of colonizing other planets. And again, when Luis was telling me we're going to talk about astrobiology, I was like, oh, you mean space colonization? To me, that's the automatic kind of go-to area. And that's because I've spent so much time with sort of techno-utopian synthetic biologists who are excited about this idea. Earth is old news. We need to go elsewhere. So at iGEM in 2009, I watched students from Tokyo Tech present their efforts to engineer terraforming E. coli to withstand Mars's harsh climate and make it livable. And this is from their presentation, and these were the qualities that they imagined were necessary. So E. coli that could suntan itself to protect itself. And that was just sort of this idea of terraforming. But I think that there's more to it. And in 2013, I read a NASA job advert for a food scientist, synthetic biologist, to develop foods for the long journey to Mars, as you can't take 30,000 kilos of supplies with you. So this kind of plastic-wrapped food that's the bane of Earth won't be going to Mars. Now, obviously, I'm not qualified as a food scientist, but I thought that was unfair for a start. But I was also really curious about how they might imagine new foods and what they might imagine. And what was clear from sitting in on kind of the conference around this discussion at NASA was that like these lonely astronauts were going to be stuck eating algae, or if they were very lucky, some kind of space tofu that would last them for 1,000 days. And what's really interesting is NASA's technologies often come back down to Earth. And so I wondered if this is what we will be eating in the future. So this project, it's a very short project, that started to kind of explore this space. And it's called Seasons of the Void, and I made it with Sasha Poflep and Andrew Stellitano. So we imagined as the spaceship flies away from the sun, taking a crew to go and colonize Mars, photosynthesis would be replaced by electrosynthesis, which is a process much more efficient at converting energy. We imagined tanks of symbiotic engineered yeast cultures which would be on board, and they would sort of accumulate their secretions into space fruits, which would gorge on electricity. And these different cultures would secrete material to make these layers, which would sort of accumulate around an electrode seed. And this diagram shows how the layers, so it's a diagram of the solar system, but also of the fruit itself. And the layers would be distorted by magnetic fields kind of encountered as the ship went behind planets and experienced solar storms. So you'd sort of have this weird kind of space tofu, essentially. But what we were really sort of trying to explore through aesthetics was how the astronaut would still be engaged in Earth's rituals. They would be farming in the void. There were still seasons. We still had this connection. Despite everything, this sort of urge to escape Earth, there is still something that brings us back. So making other planets livable is pretty far off, and I think the debate this morning, we get a sense of that. And what really disturbs me about that debate is as we're searching for life elsewhere and imagining where else we could live, we're decimating the biodiversity on a pretty livable planet. So I'm definitely one of the ones who wants to stay behind rather than going off for intergalactic colonization. And synthetic biologists promise, though, that they can make our Earth better by making new life forms and delivering that through industry. These are just two examples of my vast collection of promises of better from synthetic biology. And they use these kind of visions of better, these speculations, to make material reality. And the question of what better actually means to them is something that I have just spent the last 4 years trying to find out, having written a PhD on it, and I'm not going to, that's for another day. But these questions have kind of stuck with me for a really long time. And it was around 2009 that I went to visit, is that going to go? There's an animation there. I went to visit a plant scientist called Jim Hasselhoff at Cambridge University. And he was researching how plants grow, how cells differentiate. And this video sort of shows these cells sort of splitting. It's just sort of something that I borrowed from him in 2009. And I was asking him why, as he showed me this video, why he was doing this. Why was he interested in how plant cells sort of divide? And he said, well, one day we might grow products inside plants. Now, Jim meant chemical products, but there was something about the way he said it and the other projects that he's involved in that really stuck with me, and I was just getting sort of started in this space. I went back to London, and I was working with Sasha again on this project. We were sort of thinking about this global shipping of stuff, this mad world that we live in, kind of completely unsustainable patterns of consumption. And we wondered if people like Jim were imagining a future where plants might be programmed to grow forms, to grow products, quite literally. In this future, plants would become industrial robots, replacing today's sort of production lines and manufacturing. To explore this, we designed a commodity from this future to kind of try and make sense of it. So the project is called Growth Assembly, and we have a product here that's grown in 7 different component parts, which would be harvested and assembled to construct this item, a herbicide sprayer. And when we were trying to pick what item, we decided this would be the essential commodity in this future. It would be used, everyone would need one to protect the kind of delicate, engineered nature that was making things from the existing, older, established nature that could naturally defend itself. So these are the items. There's a herbicide gourd. It's a bit like a melon. You'd put your arm through it and kind of squeeze out the herbicide poison inside it. And there was a spike that would connect to it. This is the connector fruit that sort of connects the spike to the tubes, and it grows inside a shell similar to a walnut. Nozzles grow inside fruits related to the tomato. So here's a detail of that. These are the tubes that carry the poison to the nozzle, and they're grown as long, hollow roots. This thing would be grown and harvested and assembled. And what we were really interested in was thinking about how diversity and softness might be introduced into a space that's previously been dominated by manufacturing standards and hardness. We imagined that shops might evolve into factory farms, and you would sort of grow licensed products at the same place where you would buy them. Products wouldn't be shipped around the world anymore. Instead, seeds would be shipped because they contain all the manufacturing instructions encoded in their DNA. So we showed this back to Jim, and Jim was like, no, that was what I was thinking. And that was really kind of, for me, it was an early project. And it was fascinating because it appears so seductive. It's the perfect solution. But the project was actually inspired by watching Soylent Green, the film. And it has, for me, that troubling element in it. Industry subsumes nature entirely, and human design strategies replace nature's elegance. We have 7 designed objects to do what plants have evolved to do without intention. So this, for me, was the kind of the beginning of trying to work out how to make projects in this space. And as I was working on that project, I began to think about this, which we've seen a little of already today, the tree of life. It's a human-designed model that we use to try and make sense of the complexity of the living world. And it's also a constantly changing thing as we learn more. And having met people who are experts in this, it is a very fierce field with a lot of big disagreements. So when we were working on Growth Assembly, I began to wonder, where will we classify these newly designed organisms? They may have genes from all the different kingdoms, and they may even have completely novel ones. well, we need a new branch of the tree of life. So I added one, and this was my master's project, kind of was called the Synthetic Kingdom. And it was a really useful device to go and talk to synthetic biologists. And I also went to a group of the taxonomists who fought about it. They were like, this is completely wrong, and this would never happen. Some said, well, it'll be little leaves, it'll never be one kingdom. Others said, well, it'll be really small. Synthetic biologists will never manage to make that much stuff. Drew Endy, who's one of the pioneers of synthetic biology, was like, no, it's going to be a table covering everything. And some others were like, no, it's more like spaghetti. And it became a really useful tool to discuss how designed organisms might be different or not so different at all. And what I've learned with my projects, because they use this sort of polite design language, is that sometimes they just infiltrate too far. And so it ended up on the cover of the first synthetic biology textbook, which I thought was great, because I was like, this is a place for debate, and synthetic biologists will think about this, but actually I ended up giving them their own kingdom. So, lesson learned. So, in a way, like my earlier projects, the critique as I was working out how to use this language, it was quite subtle. But in the last 8 years, so much has changed in synthetic biology. And I think we're facing a much more challenging set of... In a way, like my earlier projects, the critique as I was working out how to use this language, it was quite subtle. But in the last 8 years, so much has changed in synthetic biology. And I think we're facing a much more challenging set of ambitions and kind of technological possibility that needs scrutiny that I don't even know how we begin to address. And I think these concerns have sort of, again, been emerging today. So there's a group of synthetic biologists who believe that they can better nature, that they can remake Earth. And I've encountered some of them this year. And I think this slide really sums up this kind of ambition. This is a new marketing campaign from a synthetic biology and biotech company called Intrexon. And they are announcing that humanity are Earth's tech support. So if I quote from their website, which is a screenshot here, they say, "One planet, one health is an acknowledgement that our ability to reshape this planet makes us all bioengineers. How are each of us and our daily choices influencing the journey of life's code on this planet?" And then, more ominously, we're just getting started. Now, I find this extraordinary for many reasons. Some people in this room may think it's great. But for me, the idea of the planet as a technology, which needs and also can be solved, is deeply problematic. Another idea is this journey of life's code, the idea that life is determined by, crucially, patentable DNA code is not the whole story of how life works. It's more complex in its interactions. Yet, Intrexon is asking us to join them in a movement so they can help us, or we can help them, to save the planet. And if we're just getting started, what does this look like? What is life as it could be through Intrexon's eyes? So here are a few examples of the kinds of things that we are encountering now already. Intrexon owns a company called Oxitec that engineers male mosquitoes as a design object. Now we think about the biological thing, which is designed not to transmit life as a design object. Fascinating tool in the fight against. So we had mention of CRISPR today, the kind of programmable pair of scissors. It's a gene editing tool that's giving scientists much more precision than ever before to edit genomes. This bombastic report from Wired, it's the end of life as we know it. Well, it's probably not, but it is leading to some new technologies that may dramatically change biodiversity. And the main one in question is called gene drive. So you may have heard of this as well. It's being debated a lot. So far as we know, no gene drives have been activated in nature. And what it is just a genetic device to drive a trait through a single species, fast, through far fewer generations than has been possible before. And the idea is that so the Oxitec mosquito, you release it, and you have to carry on releasing those mosquitoes to dampen a population. But using a technology like this, you could alter a species entirely or eliminate it through the driving through of genetic material. So that's, again, another technology that could change life as we know it.
And Marcus was talking about efforts to expand the genetic code, and it all seems very abstract. This is a worm with a different genetic code to us. And I first saw this at a conference in London and was completely amazed to see this thing going across the screen, because it just looks like any other worm seen through a microscope. It looks the same, but it isn't. And that's where we get to this space that was also kind of hinted at this morning. As the first efforts to really genetically engineer humans get underway, as human genome editing trials kind of start around the world, how many generations do we have left as humans? Who gets to decide? And I'll finish off with a project that explores this dream of bettering nature to a certain group's idea of what that better nature is. And I'll show how I use design to try and explore the values at stake. So in 2013, I was very lucky to attend this conference when conservationists and synthetic biologists met for the first time. And they wanted to discuss if they had anything in common. Now, I don't know many conservationists and synthetic biologists kind of attending conferences together, and it was kind of evident why. Both very nice groups of people, but with very different views of what better means. So the conservationists are looking backwards. They're trying to protect biodiversity from human kind of influence. They want to stop humans. Meanwhile, synthetic biologists are busy inventing new biodiversity for the benefit of that same humanity. And that's it was quite a tense meeting, and there's lots of glasses of wine had in the evening, because they were trying to work out, does the survival of one field preclude the other, or can they work together? So meanwhile, there's this backdrop of the 6th Great Extinction, sort of big extinction event in the history of biology, humans probably likely its cause. And a really interesting idea emerged that I hadn't heard people talk about before, which was the idea of using synthetic biology to infect nature, to save it. I wanted to know, well, if we do that, well, what would the wilds look like in this synthetic biological future? How might this future come to pass? Whose interests are really being served? So this project, Designing for the 6th Extinction, with a very cheery title, starts to explore these questions. Now, in other projects, I worked directly with synthetic biologists, and a lot of my practice is kind of very engaged in the field. And this one, I was just consulting with them and with conservationists, sort of isolating myself a bit purposefully to kind of explore some of these issues. So this is how it looks. In the gallery, there's a large light box and some small objects and models. And the image in the light box is this most perfect-looking forest. And as you look more closely, though, it sort of looks very biodiverse and healthy, but you start to see strange organisms lurking in the undergrowth. And in this fiction, these companion species have been designed to preserve biodiversity and have been released into the wild. I decided to only describe them through the language of patent applications. So accompanying the big image, they are described as machines. So this mushroom or fungus is the self-inflating antipathogenic membrane pump. And it's a kind of fungus, fictional fungus, designed to fight a very real tree pathogen called sudden oak death, for which no cure yet exists. So the device would inflate when it detects a disease and pump serum into the tree. It's fictional, but I reference really kind of current ideas in synthetic biology, some of which have been introduced today. So, for example, this whole system would work on a 6-base DNA kind of code to separate it from nature. So the ideas that Marcus was talking about, what would that actually manifest like? I don't imagine yet we can make things as complex as this, but the idea of making an organism that doesn't connect with nature fully, but lives only to preserve nature, is for me kind of fascinating and provocative. Is this still alive? Okay, I have a few more slides. So this is the mobile bioremediation unit. It's a kind of slug. And this one has a biosafety kind of concept in it called the kill switch. So it's an organism that kills itself. And the idea behind this, that's again another real thing in synthetic biology. And the idea was to build a world within this, using existing kind of trends in synthetic biology and conservation, to then kind of look at how a world like this might be constructed. It's not meant to be a prediction, but it's a way of looking at the institutions that might emerge.
And the purpose for this, for me, is to affect debate now. So a kind of interesting ramification is that it kind of spread onto the internet. And I got emails from people in synthetic biology saying, when this kind of headline of synthetic creatures could save the planet emerged, saying, well, Daisy, aren't you worried that you're going to damage synthetic biology by promising that we can save nature, and then we won't, and then the public will be disappointed? I found this amazing because this was not meant to be utopian, but it showed, when I was reflecting on synthetic biology's promises, these kind of fault lines around the image and the vision. And just to finish off, it is possible for this work to kind of infect spaces in good ways. And so I was kind of thrilled when it appeared on the cover of Fungal Genetics and Biology, which I never imagined I would appear on the cover of, because the editors were using it to discuss what they should or shouldn't design. And finally, this is it in Ginkgo Bioworks' Organism Foundry, like the space where organisms are being designed. So if we industrialize nature completely, will it still exist for us to save? Sagan was searching for life as we know it on Earth, but if we're terraforming Earth, will Earth still be Earth if we remake it? And as synthetic biologists push forward to transform biology into design, we need to think about design as a space of satisfying needs and desires. So if we want life, what kind of life will we get? No, finish there. Thank you.
I'm going to do my best. For me, speaking for 25 minutes is much harder than speaking for 2 hours. But I'm going to put a timer on. No, a stopwatch. I'll just pull the plug on me, yeah. Okay, what a amazing and stimulating and interesting and thought-provoking day it's been so far. It's always good and reassuring to see art being included as an integral part of a scientific conference, scientific symposia, and especially those scientific symposia which are particularly orientated towards stretching the way that we see the universe around us, that we really have to reconsider a future that we can't even begin to imagine, but we're actually trying to investigate, poke around the edges of that. I was especially taken by some of the statements that Luis made in the press that I was seeing in the run-up to this, particularly the piece that stating that we should study science with all the power and insight that the humanities can bring to bear, and that metaphor is in fact generative, a constitutive part of scientific thought and practice. I thought, I love that. You so rarely hear scientists talking of metaphor. It seems like an anathema. But kind of what I want to try and propose for this talk is that, again, metaphor is this really useful tool. We begin to understand things in new and powerful ways if we apply metaphor. I also want to paraphrase a colleague of mine who was very active in the DIY bio movement. Paraphrasing him, he said that something along the lines of science being way too important to be left to the scientists. I'm interested in lots and lots of things, as I'll try and express through this, and I'm going to have to try and rush through it a bit more than what I usually would. So I'm in art, I've been working for a long time in this kind of fuzzy triangle between art, science and technology. Really interested in the way that especially science and art kind of rub against each other, how we can form parallel practice and look for that kind of tense area where they split apart and divide and try and do different things. But first, I'm going to go in a bit of context about what it is that I do and why. Where's the clicker? Here. Cool. So the first organisms that we knew of that survived in space were not the famous dogs and monkeys that kind of seized all the press from the kind of late 1940s onwards. In the early stage of the space race, the first living organisms that actually survived the journey into space were fruit flies on top of a V2 rocket in 1947 as part of Project Blossom. This was the first experiment that was particularly aimed to find out how non-terrestrial environmental conditions and influences might affect biological processes. We'll come back to fruit flies and non-terrestrial stimuli a little bit later in my talk. The primary motive, obviously, of all these earlier launchings of animal into space was much more about testing the machinery for the later human use, later human cargo, as it was at the beginning, than it was about understanding the possibilities of life in space. But since then, insects, amphibians, invertebrates, and mammals have all been flown into space for pure research on a whole range of other missions. From the early biosatellites of the 1960s to the International Space Station, biological matter has been almost constantly kept or cultured in off-Earth environments for maybe 40 years. From the initial efforts to understand the body's reactions to physical elements of space life, the violent launch and re-entry, microgravity, cosmic radiation, the focus of inquiry began to shift to include the biological effects of not being on Earth. So over the last 5 decades, space has become a laboratory. Through these experiments into aging, muscle wastage, gravitaxis, mating strategies, cell division, root development, and so on and so on and so on, we've begun to forge knowledge about the human organism, might react and adapt to a post-terrestrial future. We also begin to understand more about how terrestrial organisms might adapt to live on other planetary bodies, and what boundary states are conducive in general to life as we know it. And we've become pretty good at studying living organisms in a low Earth orbit. These procedures are excellent for finding out how organisms respond to microgravity and radiation in the near-Earth environment in low Earth orbit. But they tell us very little about how biology might emerge on or adapt according much more difficult. So it's generally more feasible with the current capabilities. It's much more feasible to try and recreate those conditions down here. And for that, we need to turn to simulation. The laboratory needs to become a simulator. So simulation is the imitation of the operation of a real-world process or system over time. The act of simulating requires that first we develop a model that represents the key characteristics or behaviors or functions of the selected physical or abstract system or process. And the model represents the system itself, whereas the simulation represents the operation of this system over time. There's the famous brain in a vat thought experiment in which an ex vivo brain is wired up to electrodes that create stimuli that mimic real sensations. It connects directly to the notion of the laboratory as a simulator. This brain in question has no way of knowing that it is not residing inside the cranium of a living organism and having a first-hand experience, but it acts in exactly the same way as if it was. The illusion is complete. So if I, for example, were to make a convincing set of conditions in the software of a computer of the surface of Mars and all the criteria, all the factors, all the data of the surface of Mars, and I transmitted this into the appropriate sensory centers of the ex vivo in vitro brain, the brain would be convinced that it is experiencing the surface of Mars firsthand. For all intents and purposes, it's there. In the same way, all organic matter in vitro will only grow because via the use of culture media and carefully modulated environmental conditions, a parallel of the real has been created. The inputs the organic material receives are the same as it would receive under natural circumstances, the data that makes it grow. And the term we use for the study of interpretation of the sign and signal worlds is semiotics. When this is applied to the perception of living organisms, we call it biosemiotics, a field of study which was developed in the 1960s by Theodor von Uexküll and Thomas Sebowick. Through this, we can basically understand that an organism, and we can extend this to include components of organisms, the organs, and we can extend this to include reacts in a fairly predictable way according to the inputs it receives from its sense environment. This is Jakob von Oetzkel. He was the father of 30 von Oetzkel. He was a German biologist, an animal behaviorist, a cyberneticist, working on relationships between an organism and its environment, leading him to develop a theory of Umwelt. For him, Umwelt was an environment world which consists of a series of elements which he calls the carriers of significance, and that these are the only things that interest the organisms in that environment. He illustrated Umwelt via a simple diagram called the functional circle, which illustrates how information received by an organism will in turn affect its own behavior in relation to the emitter of that information. Obviously, any given ecosystem will have a myriad of interconnected functional circles. Many ecologists used to assume that all organisms in the ecosystem share the same perceptual environment, but von Oetzkel realized that many organisms may have very different umwelts, even though they live in the same place. For example, a stalk of grass is perceived differently by an ant, a bacterium, a cow, a bird, or a human being. Each organism is fine-tuned to its own perceptual wavelengths. The ant will be considering maybe how safe it might be to climb to the top or where it might keep its aphids. Meanwhile, the cow will just be considering eating it. So fundamental to the practice of culturing cells and growing imaginal discs or whatever, or neurons, as well as a host of other ex vivo or in vitro laboratory protocols, is use of an incubator or a bioreactor and some form of culture or growth media. And it quickly becomes apparent that this conjunction of media and apparatus, an organism, is a form of simulator. The cells or bacteria or fungi or whatever are inside it will grow because of the sufficient simulation and replication of what can biosemiotically be perceived as habitual reality. The flux of nutrients, gas exchanges, temperatures, lighting conditions are within the ranges of what the organic material would expect and are what provide it with the signals and materials to grow. Equally, certain of these parameters can be altered or new ones added to provide experimental frameworks for growth under foreign conditions while maintaining the satisfactory illusion of authenticity. If we have data from other worlds, then we can recreate the essence of other worlds. Our presence and influence in space now spans here on for 60 years, since the launch of Sputnik in 1957. 70 years. No, 50. My math is too late to do maths. A long time. In that moment, the placement of a robotic entity in orbit suddenly and irreversibly caused our perceptual and sensory horizons to expand outwards. And this is an outwards expansion that's been going on ever since. Space is now teeming with robotic data gathering and processing platforms, a plethora of orbiters, landers, rovers, deep space probes, bristling with sensors, cameras, telescopes, communication systems, enable us to place our augmented senses and perceptions at distances of up to almost 20 light hours distant. On Earth, there's a complementary network of receivers, dishes, antennae, and information centers. We can locate our expanded sensory cortex within these machines and networks. Concurrently, the discovery of exoplanets is gaining pace, as we've heard this morning, and we're beginning to able to identify Earth-like planets orbiting other stars. The Voyager and Pioneer probes, Cassini-Huygens, and other missions have revealed to us a solar system full of exotic and tantalizing potential. We've seen that the smooth surface of Europa seems to conceal an internal ocean. We've seen that icy Enceladus spouts geysers. A smog-shrouded Titan harbors a liquid cycle, rich organic chemistry, oceans, and tectonic activity. We found ice volcanoes on Neptune's moon Triton and a Uranian moon, Miranda, with a geologically tortured surface and a cliff that is the highest known in the solar system. A few years ago, I began a series of projects called the Deep Data Project, in which I wanted to connect this expanded robotic, informatic sensory cortex with the model organisms that we use to find out about life in space. And in the context of the question, life as it could be, this project is a series of experiments looking at the evolutionary factors that could make known life become the something else that is necessary for non-terrestrial futures. Something that art can do, in my opinion, better than science is speculation. I think that art can do the experiments that science, or the way in which contemporary society demands that science is practiced, finds more difficult. But what is an experiment? An experiment is something that intervenes material in the world. It's a question posed with a certain expectation. It's an inquisitive demand made of a system to transform plasticity into precision. Designing experiments is a hugely creative act, demanding leaps of imagination and intuition. So looking at the specific features of experiments within the overall practice of science, there's one feature that stands out. In order to perform experiments, whether they are large scale or small scale, the experimenters have to intervene actively in a material world. And by doing so, they produce all kinds of new objects, substances, phenomena, and processes. And the beauty of an experiment presented as an art piece is that it is now able to bring in the metaphor and ambiguity that enrich the process of perception and allow for and stimulate new ways of thinking about a subjective inquiry. So the first of these pieces I made in the Deep Data project arc was Deep Data Prototype 1. I kind of realized I started to call all my projects prototypes because they never seemed finished. They never seemed the ideal thing. They seemed like a series of kind of snapshots of questions I was posing that I suddenly had to realize in some kind of tangible, material way. So as with all the deep data pieces, I started to kind of tap into the data produced by the PDS, the Planetary Data System, which is an archive of data products from NASA planetary missions. ESA has their own planetary science archive, which does the same for their missions. There's also Coho Web, which is a more specialized service that provides access to heliospheric, magnetic field, plasma, and spacecraft position data for various deep space probes. So by kind of harvesting all this data, I kind of wanted to match it with the model organisms that we use to find out about life in space. Model organisms, there's new ones added all the time: plants, fungi, microorganisms, mammals. And these are the organisms that the model organisms that they create our biological models, that we know how they work. Their genomes have been analyzed, their functioning has been understood. By making experiments on the model organisms, we get meaningful results back. We use model organisms extensively to test the nature of the human. We use model organisms as proxies for ourselves. Experiments we can't do on humans, we do them on model organisms, and we find out about ourselves. Model organisms, especially the smaller ones, the nematodes, the tardigrades, the fruit flies, these are the only organisms where generations have been born and died in space. And so in the deep data projects, I'm creating culture conditions, culture environments, custom-made culture simulators, really, bioreactors, mini simulators, in which I'm kind of cherry-picking various data sets from these deep space probes and applying them to the culture to see what happens. They're observational experiments. I never get to the point of taking empirical data out of these projects, which has angered a great many scientists that have come to see them. This is an observational process, and it's an experiential process. It's the posing of a question, not the reply to a question. This one, the Deep Data Prototype 1, I'm using the magnetic field data from the Pioneer and Voyager probes as they were swinging by the gas giants. You have these huge fluctuations in the magnetic field data from Jupiter and Saturn, Neptune, and Uranus. And there's tardigrades which are living in here. These are tardigrades. These are like everyone's favorite extremophile. There's even plush tardigrades now. Anyway, they're great. Yeah, tardigrades are the true survivors. These are the only organisms which, apart from the kind of undocumented artificial extremophiles we've created on the outside of spaceships, which may or may not exist, tardigrades are the only organisms which have been exposed to raw space and come back and survived. So I thought these were good candidates to expose to incredibly intense magnetic fields and to observe what happens to them. What happens to them is that they kind of seem to stop moving after a given period of time. They really aren't happy on Jupiter. This is just another kind of close-up of the mechanical side of it. So you have this, I think you see in the first picture, yeah, you have this kind of platform which has got the kind of the culture device, it's got the data receiver, and it's got a microscope camera there. So the projection screen is a kind of combination of the data that's coming in, what that data means, and then the visual effect of what it's having, kind of real-time observation of the animals, which are being experimented on. Then I made Deep Data Prototype 2, a little after. And Deep Data Prototype 2 is an experiment on a model organism plant called Arabidopsis. Arabidopsis is also grown extensively in space. It's taken to space, it's brought back from space to see how gravity affects root development, plant growth, etc., etc., etc. And this one we see there are 8 boxes. One is the Earth, one is the Moon, and then there's 3 of them are the planets Mars and Titan and Venus, which have been in science fiction, the 3 planets which could harbor life. But there's 2 for each, so we have kind of Mars and Titan and Venus, the way they were considered in science fiction. And what's happening is I'm kind of recreating the atmospheric lighting conditions, the kind of the chromatic spectrum of the atmosphere of these planets, as it was imagined in science fiction, and then the true quality as measured by the spacecraft which visited or orbited these planets, these places. Because what I'm using here, I'm using a photomorphogenic mutant of the Arabidopsis plant. The good thing about model organisms, they have so many lab-controlled mutants which are really kind of prescriptive what happens to them. You know, we kind of know a lot's going to happen to them, what kind of data we put into them, we know what we're going to get back. So the photomorphogenic mutants, depending on the wavelengths of light they're receiving in certain growth stages, they will grow to different heights, different lengths of roots, different lengths of siege, different shaped leads, etc., etc. Huh? Really? I'm not even halfway through. Wow. So is that really 20 minutes? Joe, do you mind sitting out and I'll just... So I'm growing sculptures with these plants. Now I'm going to talk about the most complex project now in 2 minutes. So our Deep Data Prototype 3, I'm using the gravity wells of newly discovered Earth-like exoplanets to experiment on nematodes, which are these. The only creatures to survive the 1986 Challenger disaster, and so really geared towards huge impacts and huge changes in gravity. A few years ago, I developed an obsession with Titan. Titan is the largest moon of Saturn. It's what's considered the most Earth-like place that we know of. Despite the discoveries of exoplanets, Titan is the most Earth-like place that we know. It has an atmosphere, it has a liquid cycle, it has tectonic activity, it has lakes, there's evidence that there's maybe a subsurface ocean, and it's a very rich candidate for biotic. There's organic chemistry going on in the atmosphere. It's really amazing, amazing place. We know a lot about it because the Huygens probe landed there in 2005. What Titan looks like on the surface. So I made this project called Drosophila titanus. How long? Let's go. This is really the project where I wanted to create something which was as close to a scientific experiment as I possibly could. But what I'm trying to do is I was trying to breed a new species of Drosophila, the fruit fly, that would be theoretically capable of living on Titan. So but the only way to do it, the only way for this to make sense, not to be a waste of time, is to follow an incredibly rigorous scientific process based on scientific foundations. But obviously, it's ridiculous. There's no way I can get them to Titan, and there's no oxygen on Titan. It's 180 degrees below freezing, so they're not going to survive there. But it's like by following this process, by the science and the art stuck together, there comes a point where they have to pull apart. They have to, because science has to do this and art has to do this. And it's like splitting the atom, you know? When you pull the science and the art apart, something really important, something really meaningful, something really exciting is happening there. Something which really affects our cultural perceptions of knowledge and new knowledge and how we assimilate that into cultural processes. So, I kind of created all sorts of different kind of it talks about eugenics and this idea of the right stuff about who gets to go to space and why, how are you good enough to go to space, and this idea of kind of biological perfection to fulfill a certain need, which a lot of the synthetic, I wish I had more time to talk about how this ties into synthetic biology and things, but I don't. But this is kind of artificial selection, it's forced evolution, which is also what's produced things like this. You know, we've created these monsters for aesthetic, economic, commercial, industrial, scientific gain, things which would never have emerged through purely natural means. So I created all these kind of nutrition experiments, temperature experiments, light experiments to kind of model as many of the different elements of Titan that we know about, to train these flies, to get them closer and closer to be able to live under Titan conditions. Getting them to, they're normally attracted to blue light. I wanted to see how many would then start to choose the orange light, you know, so you do a period of acclimatization, you do an experiment, you select the ones which match Titan conditions and you breed from those. So, you know, bit by bit, I began to think I'm changing the genome with these things, their behaviors. I messed around with their perception of gravity. It's installed as a timeline. This summer, the project, I had a catastrophic population crash, and my many thousands of carefully bred over 7 years just off in it all died. So I thought I had about another 200 years of work to do this, and now it's finished. Like this talk, it's finished. So this is like which is really good because I was really struggling how to make a conclusion about how to these death of the space pioneers, you know, the whole idea of death and risk and space pioneers and future life. So thank you for that. That's okay, very nice to meet you.
All right, we have a little moment of technical switchery now to change out the computers. So I'm going to take advantage of that since our Q&A is going to be cut short as well. Oh, to announce something, which is once we get to the reception part today, there is another special treat that Luis wanted me to tell you about, and he gave me a little thing to say, which is on my phone, which is why I'm looking it up. All right, here we go. So during the final reception, there will also be a screening of a 23-minute film about vegetables, just next door to my left here. And Luis was very careful to point out that it's important to not give too much away, but it is a remarkable film. So I'm reading Luis's description. An avant-garde film by Berlin-based filmmaker Adi Gelbert, whose birthday it is today too. Berman plays with deeply rooted astrobiological concepts to offer a surreal vision of life on Earth at a moment of cosmic contact, weaving together historical ideas of panspermia, the directed seeding of life in the universe, the early modern conception of the European conception of the universe as having multiple layers, like an onion, narratives of biological evolution, interplanetary relations between species, and the construction of organic machines made of vegetables. This experimental pseudo-documentary builds in a slow arc of gradually increasing tension in the style of cinema verite until an increasing crescendo of the storyline and aesthetic effects reaches a surprising end. No spoilers, you just have to watch it. I think you're going to want to. So stick around after for the reception and the screening. It's not every day that you get to see a film about cosmic vegetables. And with that, it looks like we are nearly ready to go here. So I am going to introduce our final, well, you can futz about while I introduce you, Joe Davis, who, as he correctly corrected me, actually is from the laboratory, not the studio, the laboratory of George Church at Harvard, and where he is artist-scientist.
And also, he's a researcher in the laboratory of Thomas Schwartz at MIT.
Using up my time.
I can't do anything right. Go.
So NASA has already realized some ideas about horticulture in space. Last year, astronaut Scott Kelly explored special problems associated with microgravity gardening aboard the International Space Station. But there's another kind of garden. Instead, let's imagine a garden on a cold, bleak ice world with briny subsurface oceans of water and ammonia, or a hot, outgassing cornucopia where planetary crust is cooled just enough to allow the formation of rocks and crustal plates. This kind of garden first appeared on Earth about 4.5 billion years ago. As far as we know, it was in the early Archean eon, roughly 3.5 billion years ago, that life left its first detectable impressions. The garden was anoxic. For the most part of the history of the life, the garden had a very different atmosphere from the one we know today. Like Mars, there was no oxygen. Rather, it consisted of carbon dioxide, methane, ammonia, sulfur dioxide, hydrogen, nitrogen, and would be toxic to air breathers on our planet today. Yet, the organism, the first one, is thought to have evolved in such a reducing atmosphere. Recent studies suggest that the late Archean Earth atmospheric pressure was 0.5 bar, more like what conditions are thought to have been at the time on our neighboring planet. 3.7 billion years ago, Mars was a wet planet with a much thicker atmosphere. But until roughly 0.85 billion years ago, oxygen was probably only 1% to 2% of its current level. That means for the most of Earth's history, oxygen comprised only 0.21 to 0.42% atmosphere. With oxygen levels at less than 0.5% atmosphere, we would have suffocated. But even 0.21 oxygenation was enough to precipitate perhaps the greatest biological disaster ever to unfold. One of the extinctions our performers neglected to mention, called the Great Oxygenation Event, about 2.8 billion years ago, when Earth probably lost the largest part of its primordial biome, and it wasn't just an ice age. Atmospheric oxygen peaked at around 35% in the Cretaceous, only to reach its current level after the KT extinction event 65 million years ago. So I've had a fascination with fossils long term. I have a big personal collection, so why would an artist cultivate such interests? It's because they possess special qualities that can transport you into lost worlds. They are teleporters for the imagination, windows into whole lost kingdoms of life, and for just this reason, there is also something terribly plaintive and poignant about the fossil record. These are relative great bottlenecks in the history of life. In the Ordovician extinction, only 12% of all living things survived. In the Devonian, 75% of the survivors went extinct. In the Permian Triassic, only 4% survived, and 75% of those went extinct at the Cretaceous Tertiary events. While scholars lament the loss of great libraries, say Alexandria, the libraries at Nalanda in northern India, the knowledge lost forever with the reconquest of Al-Andalus, and all the fanatic interdictions of art, science, and philosophy that have marked the passage of human history, but all of these pale by comparison with the unspeakable biological tragedies of the mass extinctions. We can only imagine what fields like synthetic biology could accomplish with these lost genes if even a tiny percent of them were ever recovered. But what we're left with is a toolbox that's mostly empty. Today, most organisms live in saline environments, in oceans and inland seas, both on continental shelves and in the benthic deeps, in deep ocean vents that actually comprise a large part of the overall undersea landscape, and in anoxic hypersaline basins. in marine solar salt urns, in terminal hypersaline environments where evaporating seawater ponds, lagoons, and desiccated salt pans host an amazing diversity of life. In perennial salt and alkaline soda lakes, and there are many, many of these, the Great Salt Lake and, I wish I could see. Yeah, the Great Salt Lake, but they're in Australia and Tanzania and the Western United States and Mongolia and Turkmenistan and Bolivia and China and Turkey and Kenya and Iran. They're everywhere. Human beings and other air-breathing, freshwater-dependent organisms are by far the latecomers, and in fact, there may be cause to wonder who are the true extremophiles. Salt-loving organisms, on the other hand, are called halophiles, and they are extremely diverse and live in many extreme environments that most other organisms, including human beings, are unable to tolerate. Halophilic xerophytes persist in environments characterized by extreme aridity, while halophilic thermophiles endure extreme temperatures and routine exposure to ionizing radiation. There are hundreds of hypersaline subglacial lakes, Antarctic lakes, and perennial liquid surface lakes where temperatures nevertheless range from 11.5 degrees C to minus 20 C, which we never think about as an incubator. Some halophiles are known to survive in high vacuum radiation and temperatures of the space environment, including experiments on NASA's Long Duration Exposure Facility, which went from April 1984 to January 1990. ESA experiments included BioPan and UTEF, the European Technology Exposure Facility, attached to the Columbus module of the International Space Station from February 2008 to September 2009. My several experiments in the richly poetic field that is called Search for Extraterrestrial Intelligence revealed several fundamental problems with SETI radio transmissions. Such transmissions are limited to wavelengths that transmit through Earth atmosphere. They're constrained by the need to overcome solar background. They can only be sent to relatively few target stars. Photons diverge. The inverse square law, an object twice the distance from the source will receive a quarter amount of the illumination. That means you have to be the center of the beam in order to detect it, and even the speed of light is too slow over astronomical distances. Homo sapiens appeared roughly 100,000 years ago. Homo sapiens sapiens, approximate time of divergence from the common ancestor of all modern human populations, was about 200,000 years ago, enough time perhaps for a single light speed exchange of information across our own local galaxy. Meanwhile, mitochondrial Eve evolves into a different species. You have to account for the proper motion of stars, something that Frank Drake neglected to do in his famous. Okay, so in the long run, all of these shortcomings fall into the context of three basic problems. You have to have something that's robust enough to survive the space environment. You have to have something that can reach out across geologic time, and you have to have something that's abundant enough to reach hundreds of billions of targets. And it seems that bacteria, especially sporulating bacteria, answer these problems very well. My colleague Dana Boyd and I figured out that we could fit enough bacterial spores or even bacterial cells in a pint of Guinness, that's a metric pint, 500 milliliters, so that there would be 1 million spores per solar system in the Milky Way. That's not a lot, but it's someplace to start. Biological carriers can outperform radar. They can persist for geologic time. They're space compatible, vacuum, radiation, and temperature, and easily produced in hundreds of billions of copies. My 1986 MicroVenus project explored the notion that biology could play an important role in sending astronomical numbers of directed messages across hundreds of thousands of light years in corresponding enormous periods of time. These are space toilets. We've spent millions of dollars, literally, trying to build these things, and they never work. This is ISS dumping its toilet visible from the ground. I put together a project in 1997 called Fishing the Norton Rings to investigate this phenomenon. Gemini flushed its toilet in space, Apollo flushed its toilet in space, and left human waste on the moon. Skylab flushed its toilet in space with 3 astronauts in each of the 3 Skylab missions. They would have discharged 465.8 kilograms of wastewater from feces discharges only. Space shuttle's wastewater dumps usually consisted of 165 pounds, which is the capacity for all wastewater tanks. As of February 1994, 5,385 kilograms were discharged into low Earth orbit. 7 additional missions flown between 1996 and 2011 included the longest space shuttle flights. Total discharges from space shuttle probably equaled about 10,000 kilograms of human waste. It's possible that the Russian presence in space also resulted in discharge of up to 20,000 kilograms of human sanitary waste. What's happening? Go. This computer stalled. Damn it, I'm gonna have to reboot. ISS and Mir, both. I did already.
Yeah.
It's back.
I am tech support for Earth.
Here we go. ISS and Mir also discharged. So what happens when you discharge liquid water into 10 to the minus 5th torr vacuum at near Earth orbit? You have an explosive discharge. Everything expands supersonically in every direction, and the expansion causes the liquid to freeze into particles of ice that are 1 micron to 1 millimeter in diameter, and it's dirty ice that protects it. And according to NASA's own calculations, they occupy every conceivable orbit within 33 months. This is pictures of pee-pee and caca on Space Shuttle's windshield. I went to Johnson Space Flight Center in Houston a few years back and obtained samples of the space return parts from the Solar Max satellite, and sure enough, there were about 1 million impacts per square meter from 1 micron to 1 millimeter in diameter, which we grew Enterobacters from Dana Boyd and I, and this is electron photography of some of these impacts. So, humanity has systematically contaminated extraterrestrial environments with terrestrial microbes for more than 50 years. Tragically, wastewater dumps in space and completely sterilized atmospheric probes, lunar and planetary landers have collectively ensured the distribution of terrestrial organisms into other planetary environments within the time span of our own generation. There is simply no way to compensate for a process that has been underway for decades and perpetuated on astronomical scale. Likewise, few tools are available to predict the implications of such interplanetary contamination. And we just heard about how the nematodes survived the Columbia, so I'm going to skip that. One of the problems to survive for a bacterium to survive in space is radiation from mysterious interstellar sources and from the sun. One insidious result of radiation is called molecular cross-linking, where radiation energizes an atom enough to break a chemical bond and then form abnormal bonds with other atoms. Such free radicals have been implicated in a wide range of cancers and genetic mutations. In terms of DNA damage, ionizing radiation and particle radiation each leave distinct signatures. Genes for radiation resistance are needed for planetary exploration. We will need factories in space to support exploration of the moon, Mars, Jovian worlds, and other planetary bodies, and these factories will almost certainly be biological to synthesize pharmaceuticals, fuel, industrial chemicals. And microbes are storable, dormancy with little or no energy input. Precise mineral extraction can be obtained and deposited, and atomic scale precision assembly. To grow regular plants, terrestrial plants on Mars, they'll have to be heavily shielded from radiation, and if they're grown underground, it's going to be energy intensive for artificial light. So looking for genes for radiation resistance, I have conducted now several experiments at the MIT Nuclear Reactor Laboratory, which is kind of a steampunk laboratory. It was built in 1958. This is the young Homer Simpson. This is our reactor core and a diagram of the circulation of its primary coolants. Biologist Stefan Wolfel and I, back in 1994, when the reactor was actually hotter than it is today, there were fuel particles in the primary coolant. We were using radioisotopes in lab to sequence DNA, and we would have in the reactor shell 4 hours was our annual dose. So we were racing in and out. We produced a typical microbiological survey of array of media, and then inoculated with primary coolant, and lo and behold, we got growth. We got a strange one, a cold water loving organism called a chromobacter, and numerous bacillus species. These results were reported initially in Süddeutsche Zeitung in 1994. Colonies appeared in media ordinarily prepared for micrococcus and a nutrient preparation commonly used for the culture of yeast, but we got neither yeast nor micrococcus. So we didn't have metagenomic tools back then. We only had microbiological tools, so we revisited this experiment starting last year. This is our new team. And oh my God, I'm getting ahead of myself. Yes, we have metagenomic tools. Two of our Harvard grad students produced our first vacuum-assisted filtration system. We also have access to these rabbits, little pneumatic tubes that we can send through the reactor, 10 to the 13th neutrons per square centimeter per second. We get to test whether the urban legend of Deinococcus radiodurans is true or not. We can send in the most radiation-resistant organism known, this one, and there's been some reports that a few genetic modifications to E. coli will render them radiation resistant. We will see. So, in Adrian Kish, member of the research group studying halobacterium, a halophilic archaea at the University of Maryland, says, exposed their halophiles to gamma radiation, we have completely fragmented their DNA, quote, and I mean, we have completely destroyed it by bombarding it with radiation, and they can reassemble their entire genome and put it back into working order within several hours. So we're also, because gamma rays aren't quantifiable in the reactor core, we're also utilizing cobalt and cesium irradiators at MIT. We've also expanded the inventory of organisms we want to expose, including cockroaches, because I want to put the Nuclear Non-Proliferation Treaty in a species of cockroach. We're also exposing axolotls to thermal beam ports because they're rumored to be able to regenerate after exposure. But then, in the axolotl colonies I've been involved with, I noticed that there were cancerous growths on the axolotls, and they're supposed to never get cancer. I mean, that was an urban myth. Here we have several. And so I took it upon myself to start a Facebook page to demystify salamander cancer. It turns out that they aren't 10,000 times less, though. Many laboratories responded from all over the world. One problem solved. For free. We're doing RNA-seq for transcriptomics and MiSeq and next-generation whole genome sequencing. We're interested in where the organisms are coming from, sampling at the reactor coolant input. We sampled both non-sterile and sterile connections, sterilizing with bleach and alcohol. All experimenters underwent MIT nuclear reactor laboratory radiation safety training. So here we have active filtration with some expensive filters. Our first samples. This is the hood at the reactor's primary coolant output. It's very hot. And this is our, see, I can't play, some of these are actually movies, but I can't show you because they're not loaded, I don't think.
Well, time as well.
Okay, I'm almost done. We did inoculate media as before, and we got growth in all of the media. These tools are useful in other, how much time?
4 minutes.
4 minutes is great. Salt. Salt usually crystallizes in brine, and as it crystallizes, it expands and contracts at different rates than the brine that it's situated in, and so it includes these fluid inclusions in the salt, and these have been found in Devonian salts and anyway, yes, in very old salts, and it's a typical tool used by geologists to determine at what temperature the crystals formed, which informs paleoceanography, paleoclimatology. Then in the 1960s, biologists claimed to grow organisms from these inclusions, and these reports were greeted with skepticism, but now we have metagenomic tools, and indeed, we're bringing back these organisms. So, astronomers routinely peer hundreds of thousands of years into the past, but unlike astronomical objects, these are much closer at hand, much closer than we ever expected. They're all over the world. I'm going to do a paleological expedition, Paleozoic. Anyway, I'm running out of time. Your salt, your table salt is Permian salt, by the way. We started icing roads in 1940. I was brought to this because I thought I could artificially flavor salts and make them taste like fancy French sea salts. And so I gathered up all these salts and I externally sterilized them. I found only reports but none were externally sterilized. I used ethylene oxide, a nasty poison gas, which you break this glass vial, then you close the door and run away. And we prepared hypersaline media, incubated, lo and behold, we got growth. And some of the growth we got was from salts that were 250 million years old. Wait a minute, back up. Went to get more, got some growth. Went deep into a mine, deepest North American salt mine near Cornell, New York, 2,300 feet deep, obtained samples, went into the Berchtesgaden salt mines in Germany and in several others. Here we have salts from Heilbronn, Germany, associated with the Muschelkalk in northwest Switzerland and Armenia. And we have now new samples from the Triassic from Thuringen. And so these are samples, samples, electron microscopy, finding pores in the salts where they hide, and control. We're not only externally sterilizing with ethylene oxide, we're flame polishing sequentially at 30 seconds, 60 seconds, and 120 seconds before inoculating, operating in the sterile hood, checking external sterilization with ETO-bearing organisms, autoclaving 50% of the samples to kill everything, new batches, inoculation, we got growth.
Final minute.
DNA data. This is the organisms we found. We're looking for genes for perchlorate metabolism, which we'll need for Mars. Yes, and there are salts coming from space. These are fluid inclusions in meteorites called Zag and Monahans that are 4.7 billion years old. That's older than the Earth. I mean, that's older than the Earth. And I want to get my hands on these. NASA has them. This is the primordial nebula from before the formation of the solar system. If we find signs of life there, I'm sorry, the Pope will have to get a new job. The whole history of art reflects the quest for control over the qualities of vitality and function that distinguish life and death. There are all these stories about the transformation of one kind of living thing into another kind of living thing. The dream of science and art is a universe full of life, and the logical continuation of these long-standing aspirations. My dream is for a beautiful garden, perhaps the most ambitious landscape architecture project ever imagined, to create a single cell that has all the machinery it needs to transform an entire planet. Thank you very much.
Woo! Do we have time for questions, Luis? Because there must be so many questions.
Nice job.
Okay. Raise your hands if you would like to jump in with a question. If not, I'm going to go first with a question, which is, I feel like you guys are sort of inhabiting maybe a boundary condition for artists yourself, you know, or designers, sort of embedding in science labs, hanging out at synthetic biology conferences, you know, doing these sort of slightly off. Can I help?
Nope.
Okay.
Excuse me.
So I'm curious as to how you think of your audience and responses and responses to your work. Daisy, I know you were sort of talking about, well, this can be a way to introduce much needed critique. Is it a way to get science to think differently, the way to get the public to think differently? Who are you talking to, and what conversation do you want to have? Who wants to go first?
I'll answer quickly, is that for me it's all of those things, and myself as well. So the projects for me are opening up problems and encountering problems along the way. And then my audience, it's amazing to then take it back into synthetic biology and then challenge and things develop and get assimilated problematically as well. But it's all of them, I think. I'll jump in. My initial inspiration to use science in my work is because my family is fundamentalist Christian and anti-science and climate change denying, and I felt very passionately about climate change, and I wanted to work on public misperceptions of science. And in fact, when I approach science, I see a playground of juicy and fun ideas, and I can't wait to explore and learn. And so that was my initial dive into it. But yeah, it's also super fun. But I have, as I've gone further and further, there are moments where I'm like, oh, wait, science thinks this? Why are, you know, in terms of like evolution, why is there so much focus on, say, survival of the fittest or survival of the fit, when in fact cooperation also seems to drive evolution? And so things like this have started coming up where I'm now starting to be in the conversation enough that I can start being critical and asking science, wait, why is this the mainstream paradigm? Why can we have a fuller conversation?
Who's my audience? Who's it aimed at?
Yeah, are you talking to scientists? Are you talking to...
I'm talking to as many people as I can possibly talk to. And I think something that we all have probably felt frustration with part of my activities as an artist is to produce objects that go in exhibition formats, frameworks. And there's always this frustration that the only people that are seeing it are people that are already interested or already have ideas, or the other artists or things. And so, I mean, that's an important part of the work. But, this is another important part of the work, because then you get to actually speak to scientists. I'm much more interested in speaking to scientists than speaking to artists. I'm also really interested in speaking to the general public, whether they're informed public or not informed public. So there is no target audience. I mean, the target is trying to make in whatever way a contribution to understanding, or a contribution to curiosity, to not just think about what the science is trying to find out, but why we're trying to find out, and with what means, and with what level of openness and accessibility. So I'm just always trying to strive to that point, you know, to try and get the most democratic and simultaneously stimulating way of entering into these questions.
I usually don't worry about it. I mean, I just try to make dreams come true. That's what you do, and usually, like, somebody's going to inevitably say that's not art, or somebody's going to inevitably say that's not science. It's science, so you just let those people argue. In my opinion, art has to describe the whole world, like physics has to describe the whole world, and you can't describe something that you're clueless about. We have to, we have a kind of built-in obligation to be generalists, to be as rigorous about what we do as everybody else is about what they do, and if somebody's going to say that's not art, who cares?
And is your garden going to be a salt-based one?
Is it what?
Is your garden going to be a salt-based one?
Well, yes. With the garden that I'm building now, yeah, is a halophilic garden.
Sorry, I just wanted to. Anyone?
So, Banu, did you want to jump in on this? Yeah, I think from my experience, the biggest audience that I enjoy sharing this information with is children, because for them, sky's the limit. And as soon as you see that they have a passion for what you're bringing to them, then you already know you set the seed in, and God knows what's going to happen afterwards. They're just going to go, they're kids, so they don't know there's any bound. Just like you said, you want to see dreams come true. This is your job, is to set a seed inside of someone's mind, and then
I do a lot of presentations.
Yeah, because we don't know. Yeah, we don't know what's going to happen. Right.
Teach them about DNA structure and the operation of the genetic code, just to shame artists into learning.
Yeah. And the biggest thing is that I've seen is just the fearlessness. Not to take up too much time, but the fearlessness that a child has. It's kind of like a kitten. They don't know that there's a boundary here or a boundary there. They don't give a crap. They just keep going. So that's why I like teaching babies. So.
Do we have any questions? Yes.
I have a related question that you fed right into, which is, what are your most receptive audiences? And the first thing that came to mind would be that they would be kids, because there is no judgment on. They're just hungry for knowledge, and they don't necessarily have any preconceived notions, or they don't feel they're in one camp or the other, and so it's, it doesn't matter if it's not art or not science. It's what it is, and so aside from them, children, scientist or your lay person, who finds the work that you all do most accessible, or do you connect with easiest?
Well, I feel in my work, because part of my inspiration is about trying to bring down the barriers of, perceptions. Even if I'm talking about something that's really complex, if a layperson doesn't understand it, then I failed. Because we have at our command storytelling, humor, metaphor. I mean, we have so many things at our command. Yep. And in my solo shows, I use a lot of video. But ultimately, the audience, I'm telling stories. We're theater artists first. So we're telling stories that can appeal across the range. And so, for example, one of my audiences is actually LGBT. The joke I always tell is my family is far-right Republican, fundamentalist Christian, and I'm a lesbian science nerd. And so I've done some touring to colleges and stuff. LGBT clubs will bring me in, which I love, because that's what I really want to do. Let's break down these two cultures, and let's have conversations on campuses that are not, just stuck in our two towers. Yeah.
I think kind of receptive is, I kind of mentioned briefly before that there's this frustration when you're showing art in an art context. Maybe the other people in the gallery who regularly frequent galleries, maybe the most receptive. Hey, I love your work. That's really interesting. That's really nice. But that doesn't give us anything back as an artist. I want to come into context with people that are going to challenge you, or go, no, I don't think that's right. I don't like what you're doing there. Science has proved that that's not true. Or even if people come and tell me, actually, I'm a creationist and there's no evolution. I would much prefer to have that as a reaction, so there can be a dialogue, so there can be a continuation from this kind of static object, which is an art project, an art process. That we can take that then into a new phase, which is some kind of engagement, some kind of exchange, some kind of continuing life.
We're sitting in an audience that's presumably highly literate, scientifically, artistically.
Probably.
But and so they're basically understanding what it is that we're saying. Mm-hmm. But a lot of times I open my mouth and nobody understands a damn thing I say. Mm-hmm. It's like this basic literacy is rarer than we'd like to imagine. I wish we could teach curiosity. I wish we could.
But I think you do a really good job. Now, forgive me, I missed the very beginning, but when I walked in, you were going through all the different kinds of life with your two different visuals, and you're really so funny. I mean, we were in stitches, and I know you probably didn't hear because we were all laughing very quietly, but you are fantastic. I think that a lot of what you're doing is very accessible.
Well, and I think, and unfortunately, we probably have to wrap up, but what I think is exciting is also you guys demonstrating that we all have the tools to engage in imagining life as it could be. It isn't actually just the folks at Ginkgo who can imagine that, or just the folks who understand the difference between xenobiology and exobiology. We actually all have some tools to participate in that, which I appreciate. But thank you very much, guys. And I apologize that we had to cut the conversation short.
So we've reached the end of a very long day thinking about life as it could be. I have just a few final thoughts for us, and Jennifer will read a final passage for us, and then I'll finish up the day after that. Even though we must admit ourselves to be lineal descendants of some rather nauseating gases and sundry streaks of lightning, Harlow Shapley once said, it's gratifying to be part of this magnificent evolutionary show. So I'd like to thank all of our speakers again. I'll turn to Jennifer now, who has a passage for us, a brief thought from the poet Rainer Maria Rilke.
Be patient toward all that is unsolved in your heart, and try to love the questions themselves, like locked rooms and like books that are written in a foreign tongue. Do not now seek the answers which cannot be given you because you would not be able to live them. And the point is to live everything. Live the questions now. Perhaps you will then gradually, without noticing it, live along some distant day into the answer.
Thank you. Thank you, Jennifer. And now as we turn from astronautics to gastronautics, we conclude by fulfilling the very meaning of the word symposium, symposion, drinking together, and enjoying, as Stefan once put it, some heavenly cheese and cosmic company. So thank you all for coming, and farewell, and may we meet someday on the tundras of Mars.